Question is, am I a male or a female? So this is not a trick question. It's a simple question. If you think I'm a male, raise your hand. It's up to me or up to my wife. I don't know. So if you think I'm a male, raise your hand. Phenotype, you think I'm a male, right? So, but if I tell you today, my 6 chromosome composition is XX, do you still think I'm a male? Raise your hand. Oh, so if I'm an XX, I still look like a man, you think I'm a female. That's interesting. Third question is, if I look like this, and my 6 chromosome still XY, but I do have a pair of ovaries, am I a male or a female? If you think I'm a male, raise your hand. No female, raise your hand. Or something in between, right? So this is a really interesting question. It's not black and white. It's something in between. So this is exactly what we're studying. And this particular disorder, called disorder of sex development, fortunately, I'm a normal XY and I have a pair of testes. My wife can prove that, and I have 2 daughters. But unfortunately, 1 in 250 to 300 newborns suffer from this disorder, meaning their phenotypic sex is not consistent with their chromosomal sex or their gonadal sex. So this is a particular disorder that includes pretorchidism, meaning undescended testicle, hypospadia, incomplete closure of the urethra along the shaft of the penis, and intersex situation I just mentioned to you, the inconsistent or incompatibility between your phenotypic sex and the chromosomal sex or the gonadal sex. So this particular disorder, they won't kill you, as you could imagine. It's not to the degree that we have to pay attention to rescue the patient when they were born. But you could imagine the psychological and the health impact of the individual and to their family is tremendous, right? So my lab, we use mouse as a model system because it's really not feasible and ethical to use human embryo to do this kind of basic research to find out the mechanism for sex development. So we have to come up with a model. I mean, mouse is the closest you can get in terms of you can do genetic manipulation. But as you will learn, fish and many other species are also very important to study this particular disorder. So my lab, we have three specific directions. One particular direction, we look at how the gonads are formed in the embryos, and also how the reproductive tract, the oviduct, the efferent duct, and the sexual dimorphic ductal system are established. And also, most recently, we start to look at how the external genitalia are established between male and female embryos. And today, I'm going to tell you a story about how the gonads are formed during embryogenesis and have a discussion in terms of how we can enhance the study by developing new technology to understand the process. So the field of sex determination has a dramatic or revolutionary changes in the early 1991. How many of you actually read this paper? The SRY gene published in Nature in 1991. That's actually one of the paper I read when I get into the graduate school. So that paper described this is an XX mouse, but the reason why the picture showing here is to show the external genitalia. The external genitalia develop as a male, and internally this XX mouse also develop testicle. Reason being, this is a transgenic XX animal that carry the gene called XRY, or sex-determining region of the Y chromosome. And now we know SRY is the only gene that's necessary and essential for the determination of the gonad. So it's not the genetic background of the sex chromosome background. XX versus XY doesn't matter. As long as you have the SRY gene, you can put it in the XX background. That individual will develop testicle. So this tells us SRY serve as a molecular and genetic switch to transform the bipotential supporting cell, as Dr. Zhong mentioned to us yesterday. So both male and female gonads, they come from a single primordium. So this primordium, as long as there's an SRY gene in that primordium will differentiate into the Sertoli cell lineage. On the other hand, in the absence of the SRY, the primordial bipotential cell will differentiate into the granulosa cell lineage. So you can see this is an amazing biological event because usually you don't get black and white, but this is really a black and white event. One gene can turn on the program to make a completely different organs. So this is what we know. And the most importantly, this process is hormone independent, right? Today we're talking about the comparative endocrinologists. But this process is completely hormone independent. It's controlled by the presence or absence of SRY. So they call this cell autonomous lineage specification. SRY is a transcription factor. If you have that transcription factor, open the chromatin, stimulate gene expression, and turn on a cascade of events leads to the differentiation of the particular cell lineage. So this is so-called the cell autonomous event. Doesn't require any hormone. However, there was this very interesting case. I hope that many of you have heard about this. The free martins. Anybody know something about the free martin? Raise your hand. Because I have seen a generation gap. If I go to a class, I ask, have you anybody heard about free martin? You can tell anybody that is younger than 20 years old, they have no idea what free martins are. So I kind of know the age distribution right here. So free martin was the first thing I learned in reproductive biology 101. Right? This is a basic, fundamental, interesting phenomenon. What free martin is that particularly in cow and those farm animals. In fact, this particular interesting case was documented more than 2,000 years ago by farmers and philosophers. So they found that in particular in the cow, when the female cow carried both XY and the XX embryos, so this is basically carrying two different sexes of the twins. So in most cases, the XX twins are masculinized. So they develop male reproductive system, particularly the ovaries is sex reversed into testicular-like structure. But you probably will think, Humphrey, you mentioned to us, formation of the testes require the presence of a cell autonomous gene SRY. But in this case, this female embryo, XX embryo, doesn't have the SRY gene. So how does it develop the testis structure? Doesn't make sense, right? The cell autonomous hypothesis doesn't make sense here. The reason being is they found that later on, to have the free margin scenarios to happen, the XY and XX embryo must share a placenta. If they don't share placenta, free margin case doesn't happen. So if that's the case, it suggests that there are hormone factors that coming from the XXXY embryos that travel through the blood circulation, eventually act upon the fetal ovary and somehow change the sex of the fetal ovary. So this actually phenomenon or hypothesis is contradictory to the 1990 observation of the SRY, which suggesting it's a cell autonomous hormone independent event. So that really opened up the venue that, in fact, mammalian system, particularly in this particular case, sex of the gonad could be susceptible to hormone, very much like birds, fish, and other species in this case. So we start to ask, are there truly hormone-like factors that coming from the fetal testis that actually travel through the blood circulation that can masculinize the fetal ovary? So that was the first hypothesis that we proposed. So we start to look for the potential hormones that coming from the fetal Sertoli, right? If you think that the fetal XY embryo can masculinize the XX embryos, the factors is probably coming from the fetal testis, and it's probably coming from the Sertoli cell. That is the only cell type in the body that express SRY. So we start to look for hormones. The first hormone, I'm sure everybody should know, the anti-Mullerian hormone, is one of the first hormone identified that is produced by the fetal Sertoli cell. And the function of the hormone is to cause regression of the Mullerian duct, which is the female reproductive tract. Another hormone we identify, activin. I'm pretty sure the endocrinologists should recognize activin is a very important hormone. It serves important function in adult situation, and in this case, we actually found this hormone is produced by the fetal Sertoli cell. So if these two hormones are the so-called free margin factors, so they should be able to masculinize the fetal ovary, and at the same time, they should be also very important for the fetal testis. So these are the two characteristics that need to be met to be the free margin factors. So then you probably will ask, if this two factor or either one of them are absent in the XY embryos, we should start seeing the defects in terms of the testis formation, right? So our hypothesis is this: the AMH and activin B are the putative free margin factors, and to be qualified to be the free margin factors, these two factors must be important for testis development, and they are also should be capable of masculinizing the fetal ovary. So fortunately, the knockout model for these two hormones has been made already, a single knockout situation. So we look at the single knockout first. It was very disappointing. Single knockouts of AMH and the activin B had no impact on fetal testis development. The testis develop normally, everything seems to be fine. But if you recognize AMH and activin B, they all belong to the TGF-beta family protein, the transporter. transforming growth factor beta 50. So they signal to similar but not identical pathways. So it's very possible these two factors work redundantly together to facilitate normal fetal testis development. So we decided to make the double knockout and to look at the fetal testis, whether there are defects in terms of their sex testis formation. So what we are seeing here is first we conduct the total RNA-seq between the control testis and the double knockout testis. Here is the x-axis is the fold change of gene, and the y-axis is the p-value. Right? Upregulated gene on the left is the downregulated gene. So we first look at the fate determining phase, and the mouse is E12.5. So there's nothing wrong here. You didn't see any dot. Your eyes are okay. Indeed, there's nothing wrong with this animal. At this stage, the fate determination phase, the control tests and the double knockout tests look identical. Morphologically, in the transcription, my postdoc was very disappointed. It was a very heartbreaking stage because we didn't see anything wrong. That really tells us probably our hypothesis is not correct. But then I told my postdoc, maybe we should look a little later. So we look at about 3 days later, that is so-called the fate maintenance phase for the tests, meaning all the cell types need to maintain their identity during that period of time. So what you are seeing here is dramatic transcriptome changes between the control and the double knockout. See a lot of gene being upregulated and a lot of gene downregulated. So this suggests to us that the AMH and activin B function together, and their function is not for the fate determination. They are in turn important for fate maintenance of the testis development. So then we ask, transcriptionally, they look very different, but what is morphologically at the fate maintenance phase? So what you're seeing here is a cross-section of the fetal testis that we stain two markers. SOCCE9 is for Sertoli cell, FOXL2 is for granulosa cell. So these two markers are mutually exclusive. They don't appear together. So this is the fetal testis, you only see the Sertoli cell staining. So there's no granulosa cell. But if you look at later stage in the double knockout, you can see the dramatic changes in terms of their morphology and the cell type distribution. You can see both Sertoli cell and the granulosa cell are mixed together. But the most important characteristic is this tube structure right here on the poles of the gonad. Those are the ovarian domains. So basically, this gonad developed into the oval testis structure. For fish people, you probably think oval testis is no big deal. We see oval testis all the time. But for the mammalians, this is very, very unusual. You don't see oval testis in the mammal. And oftentimes, this oval testis structure is very transient. They only develop very briefly, and eventually the testicular domain will overcome the ovarian domain, and eventually you will have only one sex present. But this is pretty amazing. You can see, even in the higher magnification, you can see double label FOXL2 and SOCCE9 positive cells, meaning the yellow cell right here, somewhere around here. So meaning those cells that they are undergo the transient fate change. They change from SOCCE9 positive Sertoli cell to FOXL2 positive granulosa cell. So we call this phenomenon transdifferentiation. You have a cell type to start with, and that cell type loses their identity and pick up another identity. So this is called the transdifferentiation. As I mentioned to you, this phenomenon doesn't happen in the mammals very often. And usually when the animal gets to adulthood, this phenomenon is resolved. So we decided to look at adult animals. This is close to 6 months of age. So this is a cross-section. You can immediately see that this is quite dramatic. You have the seminiferous tubule in the middle, and you have the folliculogenesis on the side. So this adult XY individual has both testicular domain and the ovarian domain in this individual. We can even superovulate this individual. So we also stain the marker DMRT1 is Sertoli cell, FOXL2 is granulosa cell. You can see the follicle structure located immediately adjacent to the seminiferous tubule. So this is pretty dramatic. So we were even able to produce sperm and the oocyte from this same individual. We can superovulate this XY individual, and we could collect both oocyte and the sperm from this individual. So this is pretty dramatic because it doesn't happen in the mammal at all if you look at the picture. So this is pretty amazing. This tells us that this particular factor, they are important for the testes to maintain their fate. In the absence of this two factor, the Sertoli cell undergo transdifferentiation and the organ turn into the ovary testis structure. So we want to look at the mechanism. So fortunately, we wait for 5 years. We started this project many years ago. And not the single cell RNA-seq wasn't available until maybe 3 years ago. So we wait for long enough so we can conduct single cell sequencing to look at what happened to the cell. So we get 11.5 control XX, the ovary, and the double knockout testis and control XY testis. We perform single cell RNA-seq. So each single dot representing the transcriptome of each individual cell type. We particularly look at the supporting cell, the Sertoli cell, and granulosa cell, and those changing fate cell in the double knockout. So you can see that the control granulosa cell are clustered right here. The control Sertoli cell are clusters right here. The double knockouts are clustered in the middle. If we run the algorithm to predict the transcriptome change, you can actually see that there is a transcriptome shift from the control Sertoli cell through the double knockout sex reversal cell and eventually settle in the ovarian cell. So this is a typical case of transdifferentiation of the Sertoli cell from the normal XY phenotype to the XX phenotype. So what is the mechanism? So the single knockout the single cell sequencing can really tell us not just the transcriptome change. You can also look for the potential ligand and receptor pair combination among different cell types. So how do you read this one? So for each squares representing the potential presence of the receptors and the ligand pairs between the two cell types. For example, if you look at right here, these are the ligand pair between the Leydig cell and the germ cell. So there are 86 ligand pair, meaning the potential signal transduction molecule and the receptors present between the two cell types. So where are the AMH and activin B and their receptors located? So they are right here in the middle. This square representing the potential interaction between Sertoli cell and the Sertoli cell. So this tells us the Sertoli cells are producing activin B ligand and the AMH ligands, and they are also positive for the receptors for these two ligands. So this tells us this mechanism is the so-called the autocrine and the paracrine control of the cell fate. The cells producing the ligand and they express the receptor, and the ligands act upon themselves in this case. So basically, Sertoli cells are producing this particular ligand system to maintain their fate. So this is what we're proposing. This is a developing fetal testis in the structure we call the testis cord. And inside the testis cord, you have the male germ cell and the Sertoli cells that surrounding it. And Sertoli cells producing AMH and activin B and act upon themselves as an autocrine and the paracrine regulation. And this regulation is important to maintain the Sertoli cell fate. In the absence of this particular two ligands, and the Sertoli cell undergo transdifferentiation and start to express granulosa cell marker FOXL2, and eventually the testis cord disintegrated and start to form the follicle structure. So this is how the oval testis is developed. So the first part, we show you these two factors are important for the fetal testis development. To match the function of the free Martin factor, Another important characteristic they must have is that they should be able to masculinize the fetal ovary, just like I described to you in the free market cases. So how do we test that? This is really a challenge because free market case doesn't really happen in the mouse that often. You don't really see shared placenta in the mouse embryo. And even though if they have shared placenta, how are you going to test this two factor or the free market factor? So this is pretty challenging. So how do we do that? Fortunately, several years ago, Takato Group in Japan developed a very interesting model we thought that we could take advantage of. So what they found is that when they transplant the fetal ovary from the XX embryo into underneath the adult XY animal, underneath the kidney capsule, basically you do an organ transplantation, and you wait for 3 weeks. What will happen is that this fetal ovary is almost completely sex reversed into the testis. So this tells us the adult XY endocrine environment have some sort of factor can masculinize the fetal ovary in this case, right? So we're thinking maybe we could take advantage of this model and replace the wild type XY male with the double knockout XY and ask the question, in the absence of these two factors, does the sex reversal still occur? So everybody's off. So we start the control experiment. We first transplant the XY ovary underneath the kidney capsule of the wild type normal male. So what we found is only 14% of the fetal ovary remain as ovary. You can see the foxtail tooth staining, you can see the follicle. But majority of the fetal ovary develop into the oval testis structure. This is pretty interesting, right? And also 14% they develop what completely sex reverse into the testis structure. So this number is almost identical to the Japanese data, indicating to us that we are able to reproduce the technique in my laboratory. So then we can ask the question, if we replace the normal XY male with the double knockout, what will happen to the fetal ovary? So what we see is the majority of the ovary remain as ovary. So meaning this double knockout male lose the ability to masculinize the female, right? Meaning it lose the free marking capacity to induce that effect. There are still some ovary are developing the oval testis. So we can have some discussion on why this is not a complete rescue. So this really gives us a strong indication that indeed this two factor in the adult situation, they serve as an endocrine function factor that has the free marking capacity. And using this model, we can prove that the factor from the tests probably are the free marking factors. So we published this paper almost 10 years that we started this project. It takes a long time to do the experiment. So many minds and so much effort being put in. So during the pandemic, we start thinking this is really powerful. However, it takes a long time to do this experiment, right? So how do we enhance the process that we can study sex determination in a very short span? You know, in the fish, you can easily inject CRISPR and you can get F1 or F0 generation. You can look at the phenotype. For the mouse, it's impossible. If you want to do the conditional knockout, you can have at least 3 different generations of free in the flux allele to do that. It's really time consuming, right? This is the classic model that we inject the modified ES cells into the wild type blastocysts with the hope that modified ES cells will integrate into the embryo so you can generate a chimera. So this is a typical mouse generation of the transgenic. And the F1, hopefully they will go germline, and you have germline transmission. You can get heterozygous F1, and you breed the F1 into homozygous, and you get a knockout. So you need to get at least 3 generations to get a global knockout, right? But if you want to make a tissue-specific knockout, you need to make 3. You need to make twice allele. We're taking it on at least F4 generation. So this takes at least 6 to 10 months for you to generate a sex-specific knockout. So we're thinking, how do we streamline this process that we can actually study sex determination in F0? Can we do that? So over the pandemic, we started to read paper. There was a very interesting concept called blastocyst complementation. So what they did is the reverse idea. They actually make an embryos that lacking specific cell type. For example, the beta cell in the pancreas or specific neuron in the brain. So when they inject the wild type ES cells, the ES cells will colonize the place where those cells are absent. So for example, this embryo should develop type 1 diabetes because the beta cells are absent, right? But when you inject the wild type ES cells, you can actually rescue the type 1 diabetes in the F0 population by injecting it. So we're thinking, can we use this concept? If we generate a blastocyst without one particular cell type, say Sertoli cell, and by injecting the wild type ES cells, then we can rescue the phenotype. And if this can be worked, then we can mutate any gene in the ES cells, and by injecting into this, you can study the function of the gene in the F0 population in the cell type specific manner. Okay, I hope everyone is following this. So if this really works, can really make the system much, much faster. First of all, we developed a system that the embryo doesn't develop gonad. And in this case, the embryos actually doesn't develop adrenal gland, gonads, and part of the hypothalamus. So what you're seeing here is the whole mount image to show you the control wild type. You can see the adrenal gland on the top. Here's the kidney, testis, bladder, and the ovary. So this is specific sex development in the control. When we develop this blastocyst, we call the gonadless embryos. So you can see that the gonads in the male and in the female completely disappear. So in the absence of the testis or ovary, the embryo develops as a female. So they have the female reproductive tract in both XY or the XX individual. Also, you can tell the adrenal gland completely disappeared. This tells us this model worked perfectly. And also, we do the genetic trick that 100% of the embryos develop this way. So you don't have to worry that 25% of the embryos are this, 100% of the embryos. The same. Jeff over there, he was actually, when he was a graduate student in the lab, he was breeding the Cree into homozygosity so that we are able to do this today. So 100% of embryos develop this way. So now the question is, can we rescue the phenotype by injecting the wild type ES cells? So this is the first control experiment, because when we inject the blastocyst, we don't really know the sex of the embryos, right? You cannot really genotype. So we do this experiment blindly. We don't know whether the blastocyst is XX or XY. It's completely unknown, which is perfect, because this is what an experiment should be. It's a blind experiment. But we do know the sex chromosome composition of the ES cells is XY. So in this case, all the XY ES cells should colonize the gonad in the adrenal gland and rescue the phenotype. Is that the case? So this is indeed the case. You can see the testes actually start to form regardless the genotype of the embryos, they all develop testes as long as the ES cells are the XY. Makes sense? Because the XY ES cell has SRY. And I told you, the host, the genetic background of the host doesn't matter. As long as the Sertoli cell has the SRY, it will form testes. And this is exactly what we have. all the embryos, regardless the XY or XX phenotype, they all develop testicular structure. I didn't show you the adrenal. Adrenal gland is completely rescued. You get a really nice looking adrenal gland. So reversely, when we inject the XX cells blindly into the embryos, we don't really know the sex chromosome phenotype of the embryos, and all of them develop as ovary. So this again confirmed whatever people found, that as long as you don't have the SLI, the somatic cell will follow the ovarian phenotype and develop testes. And the host genotype in terms of sex chromosome is not relevant at all. So this is very exciting. We actually start doing experiment that we can manipulate genes in the ES cells and ask the question whether we can use this system. And we actually, in fact, we mutated the SRY gene in the XXEXY ES cell. And in this case, all the embryos develop as female. So in the absence of the SRY, we only develop as female. So to prove the principle that this system really actually works, so we are in the process, we have a list of the genes that we want to knock out. So we actually can study sex determination in the F0 population in only a month at a time. And also adrenals. This is a very, very, very powerful. Oh, this is just to show you. We also have this experiment. This is the XX ES cells that carry the SRY gene. So in this case, we injected into the XX blastocyst. So all the embryos develop testes because it has the SRY gene. So you don't really need anything else. You just need the gene. If you look at the structure, you get a perfect test formation in this case. So this proves that this method can be used to have a streamlined method to study sex determination, at least in the mouse. Okay, so this method is very powerful. It can be used as a screening method to study sex determination, at least in the mouse. But this is really an open platform for you to think about, because you can actually put human genes in the mouse ES cells and study function of the human gene in this case, and ask the question whether this human version of the gene, what does it do, right? Many scenarios in the disorder of sex development, we don't really know the candidate gene, but now we have a platform, we can actually study that. And by combining with the Degron system, the Degron system is a chemical inducing degradation of the protein. In this case, you can actually control the degradation of the protein. Basically, it's a conditional knockout of the protein at different time and the stage-specific manner. So you don't really need the Cree system. You can use this chemical way to control the gene expression at any time of the development that you want, in using and directly getting that result from the F0 situation. And also increase the efficiency of the germline transmission. We can generate a germ cell depleted gonad. In this case, all the ES cells will become germ cells, can enhance the germ cell transmission by almost 100%. Finally, the cross-species kinetics. This is the most interesting phenomenon, because you can actually think very wildly, if we put the human ES cells in this case, can the human ES cells colonize the mouse embryos? And what kind of program do they follow? They're not allowed to do so at the moment, but the pig and human chimera has been done already. So conceptually, this is definitely possible. So this is really put us one step further to understand the gene function in the human sex determination process. So with this, I really need to thank all my lab people. Really right here are the major driver for the double knockout study and also the complementation. Here, Barbara also did a lot of study on the characterization of that. And definitely, I need to recognize the intramural funding from NIEHS. Without those resources, this won't be possible. Finally, thank you all for the attention. I'll be more than happy to answer any question that you may have. Thank you.
Okay, thank you, Humphrey. We have time for a couple of questions.
So, I just wanted to about mice. You say, change, and also with the sound, has the switch.
Very good question. So the up and down, those are the genes that classic. For example, the upregulation gene are mostly ovarian genes. Win4, FOXL2, and all the genes that you know for ovarian differentiation is upregulated. But the downregulated genes are most of the genes what we know for testicular development, SOX9, DMRT1, those genes are downregulated. So really telling us the AMH and activin B probably are either promoting or suppressing the classic sex determination pathway. So that is the first answer for the first question. Second is the Leydig cell. The Leydig cell develop normally. So all the secondary sexual characteristics are male-like. So you have epididymis, you have all the male tract, but the female tract are also present because the AMH is absent, right? The AMH is inducing the degeneration of the momentum. So in the absence of the AMH, the female tract is present. This is truly an intersex animal. It has both male and female reproductive system and both male and female gonads. Third question is the most important. Is why not 100% sex reversal? It's partial. It's because there are many other TGF beta family proteins still expressed. So we're thinking the reason why we don't see a complete sex reversal is still the presence of the TGF. For example, activin A is still there. So we're thinking it's because those TGF beta family proteins still there. So therefore, phenotype. So at the moment, we're actually using the complementation model to knock out 4 genes at the same time. Knock out AMH, inhibit beta B, inhibit beta A, and also TGF. We're knocking out all the ligand that we can think of. So in that case, we expect a complete sex. Okay.
Thank you very much for a beautiful, beautiful presentation. So using your double knockout mice, you were able to identify the trans differentiation at E15.5, and then you make the conclusion that those 2 regulators are very important. So my question to you is, in a wild type mice, did the trans differentiation occur, and at what E stage that it happens?
So the trans differentiation doesn't happen in the wild type situation unless under certain genetic background. So usually the trans differentiation case doesn't happen very much, but there are some very rare situations that you can find, but those are really, really, really rare. So usually the self-fate determination is very, what we call, penalized. Once it's happening, it's happening. It's a point of no return. So in this case, trans differentiation doesn't really happen very often. No, it's not a normal process in biology. So this process, in human cases, AMH knockout mutation is present, but the double mutation never been documented. So it's definitely a disorder situation that help us to study something.
Humphrey, nice work. So I'm also interested about why they are not 100% transformed.
Yeah.
But there's a very regional specificity, right? You're only in the distals. So if you think it's activin A, is that expression pattern actually match that?
Good eyes. So I didn't tell you it's actually the expression pattern of SRY. The SRY expression pattern is from central to pole. So basically, when the SRY start to turn out, it's turn out in the middle first, and gradually spread to the 2 poles. So our hypothesis, the central region of the gonad has the strongest signal from SLI. So meaning they are being mescalinized first. And cells on the poles, they are being mescalinized the last. So that tells us that there are different susceptibility of the cells to the effects of the knockout. So actually the phenotype we saw is actually very consistent with other oval test structure. Always it's two sides of the gonad being feminized first, whereas the center of the gonad seems to be resistant to the feminizing effect of the knockout. So that has been really consistent. We believe that is the case because of the SRY expression.
But if that's the case, then can we think about that in the center is actually more cell autonomous effect, or rather this distal is
yeah, that is an interesting thought. The only way we can think about testing is really create another in vivo situation, like an in vitro situation, to test the different concentration of the ligand. Maybe the cells will respond to that. I think that's probably the only way that we can test that. In vivo situation will be hard.
Thank you. Okay, I'm sorry, I have to close this session so we can have a tea break. If you still have questions, ask Henry during the tea break. Now I will hand over the mic to tonight. Let's give Henry a big more.
Thank you, my brother. So we're switching gear from gonadal differentiation to thyroid hormone signaling. Our next speaker is Dr. Cheyenne Zhang, and she is from National Cancer Institute of USA, and her talk will be zebrafish is a novel model for patients with mutations of thyroid hormone receptor A1. This was
I'm going to talk about today a non-sex disease. First of all, I would like to thank Professor Zhang and Professor Zhu and the organizing committee for the opportunity to present my work. Thyroid hormone receptor alpha-1, encoded by the THRA gene, mediates the genomic actions of thyroid hormone T3 in growth, development, and differentiation in all vertebrates. Because of its critical functions, mutations of the THRA gene cause a human disease known as resistant to thyroid hormone, named as RTH-alpha. Patients exhibit the symptoms of severe retarded growth, delayed bone development, anemia, constipation, and heart abnormality. So this slide shows the types of the mutation of TRα1 in the patients. You can see that the mutations are clustered in the domains. The types of mutations are point mutation, truncation mutation with a designation of X at the end, and truncation mutation by friendship mutation. Again, you can see the X. This slide shows the representative patients' severe retarded growth. And this graph shows the body height of these 5 patients. By comparing with the norm, which is indicated in solid line, that the z-scores of these patients are well below the norm reference, indicating severe retarded growth. I would like to point out in patients 1, 2, 3, the retarded growth persists from 1 year old up to 30 years old, up to 80 years old, indicating the long-lasting effect due to the mutation of TRR1. And this slide shows a representative patient with delayed skeletal development. When this picture was taken, she was 7 years old. You can see that she has DNA sutures on the cranium. She has epiphyseal degenerative in the hip joint and tooth eruption. All these abnormalities are related to delayed skeletal development caused by the TRR1 mutation. So to understand the molecular basis of RTA-alpha, our lab had developed a mouse model expressing the dominant negative mutant called PV. PV has a friendship mutation in the C-terminal 14 amino acid. As a result of this mutation, PV has completely lost its refining activity and transcription capacity. So this TRR1 PV mouse basically reproduces human RTH-alpha. They exhibit the same phenotypic abnormality as found in humans, such as severe retarded growth, delayed bone development, anemia, and constipation. Here shows, to give you a feel of the phenotype, this picture shows that the mutant mouse has growth defect, and you can see that this mouse is much smaller than the wild.
我从线上看到主持人的面。
So using this mouse model, we have learned how TRR mutant impair growth, delay bone development, cause anemia, and constipation in patients. So if you are interested in the details, feel free to refer to these publications. So this is why there's a need to develop zebrafish model of RTH-alpha. This is because the molecular actions of TRR4 mutant in early development were unknown. So why we cannot use this mouse model for such purpose? And this is because this mutant mouse model is not suitable for studying the actions of TRR4 mutants in early development, because this mutant mice have severe fertility deficiency. Usually, we can get 1 or 2 embryos per mating, frequently. So therefore, to understand the molecular actions of TRR4 mutant development, we decided to develop germline transmittable zebrafish model of RTH-alpha. So I just want to mention I have been a mouse person, and so I'm really a novice in zebrafish. So I was very excited to know that using zebrafish as a model has many advantages that we know. It's quite exciting for me, such as easy genetic manipulation, high fecundity, external fertilization, rapid development, transparent embryos, and importantly, they are cheaper than mice. I would like to point it out that unlike humans and mice, the THRA gene in zebrafish is duplicated as the THRA gene and THRA gene. The THRA gene encodes THRA beta receptors, and the THRA gene encodes TRA receptor. This zebrafish receptor share 90 to 95% sequence into the human TRR1 in the DNA binding hormone binding receptors. We can use CRISPR-Cas9 mutagenesis to mutate both the THRA gene and THRA gene. And one base pair insertion mutation in the THRA gene led to this TRα E394X transport receptor and an 8 base pair insertion mutation in the THRA gene led to TRR1 and 4,5 FSX5 receptor. Hiểu không? Interruption. Continue. And 8 base pair insertion mutation in the THRA gene led to TRR1 and 405 FSX6 receptor. This mutation is similar to mutations I talked about a few minutes ago. So we found the mutant fish with the only mild retarded growth in the mutant fish with the THRA gene mutation, with only about 5 to 10% reduction in body weight. So this slide shows the picture of our mutant fish with retarded growth. We were invited to showcase this phenotype on the cover of the journal's diary. You can see that this mutant fish are smaller than the wild type fish. We then elucidate how the THRA mutant causes retarded growth. We identified 2 molecular mechanisms. One is the repression of growth hormone, insulin growth factor excess. You can see that the mutant suppresses the expression of 3 fish growth hormone genes, GH1, SMTOA, and SMTOB. The mutant also suppresses the expression of insulin growth factor 1 and receptors, such as IGF-1A, IGF-1B, and insulin receptors. In the other pathway, the mutant inhibits the protein synthesis via AKT, S6K, S6 signaling. You can see that the mutant decreases the protein levels of phosphorylated AKT, phosphorylated S6K, and phosphorylated S6 to reduce body mass. So we then asked the important question, at what developmental stage that TR-alpha mutant cause retarded growth? And we found that TR-alpha mutant cause retarded growth as early as in juveniles. You can see that the body length, the body width, and the body weight were all reduced in the mutant fish. And the expression of the 3 fish growth hormone gene was also suppressed in the mutant fish. However, we did not find mutant larvae exhibit retarded growth. You can see that there's no differences in the body length, and the expression of the 3 fish growth hormone gene was not significantly changed. So this result indicate that TR-alpha mutant had no effect on the body weight, on the growth, and the larva stage, but act to impair growth beginning at juvenile, persist to adulthood. So the retarded growth demonstrated in our mutant fish, as found in patients and mutant mice, indicated to us that the in vivo actions of TR-alpha mutant are conserved among humans, mice, and zebrafish. It is known that patients and the mutant mice exhibit bradycardia. However, it was unknown at what developmental stage that the heart abnormality developed. So we therefore used the zebrafish model to ask the question that at what developmental stage that the heart abnormality develops. First, we determine whether our mutant fish had bradycardia. So we found the heartbeats of mutant fish were slower than the wild type fish. And the heart rates were decreased about 15 to 25% in the mutant fish as compared with the wild type fish. So these data indicate that mutations of the THRA gene cause bradycardia. So to understand why the mutant fish has lower heart rates, we then examine the histology of the heart. Compared with a wild type heart, the heart of mutant fish had a normal shape and size. So you can see that atrium was dislocated and dilated as compared with the wild type atrium. And we also found that the ventricle of the wild type fish had changed in the mutant fish. In the wild type fish, it is more like a wedge, but in the mutant fish, it was more rounded. So this histological defect could lead to the weakened heart contractility. In addition, we use mutant fish co-expressing GATA when E is red, and we found the mutant fish had defective blood flow. So this video shows that the blood flow in the mutant fish was slower than the wild type fish, and the speed of the mutant fish was reduced about 10% as compared with the wild type fish. So we attributed the slow blood flow was due to the weak contractility, and therefore we make the conclusion that mutation of TR-alpha-1 impaired contractility of the heart. We then visualize the blood vessels of the mutant fish. We cross our mutant fish with fish expressing GFP3-1. So in this slide, for a clearer comparison, we mark the color of the vasculature of the wild type fish in white, but kept the green for the vasculature of mutant fish. As indicated by the orange arrowheads, you can see that the vasculature of the wild-type fish were organized and more compact. However, in the mutant fish, the vasculature were disarrayed and fragmented, as indicated by the white arrowheads. So the disorganized vasculature in the mutant fish could contribute to a slow blood flow. So the decreased heart rate and slow blood flow could reflect the weakened heart contractility. We therefore evaluate the expression of contracted genes. We found that the expression of the contracted genes were decreased in the mutant fish at the mRNA levels. You can see that the decreased expression of SLC8A1A, ATP2A2A, RY1R2, and PON. We further determined the expression of the contracted genes at the protein levels. You can see in this Western plot, the protein levels of ATA2A and troponin T were lower in the mutant fish as compared with the wild-type fish. And the quantitation indicates a decrease of about 30 to 60% in the mutant fish. So the decreased expression of the contracted genes could lead to the weakened heart contractility. We further used transmission electron microscopy to analyze whether there is kind of structural abnormality in the sarcomeres. Sarcomeres are the basic contractile units of cardiac muscles. So compare with the wild-type sarcomeres, the width and the length, you can see that the width and length in the mutant fish were shortened. And quantitation indicate that the length of the sarcomeres and the width of the sarcomeres were shortened by about 20 to 30% in the mutant fish. So this structural abnormality could also lead to the weakened contractility. We then ask the important question. That's the reason we use the zebrafish. At what developmental stage that TR-alpha-1 caused heart abnormality? We found the heart abnormality could develop as early as in juvenile. You can see that the atrium was dilated and dislocated compared with the wild-type atrium, and the ventricle of the shape of ventricle was changed in the mutant fish to be more rounded. And we also found that the flow speed of red blood cell was decreased in the mutant fish as compared with the wild-type fish. So this result indicates that the heart abnormality could develop as early as in juveniles and persist to the adulthood. So in addition to the characterization of the growth and heart phenotype, we also use RNA-seq analysis to compare the gene expression in a wild-type mutant pituitary. This volcano plot shows that there were more upregulated gene than the downregulated gene, and the heat map shows distinct expression patterns between the wild type and mutant. We identified 723 differentially expressed genes, and 640 genes were upregulated, and 81 genes were downregulated. Gene expression pattern analysis indicate the decreased expression of non-pituitary genes such as SMTLB, GH1, POMCA, and IHB, and the upregulation of the non-pituitary genes TSH-beta and TRHRB genes. Pathway analysis indicate there are major changes in the signaling caused by TR-alpha mutant, such as developmental disorder, skeletal and muscle disorder, and other growth abnormalities. So what have we learned from zebrafish model of RTH alpha? We learned that the in vivo actions of TR-alpha mutant are conserved among humans, mice, and zebrafish. Growth retardation and heart abnormality mediated by the TR-alpha mutant occurs only at post-larva stage. Zebrafish model of RTH alpha have provided a unique opportunity to gain new mechanistic insights into the molecular actions of TR-alpha mutant in vivo, which are not available in humans and mouse models. So our germline transmittable zebrafish model of RTH alpha and mutant fish expression data with ES-REG and GFP-FLIP1 can be used for rapid and large-scale screening of potential therapeutics for RTH patients. So the work I presented today was done by a very talented fellow, Churong Han, together with Eric Olsen, and I thank my collaborators for the important contribution to the work I presented today. I want to offer my special thanks to Dr. Zhu Yong for his teaching, guidance, and advice in how best to use zebrafish in my project. When I started the zebrafish project, I had no idea about zebrafish, how to study them. And then I could not even tell the difference between a male and female zebrafish. So whenever I have questions, I reach out to Dr. Zhu, and he was always very promptly respond to my question, regardless how trivial or how naive the questions were. He even made a trip to NIH to visit the fish facility and send me specially formulated zebrafish food to make my fish happy in mating. I also want to thank Dr. Duan for valuable discussion about functions of insulin-like growth factor in fish. And I want to take this opportunity to thank Professor Zhang for providing such beautiful venue, wonderful food, and warm hospitality. Thank you very much for your attention. I welcome your questions.
Thank you. Some questions?
Excellent talk. I just noticed that size and gene mutations whereas in the heart similar. With the two receptor genes are expressed in similar heart procedures.
That's a very question, and thank you very much. And like in mammals, in the heart, the major isoform is TR-alpha. But in the mammals, there's no duplicated genes. And then in the pituitary, the major is TR-beta. So that's why you saw different phenotypes in different tissues.
Thank you and congratulations for joining the fish community. I was wondering, do you have any effect on the brain?
No, we did not do that because fish is so small. I don't know how to isolate that. So I didn't do that. Did not characterize it.
Pretty naive about the thyroid. So as I remember that in the textbook it says that about thyroid signal is regulating the EC and actually secreting that act. I'm just wondering, how much the defect here you see is because of the EC production.
You mean the production means the expression of the protein level?
Yeah, the EC expression will be affected by thyroid signal. So that's going to affect segregation.
Yes.
So I'm just wondering, how much of the defect is caused by the regulation of that.
So I kind of want, I didn't get a chance to point out the mechanism. And so this PR alpha mutant act by dominant negative effect. So again, it depends on the expression protein levels, but mainly it's through the transcription regulation via through the co-repressors. So we have not analyzed, the protein levels of co-repressors. So that's my answer to your question. Is that okay?
Last question.
It's curious why distribution is
Yes, in a mammal, in a fish, from DPF1, the gene or the express the duplicated gene and the other ones for TR beta also already expressed. So, but in the zebra fish, it was not possible for me to dissect different kinds of tissue to look at its expression pattern. But I can answer your question in humans and in mice. In humans, the major TRS form expressed, there are two in humans that TR beta and TR alpha, and then we call TR beta and TR alpha. So in the TR beta, the major isoform in the liver is TR beta. The major isoform in the brain is TR alpha. In the kidney also is TR beta. And in the thyroid also is TR beta. In the mice follow the similar kind of protein expression patterns.
Thank you so much, Robbie. Our next speaker is Professor Yoko Shi from National Institute of Child Health and Human Development, and his title will be The Sick Roles of Unliganded and Liganded Thyroid Hormone Receptors in Regulating Marble Tissue Regeneration and Adult Organ Development.
Well, what is being set up, first of all, I also like to thank President Zhang and the rest of the organizing committee for putting together such an exciting, excellent symposium and for inviting me to speak here. And it's really tough to follow two such excellent talks, so I'll try my best to tell a very simple story. So my lab has been studying how thyroid hormone regulates vertebrate development. I appreciate Dr. Chen's really outstanding introduction on thyroid hormone signature, the use of thyroid hormone. And for my talk today, I would like to begin by a brief introduction by taking human life cycle, dividing into three phases. So the first phase I would like to talk about in human is the embryogenesis, which is the first 5 months of human development. It occurs when plasma thyroid hormone concentration is very low. I just use it. And this is the part when embryonic organs first form. The second part is so-called post-embryonic development, which is actually about 4 months before to 7 months after birth. In human, that's when, in mammals in general, that's when organs mature into their adulthood. And finally, what I call child growth and adulthood. That's when thyroid hormone level reach steady adult levels. So post-embryonic development, as shown here, is a time when thyroid hormone peaks. And this is a period actually is the least studied, in part as Dr. Chen mentioned earlier, it's really hard to manipulate mammalian embryos during this period because of maternal influence. So neonates or embryos before birth relies on maternal supply of nutrients for survival. On the other hand, this period is very critical for thyroid hormone to act. Thyroid hormone deficiency is known to cause severe developmental problems that are likely, as we see some examples of thyroid hormone receptor mutant earlier. And thyroid hormone deficiency, in an extreme case, causes the formation of so-called human cretins, which are very short and with severe neurological disorders. So the question is, how does thyroid hormone regulate this developmental period? Now, we have been using frog analysis as a model. As diagram here for Xenopus, what you see is that as a tadpole is transformed into a small frog during the second month of Xenopus development, every single tissue or organ is transformed. And this transformation process are very similar to organ maturation in mammals. And they are how does thyroid hormone? Dr. Chen mentioned briefly earlier how thyroid hormone functions. I will remind you that thyroid hormone functions mainly by regulating gene transcription through nuclear receptors or TR. TR typically form heterodimer with nice cis-red amino acid receptor. Such heterodimer complex will then bind to so-called TRU or thyroid response element in chromatin, which then recruits repressor complexes, which often lead to the increase of so-called repression of his marks to keep the gene silence. So in the presence of thyroid hormone, the heterodimeric complexes then coactive forms, which lead to the increase of so-called activation histone marks and chromatin destruction, which in turn activate gene transcription. So based on this and other studies, a number of years ago, we proposed a dual function model for thyroid hormone receptor during frog development. That is, we hypothesized many thyroid hormone-induced genes are expressed at so-called basal level between stage 1 and stage 45 during embryogenesis. Stage 45, which is roughly 4 to 7 days old, is a time when the tadpole begins to feed. Embryogenesis country, we have a free living tadpole. So therefore, we can imagine the expression of such genes are no longer required. So how do we turn them off? We propose that the expression of TR-alpha gene and ARX-alpha gene very highly by stage 45. These formation of TRs are heterodimers that bind to the promoter region, recruit repressors because that's what the ligand treatment. This then keeps the gene silence. And we propose that this repression is important to make sure the animals stay as a tadpole, do not become functional. That is control timing of metabolic communication until stage 54, when the synthesis of endogenous thyroid hormone occurs. Thyroid hormone bind to the promoter ligand receptor, which then recruit co-activators to promote to active transcription of very same genes to very high levels. This then triggers mitosis. Now, among the genes regulated is the thyroid hormone receptor beta gene. As you can see, the beta gene level rises very quickly, parallel to the plasma thyroid transcription. So over the years, we and others have carried out different experiments to support this model. What I would like to share with you today is the gene editing studies and global and genome-wide analysis that we carried out in recent years. So we initially focused on the TR-alpha gene because it is highly expressed very early on. So we would predict that if we knock out TR-alpha as a predominant receptor, we would eliminate this repression during pre-menopausal. And this then will make the genes express at a basis of low repression, which in turn should make the animal develop early, initiate menopausal sooner. And we actually did a very simple experiment. We simply measure how long does it take for animal to go from stage 1 to stage 54 which is considered unsettled process. What you see is that homozygous takes a couple of time to reach them. So indicating thyroid hormone receptor alpha controls the initiation timing of meiosis. So what happens after? Now, if we take meiosis between stage 54 to 66, divided into two halves. The first 54 to 58, when TR beta and plasma thyroid hormone concentration is still low. And the second half is 58 to 66, which is considered climax meiosis, when things go really quick. In 2 weeks, this part is finished. So we ask, what happens if we knock out TR? Between 54 to 58, TR is still a dominant receptor. So if you knock out TR, you don't have enough receptor to mediate thyroid hormone signaling. So the process should go slow. And indeed, that's what we saw. Homozygous takes twice as long to develop from stage 54 to 58 compared to heterozygous or wild type. So finally, we asked what happens during climax. What happens to TR knockout with the and this presumably because TR beta is now highly induced, which can compensate for TR. So then we ask, what happens if we now knock out TR beta? If we knock out TR beta, I won't show you the detailed data. I will simply compare what we observed in distinct organs, compare the phenotype of beta. Now, stage 54 is the onset of meiosis. If we first look at the limb development, which starts before meiosis and finishes around stage 58 with the limb 44. What we found is that if we knock out TR alpha, limb development before meiosis is accelerated, whereas after thyroid hormone is given to induce meiosis, it's delayed. However, TR beta knockout doesn't have effect. Limb development either before. Now, if we look at the intestinal remodeling, the intestine and the tablet is very similar structure. Whereas in frog, it's this complex structure, very similar to the intestine. What happens is that during meiosis, mostly between stage 58 to stage 66, the inner larva epithelial cells undergoes apoptosis, as indicated by this open circles here. So the larva epithelium degenerates. Concurrently, adult epithelial stem cells, as indicated by those dots here, they start forming around stage 60 to 56. Then they proliferate, eventually replace the entire, then differentiate to form this molecular structure. So what we found is that intestinal remodeling is delayed by both TR alpha knockout and TR beta. Finally, we analyze tear resorption. Tear resorption, as reflected by the reduction of the ends of the tail, occurs mainly after stage 61. It reduces to 0 by the end of the process. No tail is left. And what we found, as I mentioned earlier, TR knockout does not affect tear resorption. However, TR beta makes this process. So this indicated two receptors have organ-dependent development, stage-dependent effects on animal development. And interestingly, if we try to correlate the phenotype of TR or TR beta knockout with the developmental expression profile, simply by RT-PCR, in different organs and different tissues, we found almost perfect correlation. So when we see an effect that makes this gene is highly expressed in that tissue. So the question then is, how does the TR alpha TR beta knockout affect? I mentioned earlier in our functional model that receptor bind to target to regulate transcription. Is that the case? So in recent years, we started to carry out RNA-seq and ChIP-seq. RNA-seq to measure transcription in different organs, in different tissues. and use a chipset or chromatin immunopresentation sequencing to measure what genes are bound by the receptor in different tissues. I will show you just one slide on the chipset. So what we did here is use antibody against TR to measure the total number of genes bound by thyroid hormone receptor in tadpole tissues at onset of metamorphosis, stage 54. Now, if we look at bound genes in the wild type tail, we have about 1,400 genes. But in the intestine, we have 4,600 genes, and hind 3,700 genes. So this shows us right away that there are more receptor bound genes in the intestine hind than in the tail. And this actually is perfect in agreement with what we know to biology at this stage. That is, if we take stage 54 pattern and add thyroid physiological level to the body, within 3 days, you will see intestinal development, limb development, but you don't see tail resorption. The reason we think is that because at this very stage, you simply do not have enough receptor to bind sufficient number of genes in the tail to mediate the resorption process. So next, we ask what happens if we knock out TR. If we knock out TR in the tail, we found the total number of genes remain bound, which is presumably due to the thyroid receptor beta. Is 1,000, so there's 361 gene reduction in the total gene count to 6%. In the intestine, we found almost 1,300 genes less bound in the knockout animals compared to 128%, in the hind is a 33% reduction. So there's a bigger effect of TR from knockout on the TR bound genes in the intestine and the limb compared to tail. Now, if we further analyze, taking these genes that are bound in the wild type animals, but not in the knockout, that is 700 genes in the tail, I think it's about 1,400 genes in the intestine and 1,400 genes in the hind. We analyze it by using gene ontology or pathway analysis. What we found is that the biological pathways or the geo terms affected by the knockout is much more in the intestine and hind. You see 27 to 4,000 geo terms affected versus 1,900 in the tail. For pathway, we have over 200 in the intestine compared to 150 in the tail. Similarly, if we focus on the pathways that are most important for the changes in the tail intestine, the tail is resorption, cell apoptosis. In the limb and intestine, we have cell proliferation as reflected cell cycle. What we found here again, in the geo terms, you see a lot of more cell cycle development programs in the limb and intestine being affected by knockout compared to the tail. Same thing is true of the hospital system. Even apoptotic process, you can see intestine limb, which involves some cell lines, unlike a tail, which is predominant cell lines. You see as many or more apoptotic pathways of your terms affected by TR knockout in the intestine. So all this is consistent with the fact I showed earlier. Limb development and intestinal development is inhibited by TR knockout, with tail resorption is not affected. So showing that indeed the gene regulations with direct binding by the receptor to target genes in those tissues are responsible for the biological effect we observe in knockout. So far what I've shown you is that TR alpha and TR beta have distinct functions, most likely through gene regulation by direct binding to targets. So then the question is, why individual alpha and beta knockout can still finish the meiosis, become reproductive adult? Suggests maybe TR alpha and TR beta can compensate each other. So therefore we ask what happens if we knock out both alpha and beta. Now based on this expression proof, we would like to predict that perhaps an animal will initiate meiosis early around stage 50, reach them much earlier because you do not have this regression. But then once around stage 58, when thyroid hormone level is high, the development should stop because you do not have any receptor to mediate pregnancy. So our prediction would be animal should somewhere stop development on 54. But actually, it's kind of surprising. We locked out. I'll show you just a couple summaries. We lock out both TR and T-beta, and then again, simply measure how long it takes for animal to the onset of metamorphosis, which is 54. What we found is that it takes about 20 days, 40 days, 6 months. Well, this is exactly the same as TR. So TR plays most in regulating the onset of metamorphosis. Now, if we look at the development from stage 54 to the next stage, 61, we found that double local animals take 51 days. For wild type, it takes 26 days. Now, I don't have the data here. If we put TR on the homozygous knockout, somewhere in between. So the double knockout development is slower than TR, which is consistent with the fact that both TR and TR beta influence metamorphosis after stage 5. So what happens then after stage 6? What we found is that if we look at wild type animals, or TR homozygous knockout, that is, have 2 copies of TR beta, or TR homozygous and TR beta heterozygous, that is, there's only a single copy of TR beta gene. What we found is those animals behave just like a wild type and develop past stage to stage 62 and eventually to sexually mature adult, although the timing is more short. So single copy of either alpha and beta gene is sufficient for animal to develop to adult. But TR and TR beta double knockout all died at stage 61. So what happened here? Now I should have pointed out, I mentioned earlier, we predict that the animals should stop development around stage 50. But we were surprised actually to stage 50. Now, if we analyze why they can develop to 65, what we found is that here, what do you see is that if you first look at the and the structure of the head region, wild type and wild type at 6, what you see is that actually that identical. And those are adult structures. So the adult structure, adult tissue of the organs can actually form up to stage 61. But if you look at the gills, you see the gills are much larger. Well, gills are lobus of. And then if you look at the tail, which hasn't begun to absorb yet, it starts after 6 months. So absorbed animals, within 1 week, the wild type animals absorb the tail completely. But wild type animals just sit, nothing change, very little change. And in 2 weeks later, so this indicates that the deregression due to knockout can allow adult organ and head structure and body skin. However, lung tissue degeneration will be more often. So this perhaps is the reason why I died. So to very quickly summarize what I mentioned, that this is dual function exists for technical development. During pre-metamorphosis, unlike the receptor controls the timing of initiation. And this is mainly done by TR. Once metamorphosis begins, TR and TR beta both contribute to the rate of metamorphic progression between stage 54 to stage 60. And then after say 61, either one of the receptors is essential for the complete absorption of tissues. In normal animals, TR beta they have TR that's TR beta. So how is this similar or different from other vertebrate species? Now, what's known is there are a lot of studies in mouse, and interestingly, they're highly conserved. What you can see is the thyroid signal is critical for both mouse and frog. Without T3, none of them can develop into sexually mature adult. Mouse died perinatal, frog died as tadpole. Animals without either TRG can develop into reproductive adults. So either one is sufficient for both mouse and frog. And the effects of TR knockout alpha or beta correlates very well with the receptor expression in different tissues in dogs, mouse. And the main difference we saw is that mice without TR can develop into adults, whereas frog without TR dies. Now, even here, look carefully, we see that TR is not required. But with frog, TR is essential for all tissues. We think this actually is very interesting because frogs has a 3 living period as temple. Unlike mammals, which is one year, we should survive frog living independently. And this requires certain larval tissue, larval organs tissues, which has to be resolved to become larval. Therefore, TR is essential for survival. Mouse or mammals doesn't have such organs, and therefore the function of TR for survival is not important. So we think maybe during evolution, the essential function of TR for survival is lost. Or TR may have more in regular tissues during evolution. That remains to be tested. Finally, I'd like to very quickly point out the people involved in the studies. Longwen and Yuki Shibata did most of the work in knockout, and Yuki Tanizaki did most of the work in global analysis. And our knockout study was initiated with human chance that Stephen Showing. Thank you for your attention. Welcome back.
Yeah. We have time for a few questions. Yes.
Thank you, Yongbao, for the presentation. I have a question. The survival curve that you
We actually do not see significant difference between TR beta heterozygous animals that contains TR versus TR beta homozygous is actually a big difference. Wild type versus TR beta heterozygous, big difference in TR absorption. Homozygous is delayed. Now, with the single copy of TR beta, the development is delayed, but eventually sexual maturity. So, but it is delayed. Even here. Yes.
So you mentioned most of the biological effects from the variety of TR to the target gene.
I didn't quite catch the first one.
So the biological effect TR is due to the readily binding of the target gene.
That's right.
And but as you show, different tissue have different amount of targeting gene being selected. So I'm wondering that whether this is determined by the expression level of the receptor, the TR, or there are some other replacements to suppress its binding up to the target gene.
Well, That's a very good question. And we do believe cofactors and variation of cofactors can contribute to tissue existence. Now, at a quantitative level, we think it's mainly due to the different levels of receptor expression. So, like in a tail, like TRG is very low, but they're strongly induced, especially TR beta, at a climax of muscles. So, but we also did observe there are other factors can contribute to thyroid signal, like a thyroid binding proteins. Years ago, Dr. Chen and I collaborated. Many years ago, we showed there's a thyroid binding protein. The level changes in different tissue processes, suggesting this protein function to treat thyroid out to prevent thyroid signal. So therefore, so definitely, I think I agree with you. Other factors such as co-repressors or even thyroid transporter binding will contribute, but the dominant factor is TRG.
So, one last quick question before we discuss this with you. Thank you for your talk. You are showing the heterodimer with Alexa. So is there any regulation of Alexa in relation to metabolites?
We actually did analyze RTR. We do see some parallel, not as dramatic regulation of RTR, and they do go up during the analysis, but it changes relatively. And the other thing is that there are 3 RTR genes. So at the time we were analyzed, there are only 2 RTR genes developed. So we do not know the RTR level varies tremendously during the analysis. We only analyze RTR alpha and gamma. And overall, there's some trend of change, but much less than TR beta generation. Now, I should say that RTR is really at least human development, it's really a sign of the part. So we actually have treated animals with RTR ligand, RAR ligand, without therapy, nothing. So it really helps to receptor bind. But it's protein level may not be difference, but it's a relative bind.
Thank you. Yeah, so then we thank all the speakers for a wonderful session. Thank you.
Library.
So welcome back. So we have three more very exciting talks waiting for us. So the first speaker for this particular session is by Professor Xu Guizhen. She's from the Developmental Biology and Stem Cell Biology Academic Clinic in Taiwan. So she's going to talk to us about the excess dietary sugar harms stem cell. Professor Shi.
Thank you. So, hi, good morning. Really, no place like home. I'm very great. I have this chance to come back my mother university and share my research work with you. Before I start my talk, I would also like to thank Dr. Zhang, President Zhang, to invite me to come back my home. All right. So many people like sweet food, not only because they taste good, but also because they can make us happy. But you know, sweet diet often leads to obesity and diabetes, both of which are metabolic disorders and associated with dysfunctional stem cells. However, whether the high sugar diet itself or metabolic disorder cause the dysfunction of stem cells remain unclear. So why do we need to care about stem cells? Because they can continuously generate differentiated cells and replenish the cells that are lost from the tissue, thereby maintaining tissue homeostasis. In the case that stem cells are dysfunctional, then the tissue usually undergo degeneration. So from here, you can know how important the stem cells are. Stem cells are therefore tightly regulated. They reside in the niche. They not only regulated by niche signal by stem cell intrinsic regulator, but they also need to respond to physiological states and nutritional input to fit the fitness of the organism. However, the processing, the mechanism underlying this processing remain unclear. So from the past, my lab has been working on how niche is established to recruit stem cell and how stem cell responds to aging and diet. Today, I will talk about how high sugar diet impairs stem cells, and can we find a solution to let us enjoy the sweet diet, and at the same time, we can protect our stem cell. So to do this, we use Drosophila as a model for several reasons. First of all, the fly ovary and intestine respond rapidly to diet, and they contain well-characterized germline stem cell and intestinal stem cell respectively. So the Drosophila, in the Drosophila, the functional unit of egg production is called ovario. The anterior most structure is called germline, which houses 2 to 3 germline stem cells at its anterior tip. And each germline stem cell contains a round-shaped fusum, which is a unique cytoplasmic organelle. In contrast, the differentiated germ cell cysts, they contain a branch fusum. This differentiated cyst eventually develop into egg chamber and then mature into eggs. The Drosophila gut can be divided into foregut, midgut, and hindgut. Among them, the midgut, the function of midgut is highly relevant to mammal small intestine. And this region contains very high activity of intestinal stem cell. They localize closer to the basement membrane, and they contribute to build all the cell lineage in the small intestine. Most importantly, all the cell fate in the intestinal intestine can be identified by different cellular markers. For example, in this image, the green cell labeled by escargot NCDA-GIP mark intestinal stem cell and their progenitor. So you can see after immune staining, we can quickly identify stem cell just based on the cellular marker of the cell size after challenging with different diets. And second,
There are several key signaling pathways are very conserved between fly and mammal, including insulin signaling pathway that control glucose uptake. So from here, without the explanation, you already know insulin signaling is highly conserved between fly and mammal. It's not only control the glucose uptake, but also control the cell growth through promoting protein synthesis. But today we will only focus on AKT and glucose transporter. In mammal, insulin is secreted by pancreatic beta cells. Upon insulin bind to its receptor, the downstream signaling cascade recruit AKT to the plasma membrane where it is phosphorylated. Then the phosphorylated AKT can promote the translocation of glucose transporter from cytoplasma to the plasma membrane to do the glucose uptake. Finally, the body size of the fly is very small, so we can easily challenge fly with different diet conditions. Most importantly, a model of diabetes in fly has been established. In 2013, in Kenga's lab, they treated the fly with a diet containing 1 molar sucrose for 3 weeks to develop an insulin resistance. They confirmed this by insulin response assay using Western blot. So what they did is actually get the fly assay from different diets and then treat it with or without insulin peptide. So the assay from control diet, as you can see, when they add the insulin peptide, the phosphorylate AKT here use the readout of insulin signaling reporter is highly activated. However, the lysate from high sugar diet treated fly, the increased level of phosphorylate AKT is not as much as in the control group. So this indicated that under high sugar diet treatment, the cell have poor response to insulin. So to study the effect of high sugar diet on stem cell, we first try to distinguish high sugar diet induced obesity or high sugar diet induced diabetes affect the stem cells. To do this, we try to generate obese fly without insulin resistance. So we try several different concentrations of the sucrose and different feeding periods. At the end, we found that fly fed with 1 molar sucrose for a week can develop to the highest degree of the obesity, and they are relatively healthy. So here are the adipocytes from control diet and from the high sugar diet. As you can see, the adipocyte from the high sugar diet contain larger oil droplets labeled by the body P showing green color, indicating that this fly are obese fly. We also perform insulin response assay using immunostaining. So you can see the adipocyte from the control diet has very low level of the phosphorylate AKT. Upon insulin peptide treatment, the expression level of phosphorylate AKT is dramatically increased. The adipocyte from high sugar diet already shows stronger expression of phosphorylate AKT. Interestingly, upon insulin signaling treatment, phosphorylate AKT is enriched in the plasma membrane, indicating that this adipocyte increased the sensitivity in response to insulin. However, when we detect the glucose or the triglyceride level in the hemoglobin, we did not find any difference between the control diet and high sugar diet. So this really means we generate an obese fly without insulin resistance. So interestingly, when we assay the egg production, we found that high sugar diet treated fly showing red, they decrease the egg production as early as 3 days after high sugar diet treatment. And then in 2 weeks old fly, with then the ovary are much smaller compared to the control diet. So that means high sugar diet disrupts oogenesis and decreases egg production. Then we further analyze germline stem cell proliferation using correlation. So EDU show you in red and germline stem cell outlined by dashed circle. We counted the percentage of EDU positive germline stem cell, and as you can see, obviously there are about 50% of reduction in germline stem cell proliferation. We also analyzed germline stem cell number in the fly culture under control diet and the high sugar diet, but we did not find any differences in terms of germline stem cell number. So here we know that high sugar diet decreased germline stem cell proliferation prior to insulin resistance. Then we further ask, does excess sugar or excess calorie cause the reduction of germline stem cell proliferation? To answer this, we fed the fly with similar amount of calorie compared to the high sugar, but essentially substitute by protein or fat. So after a week feeding with high fat diet and high protein diet, we found that this fly all developed into different degree of obesity. By the quantification, we found that high sugar diet and high fat diet, they produce similar degree of the obesity, but we did not find germline stem cell proliferation reduced phenotype. So this suggests that high sugar diet has a specific effect on germline stem cells. Then we think about if we can remove the excess sugar from the diet, can we reverse high sugar diet induced phenotype? So what we did is that we culture fly under high sugar diet for a week. Then we switch those flies from high sugar diet to control diet for additional week. And then we examine the phenotype. So firstly, we examine the obesity by examining oil droplets in the adipocyte. And as you can see, after a week switch to control diet, the oil droplet size is greatly reduced back to the level that you can see in the control. So this means dietary switch from high sugar diet to control diet reverse obesity. Then we further look at the egg production. Now please pay attention to this orange line. So after we switch diet from high sugar diet to control diet, as you can see, the egg production is also rescued back to the control level. More interestingly, ovary morphology or ovary size also back to the control level. So this means dietary switch from high sugar diet to control diet reverses the disrupted oogenesis and egg production. Then we want to ask, what happened to the germ cell at the cellular level on the high sugar diet? So there are several papers report that when cells culture under a high glucose medium, those cells accumulate high level ROS, and the ROS has been known to activate junk signaling in several different cell types. So we first examine glucose uptake in the glomeruli under different diets. So we incubated with the ovary with 2-MBDG, which is the analog of glucose, but with fluorescence. And this glucose analog cannot be further metabolized. So you can see that on the high sugar diet, all the glomerular cells have increased 2-MBDG signals, means that they have increased glucose uptake. And they also have high level of the ROS as revealed by DHE staining. Similarly, they have the high level of the junk signaling as revealed by P-junk staining. So we know that high sugar diet increase glucose uptake, ROS level, and junk signaling in the germline, including germline stem cells. Okay, so then we started to do the dietary switch experiment and to see whether ROS level could also be back to the control level. And this is the case that after a week switch diet from high sugar diet to control diet, we did see the reduce of ROS accumulation and it's actually back to the control level. So then we directly genetically manipulate ROS level and junk signaling. What we did is that we overexpress an anti-oxidant enzyme, SOD1. Okay. Consistently, the control fly, you can see the reduction of germ-like stem cell proliferation under high sugar diet. When we overexpress SOD1 and culture fly under the control diet, the proliferation is already reduced. So this indicates that basal level of ROS is important for germ-like stem cell proliferation. But interestingly, when we culture SOD1 expressing fly under high sugar diet, they did not show further reduction of the germ-like stem cell proliferation. Similarly, when we disrupt junk signaling by knock down junk kinase or junk kinase, we did not see further reduction of germ-like stem cell proliferation on the high sugar diet. So this means that it says dietary sugar decreased germ-like stem cell proliferation field out as junk signaling. Okay, so then we further ask, does it say dietary sugar reduce germ-like stem cell proliferation feel its own action on germ-like stem cell or fire another cell types? To do this, I thought we need to identify the glucose transporter in the germ cell, which is even not known in the mammalian germ cell. So to identify the glucose transporter, we collect all the RNA line that target to the known glucose transporter in Drosophila. However, after a few months' work, we did not identify any single glucose transporter that's responsible for glucose uptake in the germ line. We did find that some of the candidates, when we knock down and treated with the high sugar diet, they don't have accumulated ROS level. So we think probably more than one glucose transporter responsible for the glucose uptake in the germ cell. So to know whether to answer this question, instead of finding the glucose transporter, we knock down insulin receptor to disrupt insulin signaling, which is known to regulate glucose uptake in muscles, adipocyte, and many other peripheral tissue. So we first examine the glucose uptake in the insulin receptor knockdown germ line. So you can see the control first. Under the high sugar diet, they have the increase of glucose uptake by using this glucose fluorescent probe. Knockdown insulin receptor, the glucose uptake is slightly reduced, but under the high sugar diet, we did not see the increase of glucose in the germ cell. So this indicates that insulin signaling controls glucose uptake in the germ line. And interestingly, when we examine ROS level, so in the control group, you can see the ROS level is increased, consistent to our previous report. But in the insulin knockdown germ line, we did not see the increase of the ROS accumulation on the high sugar diet. This means that germ-line disruption of insulin signaling does not show increased ROS on the high sugar diet. Then the most important thing is that whether the reduced germ-like stem cell proliferation can be reversed when we knock down insulin receptor. So we perform the EDU incorporation AC. You can see in the control group under high sugar diet, germ-like stem cell proliferation is reduced. However, when we knock down insulin receptor, the germ-like stem cell proliferation is not further reduced, even though we culture it under the high sugar diet. So we thought that germline disruption of insulin signaling does not show decreased germline stem cell proliferation on the high sugar diet. Then we conclude that even though we cannot identify the glucose transporter, but through this insulin receptor data, knockdown data, we propose that it says glucose in germline stem cell likely directly decrease germline stem cell proliferation. And then we ask whether this phenomena also can be occur in different stem cell type. So we take the advantage of Drosophila intensity because they carry well characterized stem cells. We perform the glucose uptake assay. We examine RS level. We also examine junk signal level, and they all increase on the high sugar diet treatment, similar to what we observe from the ovary. Differently, high sugar diet decrease intestinal stem cell maintenance. In this image, green color mark intestinal stem cell and the progenitor. Without my explanation, you already can find that green cells are fewer under the high sugar diet treatment. So we disrupt junk signaling or remove RS by two different anti-oxidant genes, SOD1 and catalase. And what we found is that when we knock down this SOD1 or catalase, their maintenance, the percentage of progenitor and ISC, the number is already reduced, suggests that basal level of RS is important for ISC maintenance. But when we calculate under the high sugar diet, we did not see further reduction like what happened in the control group. We also disrupt junk signaling by using two different RNAi, and we got a similar result. When we disrupt them, they don't respond to high sugar diet. So this is the take-home message for you. It says dietary sugar impairs stem cell prior to insulin resistance, and RS junk signaling may be the common pathway for stem cell response to, it says, dietary sugar. And my solution for people who like eating sweet food is that you need to eat sweet food to make you happy, but you also need to have some food that contain high antioxidants. So at the end, I would like to thank my people. People who did the job are shown in red. And I would also like to thank our collaborator, Dr. P.I. Wei from Changchun University. She helped us to check male germline stem cells on the different diets. And she also found a consistent response, like the response is similar to female germline stem cells. And I would also like to thank our co-facility, funding resource, and the great flight community. Now I'm ready to take your question.
Thank you, Professor Xun. We have time for a couple questions.
I was wondering, when you switch to regular diet, I know after 7 days, the fat content decreased. But what happened after 1 day before the fat content decreased? Do you see a reduction, recovery of cell proliferation in the fat content? I'm just saying, because if the function is directly in the stem cells, then as soon as you switch diet, you should recover cell proliferation.
We did not do this experiment. That's an interesting point. I'll go back to the students to do.
Very nice work. Two questions. Have you actually beaten the price with the the RS scavenger like the NGC to see that they actually rescue the phenotype in the stem cell, also in the intestine, also in sugar?
We have tried to suppress RS production in mitochondria, and the germline stem cell proliferation seems to be rescued. the reason why I say seems to be rescued is because students just did one experiment. So later on, they will further confirm the result.
You use the genetic manipulation talking about.
No, we treat without as suppressor, but I couldn't remember the name right now.
The other thing is maybe you can elaborate a little bit. You overexpress SOD1, right? You can express it. So the SOD1 is converting the superoxide to the H2O2. So you still have the species actually triggering it. But then it's the H2O2. Why the SOD1 can actually maybe you can elaborate.
I see. So why only overexpress as OD1 can restore other RS species still
because SOD1 is not really reduced RS. I mean, it's converting the superoxide into the H2O2, but H2O2 is still RS.
I know what you mean. Well, we also did the catalyst expression in the joint, and we got a similar result that strong stem cell proliferation on the high sugar diet could be rescued. So it seems that both SOD1 overexpression and catalyst overexpression can reach to the same result. So I guess maybe this is some specific RS material does the job. I don't know the detail.
One quick, so two related, one question. So I enjoy your talk. So you try to manipulate the glucose uptake by indirectly by RNI insulin receptors. How penetrates RNI? It's actually knock down insulin receptor in those germ cells. One. Two is, I don't know about Drosophila in the workplace system, there are glucose transporters that some are insulin sensitive, others are not. What about the flies?
So what's the
glucose transporters? Some are insulin sensitive.
Okay.
Some are not regulated by insulin.
I see.
We have insulin sensitive glucose transporters.
Yes, we do have.
Not
I know. So the the penetration. Okay. So the penetration, because we also had the mutant line. So and previously we worked on how insulin signal control genesis. We found that this insulin RNA knockdown can phenocopy insulin receptor mutant in the germline. And we also did the qRT-PCR, is about 80% of reduction. So we think the penetration is quite good. And second, yes, there are some glucose transporters that are independent of the insulin. So yes, you are right. So when we did the RNA screen, we actually collect all the glucose transporter family. You know, they are a big family. Some are the insulin dependent, some are independent. However, we did not find anyone can reduce glucose uptake on the high sugar diet.
Great. Let's thank Professor Xu again. Our next speaker is Professor Wang Zhenzhe Jeff. He's from Auburn University in the United States, and he's going to talk to us about how to manipulate the adrenal cortex dysfunction, development, and aging.
Can you hear me? Yes. So first of all, I would like to thank President Zhang and Professor Zhu for inviting me to come here to give my talk here, to share my research with you. So today I'm going to talk about the adrenal gland. I'm going to share you 3 stories about the adrenal gland. One is about the function and the development and the aging of the adrenal gland. These 3 figures actually represent the 3 categories: the function, development, and aging of the adrenal gland. Okay. The signals you see here, those dots are oil droplets. Okay. Adrenal cortical cells use those oil droplets for cell adhesion. That's about the function of the adrenal. Those green cells are the cells coming from the outer part of the adrenal cortex. This is the image from the lineage tracing experiment. So you can see those green cells can cover the entire adrenal cortex. Those cells from the outer cortex move inward and replace the entire cortex over time throughout the lifetime of the organ. If this is true, what you can imagine is that in the inner cortex, those cells are relatively old. Those aged cells should express some marker genes that differ from the cells in the outer cortex. So these are the old cells. And those cells will undergo regression of apoptosis and will be replaced during aging by the cells from the outer cortex, right? And thyroid hormone can change them all. So let's start to talk about this one by one. The function. Again, those signals represent the oil droplets. Oil droplets contains cholesterol. Cholesterol can be used to synthesize steroid hormones. And the first step, this enzyme called CYP11A1. When I was a master student in Dr. Banju Jones' lab, we were studying the CYP11A1 knockout mice. Okay? As you can imagine, if there's no this enzyme, the cholesterol or the substrate cannot be converted into the product. The substrate will accumulate it in the cells, right? So this is the adrenal of the wild type mice and the CYP11A1 knockout mice. Those oil droplets in the knockout mice are bigger, right? Makes sense to you? So here comes the question. I told you that thyroid hormone can change the function of the adrenal. So what happens if we treat the mice with thyroid hormone? What happens to those oil droplets? Those oil droplets become bigger. And more interesting thing is that the enlarged oil droplets are mainly in the inner cortex. It's an area-specific pattern, right? So what we can say is that thyroid hormone treatment leads to increased lipid accumulation. All right? Why? We don't know that yet. But this is the phenotype that's very interesting. How about the development of the adrenal? pCNA labels the proliferating cells. Those proliferating cells are highly accumulated in the capsule, as well as the outer cortex underneath the capsule. That's because the capsule contains the stem cells, and the outer cortex contains the progenitor cells. Those stem and progenitor cells proliferate and differentiate and contribute to the functional cortex. When I was a PhD student in Humphrey Yale's lab, we were using Sony Hedgehog knockout mice to study the interaction between the capsule and the outer cortex. That is the Sony Hedgehog conditional knockout. So when I finally get a chance to establish my lab at Albert University, I asked my PhD student, hey, this is Sony Hedgehog. It is expressed in the outer cortex. That's the root trace itself. Okay, so what Sophia did was that she permanently used this lineage tracing model to permanently label the Sony Hedgehog positive cells. Those cells and their descendant cells will be labeled by GFP. All right. So she starts the labeling at postnatal 22 and postnatal 24 and analyze the tissue at 3 time points. First one. Those Sony Hedgehog positive cells and their descendant cells expand and make the Sony Hedgehog positive dome thicker and thicker over time. So what that means? It takes four months. It takes 4 months to replace the entire cortex. So the adrenal cortex is continuously renewing. So again, if this is true, the cells in different layers of the adrenal cortex should form concentric zones. Okay? Concentric zones. And each zone should express different marker genes, represent the different age of those cells. So this is a good example. The 20 alpha that actually see here in red color expressed in the innermost concentric zone of the adrenal cortex. Those cells are very old. On top of it, the CYP2F2 cells are younger. And actually, CYP2F2 is the marker genes that we identified in my lab, right? And you may say, hey, Jeff, could you give me more examples? Of course. AKR1B7, mainly expressed in the zona fasciculata. Okay? See? It's on top of the 20-alpha HSD. You cannot hear me? Okay. Right. Maybe put it here would be better. From the beginning? Are you kidding me? No. This is not right. Okay, let's move on. Go. Come on, almost there. All right. Lunchtime. Excellent. Thank you. So AKR1B7 expressed in the zona fasciculata, as you can see, it's not in the zona glomerulosa in the most outer cortex, and it's not in the most inner cortex. So there's a zonal specific expression of AKR1B7. Same to the 3-beta-HSD. 3-beta-HSD is a steroidogenic marker. The second step of the steroidogenesis pathway. So as I told you, the cells in the most innermost cortex, they are very old, right? They are dying. So they are not really functional. So that's why they don't express high level of 3-beta-HSD. Okay? What happens if we give them thyroid hormones? The inner cortex become expanded. But the most striking phenotype is that, do you see those yellow cells in the innermost cortex? Yellow means red plus green. Hey, the inner cortex cells, cortical cells, they don't express the AKR1B7, but by giving them thyroid hormone, now they start to express AKR1B7, the genes that they were expressed when they are young. Same thing here. The cells in the innermost cortex, right next to the medulla. They shouldn't express 3-beta-HSD because they are very old. But by giving them thyroid hormone, those old cells now start to express 3-beta-HSD again. So what's that mean? Thyroid hormone treatment makes the inner cortex young again. That's about the development. So how about the aging? Like what I just told you, the inner cortex or whatever the cortex, they'll undergo regression, will be replaced or renewed by the cells from the stem cells and the progenitor cells in the outer cortex. So this is the evidence. 20-alpha HSD cells in the innermost cortex, as time goes by, when they're getting old, become thinner, and disappear eventually. That's what happened in male mice. So what's going on in female? In female, those 20-alpha HSD cells also regressing over time, but in female, it takes very long time to regress, like 7 months or 8 months or even longer. The interesting thing is that if you let the female mice get pregnant, the 20-alpha HSD cells will regress immediately, like 2 or 3 days right after pregnancy. So there is a sexual dimorphic regression of the inner cortex. Again, as a male, regression is a normal regression when mice are getting old. What happens if we give them thyroid hormone? If we give them thyroid hormone from P14 to P28, inner cortex become expanded. If we give them from P21 to P35, we prevent them from dying. What's that mean? Thyroid hormone treatment changes the cell fate of the inner cortex. So here comes the question. What does thyroid hormone do to the adrenal gland? Or in another word, how does the thyroid hormone change the cell fate? That's the big question we would like to answer, right? But just like what we mentioned in previous talk, what thyroid hormone can do. Thyroid hormone changes genes expression, all right? This is a simplified model. Thyroid hormone binds the receptor and changes genes expression. To answer this question, what we can do is that we can do some RNA sequencing to find out what those genes are and to see whether those genes controls or evolved in the cell fate changed by thyroid hormone. Make sense to you? Unfortunately, the mechanism or the control of the thyroid hormone signaling is more complicated than we imagine. Okay, it has transporters, it has the iodinase, it has co-repressor or co-activator. When thyroid hormone level is low, what happened here is that the co-repressor will bind to the thyroid hormone receptor instead and actively suppress the gene expression. What's that mean? That means the thyroid hormone signaling relies on the homeostasis control by the co-repressor. It's a beautiful balance. Okay, thyroid hormone signaling is not just we have thyroid hormone, so we turn on something. No, when you have low level of thyroid hormone, active repression. So that means when we try to answer question 1, we don't want to ignore the possible effect from the co-repressor. Okay, so we are going to answer these 2 questions side by side at the same time. Let's start to answer the question 1. Like what I said, to understand the thyroid hormone effect, we can do RNAs. So we have saline treated group with T3 treated, very straightforward, right? By comparing these 2 groups, we have some T3 responsive genes. Very straightforward, very easy to understand. This is what happened in male. And then, of course, we need to do female too. And this is what happened in female. Female is more responsive to male. They have 2 times more genes. They are responsive to T. The beauty of this experimental setting is that we can simply compare the saline-treated group in male versus the saline-treated group in female, and then we come out with a gene, the list of the genes called male-dominant genes. Those genes are highly expressed in male. And of course, we will have female-dominant genes that are highly expressed in females. We overlap them. We call those overlapping genes sexually dimorphic T3 responsive genes. Make sense to you? Now we have a list of genes, 114 genes. We can do geo-analysis, gene ontology analysis. So what are those genes? Those genes link to lipid metabolism, cholesterol metabolic process, fatty acid, blah, 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 so on and so forth, about lipid metabolism. Just like what textbook says, right? Thyroid hormone controls the metabolism. How about question 2? The co-repressor. What we can do is to knock it out, right? NCOR1 is the major co-repressor we found in the adrenal cortex. So let's do a conditional knockout and knock out NCOR1. So we have wild type, we have NCOR1 conditional knockout mice. Again, we have male, again, we have females. That's the phenotype. Again, at P35 in wild type, the innermost cortex already regressed in male. However, in the conditional knockout mice, there are some CP2 cells remaining. In female, we see expanded or the stronger expression of those inner cortex markers. So what's that mean? If we don't have mTOR1, we see a delayed regression of the inner cortex. We see expanded inner cortex domain. These phenotypes, just like T3 treatment. It totally makes sense because mTOR1, the co-repressor, suppress the thyroid hormone signaling. So if we don't have the suppressor, it looks like T3 treatment. Bingo, beautiful model. Let's do some RNA-seq, right? Very straightforward stand-up procedure. We knock out something, we do an RNA-seq. And now we find 75 genes are differentially expressed when we compare the wild type versus the knockout mice, both in male and female. Hey, if you did RNA-seq, this is not normal that you only identify 75 genes, differentially expressed genes, right? Most of the time we identify hundreds of genes. 75, well, it seems like that's just too small to label, right? Maybe you even compare two wild type groups that you will identify 75 differentially expressed genes. Let's pull out those 75 genes and draw a heat map to see whether those genes are specific or not. Euthyroid condition, wild type versus knockout, wild type versus knockout. Like what I said, some genes are upregulated. When we put together with the thyroid hormone treatment group, bingo, those genes are also upregulated in thyroid hormone treatment group. So that means these 75 genes are specific enough. Even though we only identify 75 of them, but they are specific enough. They are controlled by thyroid hormone signaling, right? Similar to the T3 treatment, they are upregulated. But if you still remember about that, what I just mentioned, the homeostasis concept. The control of the thyroid hormone signaling requires the homeostasis. The reason we see an upregulation in this mTOR1 condition in knockout mice, that's because these mice, they have thyroid hormone in their body. They have thyroid hormone. That's why you see the upregulation when we knock out or remove the co-repressor. If we make those conditional knockout mice hypothyroid, what will happen? Theoretically, we should rescue this upregulation. Make sense to you? So we ask our PhD student, hey, let's include another group, so-called hypothyroid group. All right? Oh my God, there's too many. But she has no option, so she make it. This is the conditional knockout mice under hypothyroid condition. Okay. Do you see here? The upregulation now has been rescued. Okay. So what's that mean? That means those genes that we identified are very specific. So let's do a geoanalysis to see what are they, what those 53 genes represent. Number one, rank top one, cholesterol biosynthetic process. Cholesterol biosynthesis. Hey, that's interesting. Hey, Jeff, you just said the steroidogenesis use cholesterol. And from the textbook, those cholesterol are mainly come from the blood circulation. And if your geoanalysis says the top one geo term is cholesterol synthesis, does that mean adrenal gland can synthesize its own cholesterol? The answer is yes, okay? It has been known for a while that adrenal cortical cell can synthesize cholesterol locally. And this is the cholesterol synthesis process, all right? It's also an enzymatic process through several different processes, steps. So if that is the case, if that is true, if T3 or thyroid hormone regulates the de novo cholesterol synthesis in the adrenal gland, that means the key enzymes for cholesterol synthesis should be expressed in the adrenal gland. For example, like DHCR24 should be expressed in the adrenal gland. Right? Makes sense to you? And the answer is yes. DHCR24 is expressed in the adrenal gland, especially in the inner cortex. This is what we discovered nobody knows before. So what's next? Now we identify a new gene expressed in the adrenal gland very specifically, in a specific area. Let's knock it out. How about that? Okay. Does DHCR24 drive the T3-mediated cholesterol synthesis? To answer this question, let's knock it out. So we make a DHCR24 conditional knockout mice using a very similar strategy, SF1 cream. Okay, so SF1 cream can remove DHCR24 in adrenal cortical cells. Yes, we remove it. DHCR24 knockout mice, they don't express DHCR24 in the adrenal gland anymore. So what happened to those DHCR24 conditional knockout mice? Again, in wild type, if we give them thyroid hormone in male, you see the increased oil accumulation. In female, we see the increased oil accumulation. Female has better response to T3. So what happened in DHCR24 conditional knockout mice? This T3-mediated expansion of the inner cortex is partially blocked, especially in female. Partially blocked. This is the result from the DHCR24 condition. Do you still remember what happened in our NCO-R1 conditional knockout mice? We found cholesterol synthesis is the top GO term due to the lack of NCO-R1, right? How about in our thyroid hormone treated mice? The first project. In the thyroid hormone treated wild type mice, lipid metabolism is the top GO term. I hope in the past 25 minutes, I used 3 independent projects, 3 independent models to convince you that thyroid hormone can control the adrenal gland system metabolism through the upregulation of this cholesterol synthesis. And the most important or most interesting thing is that those phenotypes, it has sexual dimorphism. Female are more responsive to T. Female are more responsive to T. Of course, we haven't got a chance to give you the detailed mechanism, like how T3 changes the cell fate. But this is a good start. Give you some hint. Okay? And I would like to thank my current and former lab members and somebody from whatever institute, service provider, anyway, to give us money. Any questions?
Thank you very much for a very dynamic talk. I guess mentor. Okay, so 2 questions.
Yes.
Number 1, you have shown that DHCR24 mediates de novo synthesis of cholesterol in the adrenal. Have you gotten a chance to analyze whether it is the direct target gene, T3? That's number 1. Number 2, there seems to be a lot of dynamic changes in the cortex, outer cortex. Did you get a chance to look to determine whether TR has a different distribution among all the different layers?
Yes. For the first question, whether the DHCR24 knockout conditional knockout mice, we did the RNA-seq analysis, right? And we have some differentially expressed gene lists, but we haven't really looked into the details. But we did identify another candidate genes called SCD1. And that SCD1 is responsible for lipid metabolism, whatever, something like that. So maybe SCD1 with DHCR24 are both of them responsible for the phenotype we see here. For the second question, the answer is yes. Thyroid hormone receptor, especially thyroid hormone receptor beta 1, is specifically expressed in the inner cortex. So that's why the effect we see is zonal specific, because that expressed in the adrenal gland inner cortex. Only TR beta 1. Any other questions? Yes, please.
I just sort of have a wild guess or suggestion or thinking in my mind is that your sexual dimorphism with the female being more responsive to some treatment. Are there any possibility that there is heterodimerization between your thyroid receptor with the estrogen receptors and the balance between the amount of dimerization with the RXR and the RAS?
The short answer is that we believe so. That's the reason, yes.
Yes, so you identified CYP2F2 as the marker for old cortex.
That's correct.
Yeah. So is it involved in the degradation of the cells? So have you tested? You must have done it.
Yes.
Just to see how it's involved.
So like what I said, if we identify some novel genes, what we can do is to knock it out. And we did knock out CYP2F2, and unfortunately, there's nothing happened. So CYP2F2 knockout mice, they still have normal adrenal gland development.
If not, last one.
Yes, please.
I'm very interested in your sex dimorphism in response to T3 between male and female. So can you tell us what's the difference between male and female adrenal after response to T3? What exactly the difference? And whether, because the female has more T3 targeting gene compared to the male, whether those genes are also expressed in male, but it's not turned on.
To answer the difference between male adrenal and female adrenal gland, we did RNA-seq analysis to compare the male versus female from postnatal day 0 to postnatal day 49 every other week, not every week. So P0, P7, P14, blah, blah, so on and so forth. And what we found is that starting from P28, the female adrenal glands start to be different from the male adrenal. We don't know yet, but we have the data, the RNA-seq data show that they go to different directions. And yes, this will be the most important thing to identify what drives this difference as early as at postnatal day 28, right? Yes, testosterone already been higher at that stage, but we don't know that. We don't know whether testosterone is the primary driver. Maybe there is something else to drive the sexual dimorphism of the adrenal gland at that early stage. Or E2, yes, probably. All right.
All right. Thank you, Jeff.
Thank you.
We're going to switch here to Professor Li Qiying from the Department of Biology, National Changhua University, and he's going to talk to us about the control by the parasitic barnacle and of its brachion host mechanism and the potential application.
Yes. First of all, I'd like to thank Professor Chan and Professor for organizing this symposium. I learned a lot yesterday, today, up until now. I'll move the slide. Okay. Let's see. No. So I learned a lot from yesterday and today's talk. There's so much data and so much new findings that I think I update my vertebrate endocrinology since I give. So yeah, it's good to learn the new things. Okay, but before the lunch, I like to take a wild U-turn. Okay, directing your attention to the frustration system. Okay. So the system that we are working on is a system that involves brachiurins, host, right? Okay. And the picture show that there's some like egg-like tissue here. It's abdomen. Most of the time, you probably will think that it's an ovigerous crack over her own egg mates here. But it turned out that in this case, it's a female reproductive apparatus of the parasitic particle. So I'm talking about trying to talk about how the parasite, the parasitic particle, when they try to live inside the system of the host, how they control the host physiology. Okay, a little bit about the life history. Okay, so the history of a parasitic barnacle. Okay, it actually has a three living phase here. Okay, beginning with the lava released from the mother. Okay, so the lava released and then another stage of lava, and like other particles, okay, whether it's parasitic or non-parasitic, the seabreeze lava will settle on some surface. Okay, in this case, the parasitic particle they will settle on the soft tissue of the host, like the gill. Okay, and doing that, it will metamorpholize into another stage of lava. Okay, and that lava will inject with a spatialized structure, needle-like structure, which is called stylate, inject yet another stage of lava, which is called vertigo, okay, into the hemolymph. From there, the tissue will go first to the nerve tissue of the host and colonize there. Okay, from there the parasite tissue will proliferate. Okay, eventually the host hemolymph will actually occupied by the proliferate structure of parasite, okay, which is sometimes collectively known as internal. Okay. There's a network of tube-like structure called rootlet. Okay, that we believe that they will absorb the nutrients from the host, and in addition to that, probably will secrete some controlling factor that control the host. Okay, and after the maturation of the larva inside the host, the host, and the primordial reproductive tissue will mature. Okay, at the same time, and that reproductive tissue will emerge from inside the host to the outside and represented as external, just like the orange structure that I just show you. Okay. So that is actually the female reproductive apparatus, okay, from the female surface infect and parasite. within the host. And what makes the story more interesting is then after the emergence of external from inside, that then came the male sequence. Okay, it will enter the newly emerged external, the male sequence. And only the sperm producing tissues will finally stay inside the female reproductive open here. Okay. From there, it will produce sperm and fertilize the egg, produce here, okay. And then finally more larvae release, okay, when they are mature. Okay. So this completes the life cycle of the parasitic, typical parasitic barnacle. Okay. So this shows you the development of the external, which it's easy for us to determine the stage of the external by looking at external characteristics. Okay. For example, the color of the external. Okay. This is a newly emerged external. From here, it will turn into orange, yellow, and brown. We will call it creamy, orange, yellow, and brown external. Basically, what it did is from here, the oocyte will begin to make it to develop from this creamy, we call it virgin external, down to orange and yellow to be the present of the male signal without any male penetration into the external, the development of the external welfare. Okay, not going through the whole cycle. Okay, but in that way, when the male enter the external, then it begin the development of the external. And what happens is the ovary will begin to develop inside, okay? And there's a ovary duct that will move so that the mature oocyte will be released into a chamber. Actually, that was not shown by this external observation, but there's a chamber in the inside the external that will receive the ovulated oocyte and got fertilized by the sperm, and then the embryo will develop within that chamber. And that with the development of the embryo inside the external, the color will actually change to the brown color gradually. Okay. By the time it reaches this stage, the larvae is ready to be released. Okay. So yeah, maybe a short clip to show you the release of larvae from the external. Okay. So it's filmed in the night, so you can see the profile. Right? Here's the external, and there's the opening here. White spot actually is the larvae. When the external push, we contract and push the larvae into the and what attracts us to study this system is it has been known for a long time that the parasitic barnacle actually control many aspects of the host. So in the literature, you will be reading that the host will be sterile by the barnacle, and the male host will be feminine. Turn the genetic male into female with turning into individual with female characteristics. And there's also the control of it is called endocytes. Okay. host usually will undergoes a series of molting in order to grow. But as long as the host has the external, we say the abdominal region in the neighbor wall, so that people think that the barnacle control the host molting system, so that it stops it from molting. And there's a lack of immune response from host. Or weak, very weak immune response. And that started us as to be interested by the system, and the rationale for the parasite to control its host in this aspect are really it's easy to understand because you have a female parasites, female reproductive system here, and that represents that the parasite need to develop its reproductive system organ. Okay. So that put the parasite in competition with the host, right? In terms of reproductive development. So somehow it need to inhibit the development of the host, the natal system. The other thing is this one. The second reason is it's a female reproductive organ. So if you want to control the host by providing the resources for the parasite's development, well, it's probably no problem if the host is a female, right? The female will produce the resources, the molecule for its own maturation anyway. Okay, you just grab the resources from the host to the parasite. But what if the infected host is a male? You need to feminize it so that it will do the female reaction or provide the thing that female usually do. And another thing is you need to stop the molting of the host, which is obvious because the external is actually connected to the internal inside the animal. So once the host molt, that will disrupt the connection of the external, and so somehow you need to stop it from molting. Okay. And here it's the lack of immune system. This is the external, internal that I'm talking about. This is a section of the ovarian host, ovarian tissue. Okay. You can see the oocytes are arrested, okay, with a lot of roulette of the parasite that infiltrates ovary. Okay. And the prostration immune system really is very sensitive. If you have some pathogen that enter the system, normally the immune cells, the hemocyte will come around the pathogen surrounding it, which is a reaction called encapsulation. And then it will minimize the pathogen and then destroy it, remove it. But we've seen hundreds and hundreds of tissue sections that are infiltrated by the roulette. We rarely see any immune response. Right? So somehow they need to stop it from mounting immune system, immune response toward the infiltration of roulette. Okay. And there's a lot of things that could be studied, and we actually have data about these aspects of host control. Okay, but the main data that I want to show you are regarding the so-called sterilization and feminization. Okay. So here we go. There's a tissue sectioning of the parasitic animal and non-parasitized animal. And this is the animal's ovary, testes, and hepatopancreas, which is the tissue with the function of liver and pancreas. And as you can see clearly that the effect of the parasite on ovary is so severe. Okay, these are the arrested oocyte, developmentally arrested oocyte. These are the roulette that I just show you. Okay, so it's completely arrested in development. So oocyte development arrested. And here, this is compared to the non-parasitic ovary. Okay, got the mature oocyte. Okay, so but the situation in testing is different. Okay, because we can see the number of mature sperm produced in the host testing are at least comparable to the testing in the non-parasitic animal. Okay. Sometimes we can see some extra secretion with among the mature sperm, okay, but other than that, it's quite, at least from histological observation, the spermatogenesis remain intact. Okay. And we cannot see within a lot of testicular section, there's no roulette infiltration into this tissue compared to the ovary, where you can easily see a lot of roulette infiltration. And here again is the tissue, capital pancreas from both type of animal. Okay, these are the secretory lumen of the tissue. Okay. And you can see a lot of infiltration is heavily infiltrated by the roulette. Okay. The reason I want to show the result of this tissue is because this tissue is the tissue incrustation that produce metallogen. In addition to the ovary, incrustation, pancreas is the main tissue that produces metallogenesis. Okay. And I'm going to show you the data. Okay, the metallogenesis synthesis in ovary actually in the parasitic animal remain active. Okay. Although the oocyte are severely arrested developmentally, but the metallogenesis synthesis is still going. Okay, and that's the same. Okay, in hepatopancreas. Okay, they are active whether it's parasitic or non-parasitic. In fact, in both sexes. Okay, I'm going to the data now. Okay. So a little bit recap of the point that we just mentioned. Okay, sterilization occurs in female only, not male. Okay, and I think the reason is the competition between female host and parasite. It's because of that you have a need to mature the ovary for host and parasite, right? And for the parasite to control the host, you need to stop the development of the host ovarian maturation. Okay, so yeah, got to be stopped. Okay. And in terms of metallogenesis, which is the precursor protein for the formation of milk protein, metallin. Okay, so it's obvious that the host ovary produce metallogenesis still once it's got parasitic. Okay, but the oocyte did not develop. Okay, that make us to suggest that the host ovary will produce metallogenesis. Okay, all the same. Okay, but its own oocyte would not be able to pick up the metallogenesis. Okay, to be processed inside the oocyte for metallin production. Okay, and one thing is I think that's important is
infiltration by roulette is required for host control. The reason we have this conclusion is in testing there's no roulette, so the spermatogenesis remaining intact, but in the oocyte, the development of the oocyte in the ovary start there's a heavily infiltration. So we think that the presence of the roulette in the tissue probably is required, and that has some implication that we think that the roulette is going to produce parasite the right regulatory factor. Some chemically still unknown regulatory factor that control the host. I think that's the implication. Okay. Okay. Now we turn into the this feminization result. Okay, so we have the host sex is female and male tissue, and the parasitization status of the host is host of non-parasite. Okay, HMP host with orange external, male not parasite, orange, yellow external. As you can see that the expression of vitalogenins are present. Okay, whether the female are not non-parasitized or parasite, it didn't matter. Okay, so still going even with a parasite. Okay, for the male, the hepatopancreas did not express vitalogenin, okay. But once it got parasitized, the expression of vitalogenin begin. So that's one aspect of the so-called parasitic feminization. Okay. And the next one, okay. Well, the next question we ask is what the host, whether it's male or male, the host express vitalogenin. Okay. But how about the parasite itself? Okay. In fact, we can actually see that parasite express vitalogenin itself. So it's not only the host, the parasite also express vitalogenin. Okay. And this is the these three. Okay. These are the hepatopancreas of the host of the parasitized male. Okay. Parasitized female hepatopancreas and ovary. And the y-axis in all three figures are the expression label of the parasites, vitalogenin. As you can see that there's no obvious correlation between the parasites vitalogenin and host vitalogenin expression and in the hepatopancreas of female or the ovary, but there's a strong negative correlation, inversely correlated between the male hepatopancreas vitalogenin and parasites vitalogenin expression. And the explain what the next one we think we will do is well, that's a gene expression. How can we determine that the parasites vitality in the ovary contain vitalogenin, process vitalogenin from both host and parasite. And so we did this is the host vitalogenin protein with a three signature domain of the vitalogenin family. Vitalogenin N-terminal vitalogenin domain. DUF 1943 and the C-terminal VWP domain. We have the parasite sequence, the host vitalogenin sequence, parasite vitalogenin sequence, and the three signature domain. Okay. And then let's focus on the data here, the VNP, which is the parasite repellent. Okay, so that's the protein, that's the yolk protein we extracted from the external. Okay, and we take it, we purified it, and we digest it, we trypsin, so that there's many tryptase fragment, and then send it out for sequencing. Okay, using mass spectral photometry, okay? And we get the sequence of the tryptic fragment back, okay, and use the sequence to map to the host of vitellogenesis or parasite vitellogenesis sequence. So the results show that yes, the vitellant protein in parasite actually have sequence coming from host vitellogenesis. Also sequence, although the sequence number, the fragment number is lower, but yes, there are also some fragment that come from parasite vitellogenesis. So this concludes that yes, the yolk protein appear in external actually come from parasite and host vitellogenesis. And that has some implication, because when we usually, if you read the literature, we usually talk about host control. That means that the parasite is on the top of the commenting chain, although it will send a signal to you and produce vitellogenesis for me. But this data really show that in addition to that, parasite want additional layer of assurance that my ovarian maturation will be complete. And so in order to that, the parasite itself also produce vitellogenesis for vitellant formation. Okay, it's one layer of assurance. Okay, I know that. Yes. Okay. So yeah, that's the point that yeah, it's a parasitic fertilization, and the male level expression is negatively correlated with parasites, VG, okay? And both both host and parasite contribute to the parasite maturation. Okay? And finally, I think that it's just maybe give you some ideas is what caused the parasitic fertilization. Well, to make a story very short, is it's been known for a long time that station androgenic gland is the gland that determines the sex. It's important for the sex determination. Okay, that the presence of that gland will make the male male. Okay, so later on it has been shown that yes, the hormone produced by that gland is called insulin-like androgenic gland hormone, IAG, which is the reason why it's called insulin-like is because it belongs to the insulin's superfamily. It's chain A and B that coordinate three-dimensionally by some isocytol. Okay. It's there. There's a tissue very small here, and it's actually infiltrated by the ruling. Okay. Well, just give you an idea. The expression of the androgenic hormone actually is decreased significantly, decreased, almost shut down. Okay. Once the parasite got infected, okay. And that we think that the destruction of suppression of IAG actually remove the inhibition of this hormone, the inhibition of this male hormone on the synthesis of the vitellogenesis by the hepatometrix. Okay, so if you remove that inhibition, then the male begin to synthesize So yeah, this is just to some point that I just mentioned. Okay. Okay. So what the way we look at this study, I think the more perspective and this mechanism of host and parasite interaction for this parasitic problem are really the first time for any this animal and parasitic problem. Okay. And we think that the data that we have actually can provide suitable bioassay for our next step to purify and determine some putative parasite, the right regulatory effect. And in this case, we think that the PDR that inhibit expression of IAG by definition, by function, is a feminizing factor that we think might have some application in aquaculture. So yeah, this is the student postdoc from my lab, and I think Shicheng and Suli and Jianxin are the one produce the data that I report today. Okay, I thank them for their hard work, and I think the life is not easy, and for the penguin in the Antarctica base took this several years to go there, and life is not easy. Not easy for the barnacle that try to colonize inside the host tissue. Okay, so thank you very much for your attention. Okay.
Thank you, Dr. Lee. I don't think we have time for questions, but I'm sure Dr. Lee won't mind if we want to talk about crab and parasites over lunch. So very interesting work. Thank you very much. So, President, we are having lunch. Okay. Thank you. What does it say?
还有办法再开吗？
好像没有点到。
What molecular mechanisms that make germ cells conductive. Okay, thank you very much. So first of all, I thank the organizers and Professor Jin Chen and also Professor Yong-Soo for inviting me such a conference. Now, actually, I have visited Taiwan several times. This is the second time in Zealand, but I would say they found I was infected with COVID-19 months ago for the first time, but I'm fully recovered from it, and I'm not infected. But not as after effect. So please allow me. Okay, so let's get started. And today I'm talking about the mechanism that make germ cells dead. So to understand the mechanism, so we have been using Medaka. And ultimately now, the Medaka employed XY sex determination system. The sex determination gene, Y chromosomes here on the DMY determined male. And the next slide shows you a general scheme of gametogenesis. Here, the green cells and the upper part is spontaneous, the lower part is genesis. And you can see a lot of transformation, oocyte growth, maturation, and duration. And these events actually happen occurs in the late stage of oogenesis. And these events actually have been intensively analyzed. But my talk will be at the very early stage of gametogenesis here. And this is early stage of gametogenesis before meiosis. So the early stage of gametogenesis, germ cell divide mitotic, and it's now. And many years ago, so we identified germ stem cells. So many years ago, we identified the germ stem cells in the ovary for the first time in vertebrates. So now we know that many animals and many vertebrates possess germ stem cells in ovary and testes, which is different from mammalian cells, just germ stem cells in ovary. So the presence of germ stem cells in ovary and testes raises one question. So how the sex of the germ cell determined? How is the sexual switch towards the oogenesis spontaneously turned on? Because germ stem cells are sexually different. The sex of the germ stem cell is not determined, even if they are present in adult ovary and testes. So this would suggest that there should be some mechanism that determines the sex of the germ stem cells in ovary and testes continuously. So we wanted to address the mechanism of the germ stem cell transmission. So we did experiment, but no, I didn't go into the detail. But what we found is that so yeah, so this is the example of the indifferent germ stem cells. So this is a picture where germ cells are taken from the sperm testes and put them in the ovary. And you see the germ cell, the brown germ cell actually developing as oocyte. So this came from the germ cell came from testes, but they grew as oocyte. So that's why we are addressing that question. How germ sex of the germ cell is determined. And we actually did an experiment and what we found is that Fox's Professor Chan explained yesterday. But now the official name of FOX3 is FOXL2M. So because of new FOX3 come in based on the study of the phylogenetic analysis. But let me call in this we might talk FOXL3. The FOXL3 is expressed in certain population of germ stem cells, and also it is expressed in mitotically active germ cells before they enter during oogenesis. But during spermatogenesis, we can't see the expression of FOXL3 in mitotically active germ cells. Then the next question: what and we actually generate the mutant. So we disrupt the function of the gene in the ovary during oogenesis. What happens? Actually, oogenesis turned into spermatogenesis in the ovary. And this slide shows you a typical example of mutant focal mutant homozygous ovary. And this is actually histologically ovary. It is the typical structure of variant epithelium, which is indicated by star, which is separated by junction epithelium from stromal compartment. But you see many tiny dots. They are spark. The spark development inside the ovary. So these are sperms, and they are functional sperm. If you take out the sperm, and if you do artificial insemination, so you can get the next generation. And the next question is, what is Focal 3 is doing when germline stem cells meet the oogenesis? And we did transcript analysis and compare the wild-type germline, female germline stem cells, and mutant germline stem cells. To make the story short, what we found is that two factors, FBX047 and regulate A. So FBX47 is one of the components of ubiquitin D3 ligase, and it's recognized as substance. Regulate A is a major component of cohesion, which is involved in sister chromatid cohesion, homologous recombination, and DNA looping during meiosis. It's a very important factor to promote meiosis. And this factor actually activated directly by Focal3 transcription. So we actually identified 6 elements, 6 sequences experimentally, which recognized by Focal3. The sequence is A88C88. This 6 elements are found in the region of FBX047 gene and region and regulate A gene. And Focal3 recognized binds to the sister end in this gene, which is ruled by chip analysis. Then I'm going to show you the phenotype of these genes, phenotype of the mutants, to know the function of FBX047. So this is the result of homozygous mutants. So they are not lethal. And the XX and the XY mutants figure ovary and testis respectively. And ovary and testis, they look normal. Actually, testis, right panel, left panel, the testis produces functional sperm. And ovary looks normal over there. But you look at the cross section here, so you cannot see any follicles. You cannot see any follicles. And you can see expanding germline cells via germline stem cells are present. And if you look at the inside, on the right panel here, you see many dots. And we examine the how gametogenesis proceeds. For this purpose, we check the chromosome structure. Okay. The upper panel over there is chromosome structure in normal oogenesis. And the middle part is chromosome structure during normal spermatogenesis. So you can see zygoten, lacten, and diplotin chromosomes. And in the case of spermatogenesis, which are followed by second spermatocyte, spermatocyte. But chromosomes isolated from mutant ovary look more like chromosomes found in normal spermatogenesis. And if you look at the expression of spermatogenesis, gene down left panel. So you can see expression of the spontaneous gene. This is the example of SIC1. So we would say that the FBXO47 it acts as depression of spermatogenesis during oogenesis. Then also we will address more function of FBXO47. Then to do that, so we did epistatic analysis. Okay, in mammals, so several genes have been identified and functionally analyzed during late oogenesis. Okay. For example, LHX8 and the figure alpha is essential for follicle phase. In mammals, it is important to promote secondary oocyte development. And we generated the mutant of this gene using metadata. And we did, using this mutant, we did epistatic analysis. So it may be difficult without please look at the panels in the red box. And left column on left side shows the gene that express one. The FBXO47, LHX8B, and alpha are not detected in the ovary of FBXO47 mutant, no expression. But you can detect FBXO47 expression in the ovary of LHX8B mutant. We can detect the figure alpha and the box expression in the mutant. In the ovary of the figure alpha mutants, so you see the expression of FBX47 and LHXB, but we don't see expression. So this result indicates that the Fox LC actually activates FBX47. And FBX47 is important to promote follicular pathway by triggering this gene, FBX47, to figure out box within this. So the one row of so now we can say that on the row of the 3 is the follicular pathway. Next, I'd like to show you the phenotype of black weight A. Black weight A is not again embryonic lesion. The XY mutant and XX mutant develop testes and they look normal. But in the whole, the fertility rate is very low. The mutant testes produce plasma. On the contrary, again, class 7, we cannot see any follicle. Instead, we can see very weird aberrant type of marks. I think it's reasonable because regular day is a major component. Then, actually, we did chromosome examine the chromosome changes by prophase 1. And the rest one shows ovary. Ovary, you see a typical zygote, actin, different chromosome structure, obtained by SYCP, which is a element of the synaptic complex. On the other hand, the germ cell in the mutant, calculate the mutant, homozygous mutant, zygote seems to be okay. It shows a typical chromosome structure. But if you look at the actin-like cells, the signal is very rapid. So we would say that the meiosis does not proceed beyond the actin state. Again, then we did epistatic analysis. That's very interesting. And if this gene over there of the follicogenic pathway from FBXO47 to now, they actually detected in the ovary of liquid A complex mutant ovary. And liquid A expression over there can be detected if the ovary of FBO 47 mutants. This indicates that the two pathways, meiotic pathways and the follicular pathways, are genetically associable. The two pathways is genetically independent. Yes, so this is a summary slide, and FOXL3 triggers two pathways by activating the two pathways transcription. One is the pathway to genesis. The other one is pathway female specific meiosis. The phenotype is only in people. So regarding meiosis, it's a kind of surprise to us because we had aware of sexual difference in meiosis. But this finding really suggests that meiosis differs between oogenesis and spermatogenesis at the level of molecular mechanism. In addition, I actually showed FBXO47 mutant, so partial spermatogenesis. And I didn't go into the detail, but if we examine the mutant of LHX data, so they also showed partial, but mutants of the figure doesn't show anything. So this shows that FBXO47 and LHX act as the repression of the spermatogenesis during oogenesis. And this result suggests one more about feminization establishment of genes. In FOXL3 expression is a timing switch, when germinal stem cell enter oogenesis. But the feminization of the germ cell is not established yet at the initial time because these genes are deleted, then oogenesis turns into spermatogenesis. The feminization of the germ cell seems to be established before meiosis when figure alpha is expressing and FBX47 and LHXA are down. Then, so recently we found another pathway where microtubule is involved. So during the analysis of the germ cell transcriptome, so we noticed the sexual difference of microtubule components. So this is an example of GO analysis. Okay, so you see several terms of a microtubule. So we decided to visualize microtubule. And it may be hard to imagine the germ cell figure, but black region. So this slide shows the single germ stem cells. The black region is nucleus. And the green structure is a structure composed of microtubules. So we call it empty core, microtubule form, because it looks like a form. So empty core covers two dots over there, so can which is visualized by gamma cube. And now we know that we found that empty core structure is getting replaced with organic body at the late stage of meiosis, development stage. And what is about the body? The molecular body is represents oocyte polarity, which is surrounded by the mitochondria. here. And also maternal RNA deposit around the garden form. So the polarity is very essential. Oocyte polarity is one of the major characteristics. The oocyte polarity is essential for a three-dimensional formation of the axis in the development, embryonic development after fertilization. So we are thinking that the formation of antiphon shows the process of oocyte polarity. So polarity is very important character of the oocyte. And here, so these are germ cells during oogenesis. And you see empty construction things like green. So this is stem cells, and when germ cell make germ stem cell to the oogenesis, germ stem cell mitotically divide to produce daughter cells. And the daughter cells are connected to each other by intercellular bridge. And you see the microtube extending from the antiphon actually connects the daughter cells. And this one shows the germ cell during genesis, and you can see the antiphon structure in the mitotically divided germs. And we want to see the molecular mechanism that causes the sexual difference of the antiphon. And as I said about DMY at the beginning, so Medaka has a big white. And we know that DMY activates the transcription of DMRT1. DMRT1 is very essential for masculinization of somatic cells. And in germ cell, FOXL3 is expressed for fertilization. And first, we did examine relation of FOXL3, which is expressed in germ cell, and DMRT1 expression in smart cell, male smart cell. Then we check the expression of FOXL3 in DMRT1. And left panel shows the FOXL3 expression in the developing ovaries. So you can see red and brown signal for the expression of FOXL3. And no expression at all during developing testicular development. But in the absence of DMRT1, you will see again red signal over there. So this indicates that some signal from DMRT1 expressing small cells are from germ cell, the repressive expression of the 3 during spermatogenesis. Then we examine empty construction using mutant. Again, so you can see empty construction over there, the left, most left, and the next to the left. So you see germ cell during spermatogenesis. See any construction. But mutant of FOXL3 in this structure still maintain. This is contrary to our first initial expectation because FOXL3 is very important for oogenesis. And not FOXL3 might use the this structure, but we can see the structure. In the absence of the DMRT1 with FOXL3, germ cell genesis, but again we can see the construction in the germ cell genesis. This slide, this result shows that DMRT1 signal from DMRT1 expressing cells does not only repress the FOXL3 expression, but also promote the degradation of MT4. Okay, so this is a summary slide that we found that a new pathway leading to oocyte plurality here. And this is independent of two pathways over the nucleic pathway and the nucleic pathway. So this during spontogenesis, some signal DMRP1 replaces germinal stem cells in the replaces FOXL3 in the germinal stem cells, and also replaces degradation during spermatogenesis. That was the sexual difference between tests. And going back to my first question, what makes germ cell eggs? So we are now thinking that eggs may be made of genetically independent which are empowered. And we call this genetically pathway module because module can be defined as a unit of series of things that leads to major specific help to become oocyte or to become egg. Egg has to have oocyte cap. So for example, over there, polyglotinic pathway, the FBX of 47 to about is one series of a unit of gene that leads to folic. Again, blood population together with biotic gene promote female specific meiosis. The module is genetically independent. That's very important. So this means that if one module is deleted or wrong with a module, then another module can function in parallel independently. And we are thinking that this concept of the module might explain the diversification of the reproductive system. So I don't go into the detail, but this is just a hypothesis. But it could be explained. So for example, here, if the biotic module is deleted during evolution for some reason, then we can possible to speculate that polyglotinous module acts independent of meiosis, which leads to very potent activation of potent genes of mixes because it produces it should produce diploid. And if polyglotinous and spermatogenesis, that are very important to call the sexual dimorphism. They are deleted, so it's possible to speculate that one independent, which might lead to us. So we are thinking that module may be very important to understand the diversity of the reproductive system, and also important to understand how X is made. Okay, so then I'd like to totally change my topic because this is I found this endocrinology. So I want to explain previous 621 to 21. So 621 is very essential enzyme for the production of the cortisol. And the lack of the cortisol is a major cause, one of the major causes of congenital hyperplasia in human body, which is frequently associated with polycystic ovary syndrome. The our mutant has 9 base insertion and exon 1 can result in 1. And the mutant is not the embryonic lethal. It's not embryonic lethal, but it shows a delay of health over there, right blue lines over there. So delay a couple of days. But once the larva hatch, then larva die gradually. And the survival rate is very low in the mutants. Only 10% don't survive 1 or 1.5 after hatch. That's the time when they are mature. And CH, congenital adrenal hyperplasia, often associated with hyperplasia pituitary and adrenal. The same with mutant. Over there, so if you look at the pituitary stained by Ponca expressing cells, Ponca is a precursor of ACTH, and you see the expansion area over there, 621. And this is the appearance of interrenal excess in XY, and you can see the hyperplastic renal. Interrenal is equivalent to the mammalian renal. You can see hyperplastic renal. And we confirmed the lack of cortisol in the mutant. Here, the graphs down below are the level of different steroids. And the blue bars of the heterozygous mutant. The light bars show the level of homozygous mutant. And if you look at the cortisol and the cortisol, we cannot detect the cortisol mutant. Instead, so we can see the accumulation of a precursor steroid like 17 alpha hydroxyprogesterone and 21 deoxyprogesterone. And actually, the cortisol, additional cortisol, rescue the phenotype of type 1. We put the eggs in the water containing cortisol from 2 dPA 2 days after 14 days to the hatching time 7 days after 14 days. And then the hyperplastic pituitary is was recovered. The size of the spacing area is small as the heterozygous mutant. The same with the size of the renal. The size of the measured by 621 area becomes as small as the heterozygous. And lastly, so I'm going to tell you the phenotype of production. So although the survival rate is very low, the adult homozygous mutant show a normal secondary characteristic, and also homozygous mutant show ovary. It looks normal, but if you look at the inside, so the follicogenesis are very much disorganized. And consistent with this result, the homozygous mutant never spawn. Okay. And we want to know what happened to follicular growth or saturation. This is oocyte growth during normal oogenesis. And Medaka spawns egg every day with the onset of the night that is shown time 0. So Medaka experience 10 days, 10 hours dark before onset of the night, which follows 14 days hours dark. And if you sample the largest follicles at a different time point, you see the growth of follicles, and 5 hours before onset of the light, the pink area. So you can see the maturation happened at the time 5 hours before onset of the light, which is followed by ovulation and spawning. And if you look at the mutant follicles, you see several lines of oocyte growth. Actually, oocyte growth occurs in the homozygous mutant, but you see multiple time point of oocyte maturation. The maturation also occurs in the but ovulation never happens. So this is confirmed by individual experiment. If you take up the largest follicle 3 hours before onset of the light and put them in the incubation media and see what happens to the follicles. And here, these are taken from normal ovary, and you see spontaneous, Then lastly, I'd like to show you pathogenic activation. So the follicle never appeared, but oocyte begins to develop without any fertilization inside the follicle. So this slide shows you 2 cell state without any fertilization. So we are analyzing the action of the food and so we are thinking that the metabolic mutant is a good disease model for analyzing oxygen protein, and the mutant is good providing good tool to analyze a role of cortisol that has not been yet very level. Okay, so I like to close my lastly, so I like to acknowledge the people, the medical teaching, who is assistant professor. She has been working on the pathway created by OCC. And also current PhD student who is working on the mutant together with Professor Ho Choi in case Korea. And also the represent all the members. Okay, thank you very much.
Thank you for a great talk. After about 3-4 minutes for questions.
So I hope very interesting. So do you know how does control oocyte? This is deliver or transport the oocyte to the oocyte, or it actually does something to mitochondria and that produce energy from the oocyte.
Okay, so I didn't talk about the detail, but there are the cytochrome reason why the body is very different. Mitochondria or some other reached in the region, but present in the also expectation, another evidence of the absence of based on so that's why answer your question.
Actually, I have a very similar question. So the image actually reminded me about insulin spectrums and software.
Yeah.
So wondering whether they actually have a function for the transport. Are they actually transport things?
Yeah, that's very interesting question. So I know the case of the Safra, so through feed zone, so the any protein or something like that. And we are not sure. We are not sure actually. But it's very possible. It's very possible. Because I said that the germline something might divide my totally and produce many cells. But it seems like the loss of several of the page become impossible. So that might suggest some similar mechanical metabolisms. The CIP so I don't know the expression of the nuclear receptor, but 17 alpha 28 beta actually can escape. And also progesterone is produced. So we actually control the expression of these groups.
Thank you. Next speaker is Dr. Swinton Jun from Brazil. He'll be talking about screening through steroidal receptor.
It's good. Okay. All right. Okay. I'm also like to echo everybody's point about it's really a great pleasure to actually come to this meeting. It's very nice personalities. I also would like to thank Dr. Tu and Dr. Chang to invite me to actually share our here. So my lab is interested in reproduction and we use the population model. So we're going to shift gear a little bit to talk about ovulation and how we can use ovulation in the population to actually screen the several All right. So as we all know that we have very effective contraceptions for females, but they still have a lot of challenge in the field. That more than 40% of the pregnancy are actually unintended globally, and there's more than 200 million women in developing countries have unmet needs for contraceptions. So largely because that the side effect of the hormone-based contraceptions are the leading cause for the non-use and discontinuations. So I think we're in need to get a novel contraceptive methodologies in order to alleviate some of the burdens in the society burdens. So if we instead of using the steroid-based to inhibit pituitary gonadal axis for the current contraceptions, we're thinking about whether we can inhibit ovulation directly with that
could act as a potential ways to serve as a contraceptions. However, we know that ovulation in mammals or in humans are actually very complex. So I have here quickly summarize what we already know about ovulation in mammals. So we know that the LH surge is triggered ovulation that ultimately lead to three major events happening in the mature follicles. One is the meiosis resumptions, and then second, cumulus expansions, and ultimately the follicle rupture. So that lead to the oocyte release into the into the oviduct or into the abdominal cavities. So we know much about the meiosis resumption and cumulus expansions. However, follicle rupture is actually fully understood. So it's been largely agreed that LH that lead to two major pathways for the follicle rupture. One is the prostaglandin signaling pathway. The other one is the progesterone signaling pathway that ultimately lead to the activation specific protease in the apex regions. So that's going to break down the follicle wall and allow the oocyte to release. So that's the rupture process. And that's actually been demonstrated by long time ago by Tom Curry's lab, showing that gelatinous activity is very specifically activated in those apex regions, just right before the follicle ruptures. However, in mammals, like in mice, there's 24 MMPs, and it's very hard to study which MMP is involved and how they precisely activate in these regions. So my lab is utilize the Drosophila as a model systems. Thanks for the Dr. Xu in the mornings actually give a nice introduction about the Drosophila reproductive systems. So here I quickly remind you that in the Drosophila females, there's in the abdominal regions basically mainly occupy the female reproductive system. It contains two ovaries and connected by the oviduct, and then there's uterus, and there's also some accessory glands. So in the ovaries, they are organized into called ovarian structures. So these are the egg assembly lines, and the germline stem cells and follicle stem cells located into the germarium that proliferate, give rise to the daughter cells and form the early stage follicle or stage 1 follicles. So which will go through 14 different stages into for the mature follicles, in which there's an oocyte wrapped by a layer of somatic cells. So why we use Drosophila? Because Drosophila actually has a very high capacity for the reproductions. One single female can lay about 70 eggs a day. So there's a lot of ovulation events for us to actually study this process. So here I'll quickly summarize what we actually know about ovulation in Drosophila. So what we found is that the MMP activity or MMP2 is very specifically expressed in those posterior follicle regions. And then there's the octopamine receptor is expressed in those cells, and the neuronal octopamine release into those regions that activate the OAMB receptors that trigger the MMP2 activation. So that MMP2 ultimately lead to the follicle wall breakdown in the posterior. So then the oocyte will release into the oviduct, and the remaining follicles are actually form yellowish bodies that residue at the end of the ovary. So we call it Drosophila obsludium. So through these studies, we actually develop ex vivo assays that we can recapture the entire follicle rupture process. So what we do is we use the fluorescent proteins to label those mature follicles, the somatic cells in the mature follicles. So then we can isolate those intact mature follicles and put in the culture dish. So if octopamine is equivalent to the norepinephrine in mammals, if octopamine can induce the entire process, then you should be able to see it in vitro, in the culture dish. So that's exactly what we did. So here we added away 20 minutes later, you can see that the posterior cells are actually breaking down. So the signal is diminished. and then the rest of the cells are actually squeezed toward the anteriors. So the egg is extruded out from the follicles. And this actually become very useful tools for us to study ovulations, because we can do genetic screens to find out what gene is involved in this process, and we can also do the drug screens. So here I show you an example that we knocking down the receptor, the androgenetic receptor, OAB, in those follicle cells, and then we trying to look whether they can respond to the OA signals. They just don't do anything. So which we clearly demonstrate that OA activate OAMB receptor, it's G protein coupled receptor, so that mediate this entire process. So we also did unbiased genetic screens, and then we try to study the downstream signaling pathway of the androgenetic signaling. And we found one of the very important enzymes actually called NOx. It's an NADPH oxidase. So you can see we're doing screens, and we found that, in the controls, you have a lot of follicle ruptures, 60% of follicle rupture, but in the NOx knocking down, the rupture rate is significantly dropped. So we also show that this enzyme is very specifically expressed in the mature follicle cells, not in the younger follicles. So even one stage before that. So this is actually done by my former postdoc, Wei Li, in the lab. So what she further trying to characterize is whether NOx is indeed functions as an enzyme to in the follicle cells. So as I showed that NOx is an NADPH oxidase that will convert oxygen into superoxide. And then superoxide can be further converted into the hydrogen peroxide through the superoxide diffusers, SOD. So then we'll be converting into the water through the catalyst. So what we want to see is whether NOx is actually active to produce the superoxide. So we use the dihydroxyethylene to actually label the superoxide or detect superoxide. So what you can see is that in the controls, wild type, so without OA stimulation, there's no signals. And then in the stage 13 and stage 14, there's 14 slightly have signal, but it's very minimal. But when you have OA stimulation, you have huge increase of the signals. So which indicate that OA is indeed to induce the superoxide production. And if you knock down the NOx using the ion interference, you can see the significant reduction of the DHE signals. So we want to further characterize and using a more quantitative ways to actually detect superoxide. So we use one of the probe called AIR-012. This is actually pretty specific to be oxidized by the superoxide, and it will eliminate. It will have the luminescent signals, so we can detect use the plate reader. So what you can see that in the OA treatment, you have a huge increase of the superoxide signals, but if you get rid of the receptors, you have a significant drop. And the same is true in the NOx INI knocking down case. So basically, what we determined is that the OA activate the receptors and actually lead to the calcium influx through a store-operated calcium pathway that activate NOx and that convert to the oxygen into the superoxide. And we also demonstrate that it's not superoxide actually important for the follicle rupture, but rather it's the H2O2 in this process. And we also show that OAMB activate calciums that lead to the MMP2 activations and the follicle ruptures. So just as a note that reactive oxygen species it's already been demonstrated in the mouse ovulation, and it's critical for the ovulation process. But we don't know where the ROS actually come from. So from our study, we think maybe it's a NOx is also involved in the mammary systems. So we also did other genetic screens, and we found, for the sake of times, this is actually unpublished work. So what we are trying to understand is what is the mechanical force for the follicle rupture process. where you can clearly see the separation of the cells from the oocyte. So what we found is that OAMB signaling actually lead to the active myosin enrichment in the follicle cell cortex. And we show that OAMB through the calcium that lead to the Rho1 activation. And the Rho1 lead to the Rho kinase activate that lead to the active myosin contractility that create the mechanical force to actually separate those layers. So if you're interested, we can discuss afterwards. But anyway, I just want to summarize that here's our current model for the ovulation. So basically, we show that in the posterior, the androgenetic signal activate the MMP2 that lead to the follicle cell breakdowns, and in the remaining cells that activate superoxide that also contribute to follicle ruptures. And it's also activate, the Rho1 and active myosin contractility for the follicle ruptures. So through these studies, we actually found there's multiple components or signaling pathway are actually conserved compared to the mammalian ovulation. For example, the matrix metaprotease, we show the MMP2 is involved, and the steroid signaling, like in flies, we have the active steroids. We don't have the progesterone or estrogens. We only have the active steroid signal. So it's also important in the late follicles. And now I show the ROS is involved, and we also previously showed the 5A family components are involved in the ovulation process. So anyway, in terms of androgenetic signaling, it's actually very so in the Drosophila, we clearly show that local androgenetic signaling is important for the follicle ruptures. However, in the mammalian systems, it's very controversial. So some work showing they are involved, some work showing they are not involved. So I hope I can provide some evidence in the later talk and show you that it most likely it's involved in the mammalian population as well. So the real question is, can we actually use Drosophila as these systems to screen the compound that can inhibit ovulation, and whether those compounds can inhibit mammalian ovulation? So we're trying to use this to actually to study, did a proof of concept studies. So we use these assays to actually screen the compound. We put the drugs into those mediums and see whether they can inhibit the ruptures. So we used a small library, the FDA approved the library to do the screen. So we screen about 1,000 drugs from the libraries, and we get 6 of the compounds are showing effective dose-dependent inhibition of this follicle ruptures. So this is done by my former technician, Kiwa Jones. Then the question is whether these compounds can inhibit mammalian ovulation or follicle ruptures. So we actually collaborate with the group in the Northwestern. So we tested the 4 of the compounds and using the system that they developed. It's called in vitro follicle maturation, the ovulation assays. So what they did is you isolate secondary follicles from the immature mouse ovary, and then you encapsulate in the alternate beads and culture them for 7 days into the mature antral follicles. And then you use the HCG to induce ovulation. So we use these assays to see whether these drugs can actually inhibit the mouse ovulations. So we tested 4 of them. Actually, 3 of them are showing significant inhibition, but although some of them are much more effective, some are less effective. So then the reviewer asked, how do we know that they are actually effective in vivos, right? These are just the in vitro studies. So then we try to test one of the compounds. It's called chlorpromazine. So chlorpromazine actually been studied previously, and it's recently been characterized as a reproductive toxicant by the Netherlands councils. So we focus on the chlorpromazine because the chlorpromazine actually showing the can inhibit ovulation previously through some of the previous work. And what they showed is that chlorpromazine actually inhibit LH surge that lead to the ovulation defect. But from our work, it's clearly showing that chlorpromazine can directly inhibit ovulation in the follicles. So in order to test this, we use the superovulation assays through collaboration with store shots from the Rutgers. So what we did is we treated the 21-day immature mice with the PMSG. After 45 hours, we inject the drugs, and then 1 hour later, we use HCG to induce the superovulation. And 16 hours later, we collect the oocyte in the oviduct so we can count how many oocytes actually ovulated. So what you can see that 5 milligram per kilogram of chlorpromazine are effective to inhibit the ovulation process. We also dissect the ovaries and did a histological analysis, and you can clearly see there's a lot of follicle the unruptured follicles or the follicles with oocytes inside. So that's clearly demonstrate that chlorpromazine can inhibit ovulations in mice. So then we wondered, what is the target of chlorpromazine? How they inhibit this process. So in order to do that, we use our Drosophila systems. So from the study, we actually found several ways to induce the ovulation process. So one of the ways we can bypass the upstream signals just using the ionomycins to directly, influx the calciums into the follicle cells, and whether they actually can very efficiently to induce rupture. The other way, we also find an agonist. It's the alpha-2 agonist from the humans that can very potently to induce the OAMB signals to activate this process and induce ruptures. So what we're trying to do is we first look at whether chlorpromazine inhibit MMP activities or whether they inhibit downstream components. So we use for the gelatinous assays. So what we did is, so here in the control DMSO, we added OA, and you can see a lot of follicles have gelatinous activities. However, in the chlorpromazine treated follicles, the OA additions is not activated MMP2. So which means MMP2 activity is being brought by the chlorpromazine. So then next we want to also look at the calciums. Due to the time I didn't show the data, the calcium rise is also inhibited by the chlorpromazine. So that leads us to consider that chlorpromazine is acting around the calcium rise or upstream. So then we use the ionomycin to induce the rupture. But you can see that in the chlorpromazine treated, ionomycin is not able to inhibit rupture anymore. So which means that chlorpromazine is working upstream of calcium. And we also use the dexmedetomidine to induce the ruptures, and then the chlorpromazine cannot inhibit it. So that leads us to propose that chlorpromazine is actually inhibit OAMBs. So then we wondered whether this is also true in the mammals. So the first way we tested whether chlorpromazine can inhibit MMP activities in the mouse follicle as well. So we did a similar experiment. We, used the superovulation to treated the mice, and then we dissected the follicles before the ovulation and see whether there's MMP activity being affected. So what you can see is that in the controls, we can recapitulate the previous work showing that MMP activity is localized in the apex region before the ruptures. However, in the chlorpromazine treated animals, the MMP activity is significantly disrupted. So that leads us to showing that the chlorpromazine actually did similar things in the Drosophila mice to inhibit the MMP activity or gelatinous activity. And we also find another compound that's actually very similar to the chlorpromazine, that they're doing exactly the same thing. They can inhibit the mouse Drosophila and the mouse ovulation, and it's also affecting MMPs. Maybe I'll skip this one. We also basically what we want to propose here is that the norepinephrine is actually being shown that is highly enriched the human follicles before the ovulation. It's much higher than the serum norepinephrine. And also norepinephrine-treated granulosa cells are actually increased RS production. So we think this is actually very similar between the fry and mammals. So what I want to, maybe I want to skip this. So basically what we want to do is we want to increase the throughput, and we actually use the two-step assays. Basically, we use the primary assays to use the luciferase to screen the compound that can inhibit the luciferase productions. And then we use the follicle ruptures to actually doing the validation. So anyway, we screened about 2 different libraries and about 20,000 compounds using these 2 assays. And we get roughly about 40 to 50 compounds that can be inhibit the follicle ruptures. And we are doing the validation in the mouse follicle ruptures. So here's some of the dose response. Sorry. So we actually validated some of the compounds use the mouse follicle assays, and we're showing that about 3 out of 10 from the diverse set showing the inhibition, and 10 of the 29 from the reframe libraries are showing the inhibition. So we're actually currently trying to validate using the mouse in vivo systems, and we're also interested to identify the target and then doing the medicinal chemistries to improve the compound potencies. Right. With that, I just want to thank all the people who did the work. Ping Huang is actually the major driving force. He's posting the lab, and my collaborators, Mr. Wujia from Northwestern and the Suoshao from the broadcast. Thank you very much.
Thank you, Dr. Time for a few, 1 or 2 short questions.
Thank you. So conceptually, I'm a bit confused about the issue of a ovulation inhibitor, because with the steroidal treatment, contraceptive, take it every single day, you don't have to think about it. With the ovulation inhibitor, you kind of have to know when ovulation occurs.
Not necessary, because if the compound has no side effect, you can take every day. But the real purpose is that maybe we don't need to take every day. You just need to take 1 week before before the ovulation will be back. That will be very future in this far away.
Questions?
So to
Hypomanic,
so they have similar structure, similar mechanism, similar
exactly. So they are actually doing exactly the same thing, but hypomanic is actually more potent. I didn't point out. It's about 10-fold more potent than the chlorpromazine, and it's less toxic, less side effect, because all these are the antipsychotic drugs. It's actually FDA approved the drugs, but we don't want those side effects. So that's why we want to do the medicinal chemistries to try to see whether we can block them to get into the proper areas and also increase the potency.
Thank you, Dr. So now we move to the next speaker, Dr. ISOCCE come from USA. He spoke is about the antimicrobial peptides in beetles.
First of all, I'd like to thank President Qin Zhang and Professor for the invitation to these wonderful venues. And most importantly, this is the first time to visit Taiwan after this seminar. Today, I'm going to talk about a different story on how how the neuropeptide regulating the innate immunity in the insect. Unlike the vertebrates, the insects are encased in a semi-rigid exoskeleton, which can provide support for muscle attachment and also protect the internal organ from the physical damage and also the microbial infection. And this is similar to other osteopath too, like tick. In order to survive or growth, insects must periodically shed off their old exoskeleton and form a new one, which will allow them to grow. And this process is called molting, and it's regulated by a series hormones. And today I'm going to only talk about the last hormone released when the adults emerge from the pubic case. And we can see from this slide, offer and the newly emerged adults, and with folded wings and a small body, untanned cuticle, and usually about 5 minutes after emergence, the wing expands and the cuticle starting to harden and darken. That's for the tanning process. And this process is regulated by a neuropeptide called bursicon, which means it's a tanning hormone. In the Drosophila, the tanning hormone is first identified in 1960s in the blue flies using a simple but elegant neck ligation assay using the dental process. In the invertebrate, these insects, particularly the Drosophila, when you ligate it, they can survive for up to 3 weeks. Human, 5 minutes, for vertebrates, about that. And when neck ligate fly, newly emerged flies, which could prevent on the bottom, prevent the cuticle tanning. And this insect cannot barely walk, cannot fly, because the cuticle is so soft. Then they found out dissect the central nervous system, then homogenize, and then get it open and inject about a quarter brain equivalent into neck-like untanned insect, and the cuticle become tanned. So the conclusion is must be some factor from the nervous system regulating this cuticle tanning process. And similarly, when you collect the hemolymph from the newly emerged adults, which within like 1 hour, which already contain these neurofactors releasing hemolymph, and which also can induce cuticle tanning. Now we know this is a neuropeptide, and if you inject with the recombinant protein, which can induce the cuticle tanning process. And this has been demonstrated in many insect species. Now we know this tanning hormone actually is a heterodimer, consists of bursicon, what's called a burst, and also the partner of bursicon, the P-burst. And they bind into the G protein coupled receptor in Drosophila, and then activate adenocyclase, which is going to eventually activate protein kinase A. The activated protein kinase A is going to phosphorylate a key enzyme for the tyrosine hydroxylase. And this activated tyrosine hydroxylase is going to convert tyrosine into a group, amino acid tyrosine into a dopa, which is a precursor of the tanning hormone, a tanning agent. Then eventually they're going to cross-link the protein with the chitin and harden the cuticle. So this tannin hormone actually is synthesized in the central nervous system, and as you can see on the left, and in the Drosophila and also horsefly, and it synthesized both subunits synthesize the same set of neurons in the thoracic and abdominal ganglia. And the bursicon actually belongs to a cysteine non-protein family, which contains 6 subgroups. I'm not going to read it, but the bursicon is falling into the mucin-like subgroup, and that group contains the bone morphogenesis hormones, which regulate the bone genesis, and the bursicon is regulating exoskeleton, which also kind of a bone. So they are very similar. And this family of protein, they can form a heterodimer and also homodimer to regulate different biological function by binding to a different receptors. Now we know the bursicon binding to the G-protein coupled receptor and regulate the follicular tannin. However, most recently, there's somebody found out actually the bursicon homodimer bursts and can be expressed in midgut stem cells. And if you knock down this alpha or burst homodimer in the middle gut stem cell, what happens is you can see from here it is Y type, and it is the knocked out. Okay, both. When you knock down the bursicon homodimer, the stem cell is going to proliferate further. That means suggesting is bursicon homodimer in the middle gut stem cell going to regulate the cell proliferation rate. And also, there's the same group also found that bursicon homodimer in the middle gut stem cell also regulated energetic homeostasis. So with all these different functions, we are focused on the other new novel function from this neuropeptide. Previously, in last meeting, we found that the bursicon homodimer is going to regulate the antimicrobial peptide synthesis in the fat body of the Drosophila. And then that synthesis regulated through this reddish factor, reddish. And then we were wondering, because the IMG pathway is regulated by gram-negative bacteria. When you infect with gram-negative bacteria, and then will go through this pathway, binding to receptor and a several transcriptional factor, then eventually regulate the reddish and leading to the antimicrobial peptide synthesis. Then we order the mutant for IND and it's red. And this mutant has no effect on bursicon induced antimicrobial peptide synthesis. So we conclude the bursicon cannot bind into this bacterial receptor, glycoprotein receptor, to induce AMP expression. They must have some different receptor. We also order the G-protein coupled receptor DHGR2, a mutant, which is no mutant, and also has no effect on bursicon-induced AMP expression. So then we hypothesize that time also have some new novel unknown receptor exists in the Drosophila. In the insects, there's 3 major pathways regulating immunity. The TOR pathway, IMG pathway, and the JAK cell pathway. And each pathway has their distinguished marker gene. You can check which pathway is upregulated. So we wanted to know whether this Versicon can induce, we already shown previously they can induce RNA pathway. But we wanted to know if they also can induce the full pathway and eject side pathways. To understand that, what we did is we first to knock down the Versicon homodimers and using the red flower beetle, which is a model insect, and thanks for its robotic RNAi response. To knock down, we first investigate the Versicon expression, particularly beta Versicon, pattern of the Versicon expression profile. And as you can see, in the lobby stage, the Versicon basically is expressed highly in the newly emergent adults, which can regulate critical attention, right? But when we check the expression profile, and you find that they express a lot of different states. Yeah. So we use this day 18 lobby to inject the dsRNA, and because relative expression is low, then we check the efficiency. And as you can see, different dose of the double-strand RNA, and you can receive very high, up to 70% of efficiency, knock down the targeted gene. And a temporal response, and then we use 100 nanogram dsRNA and inject into the insect, and then collect the RNA at 12 hours of injection, and then do the RNA seq. And we found identify over 1,000 differential expressed gene is knocked down insect. Then when we check further, and we identify a bunch of immune-related genes be significant downregulated after you knock down these Versicon, P-Versicon genes. Then we wanted to make sure our transcriptional data is reliable. So we selected several genes and to do the PCR. And the results indicating when you knock down the P-Vers, and all these immune-related genes are downregulated, and some the production-related genes also downregulated. So this suggesting that our transcriptional data are reliable. Then we wanted to see if the recombinant Versicon protein can induce antimicrobial peptide expression in the in vivo and in vitro. So we inject the Versicon protein into 18-day lobby, and 30 minutes after injection, we can see a bunch of all these tested antimicrobial peptide will be highly induced. And then we all know in the insects, the antimicrobial peptide is synthesized in the Fat body. So we dissect the Fat body from the lobby, and then in vitro petri dish, we incubate this lobby Fat body with the recombinant protein. And again, they are also able to induce this antimicrobial peptide synthesis. Looks like the efficiency is higher than in vivo. Next, we wanted to know if this highly upregulated transcript be translated into protein, and then they inhibit the bacterial proliferation. So what we did is here and use different dose of the recombinant protein. Then you inject in after 6 hours, you collect the insect, homogenize, and collect the supernet, then incubate with the E. coli. If there's antimicrobial peptide, they should be able to suppress or clean out the bacterial colonies. And as you can see here, and there are those dependently inhibit this E. coli colony proliferation. And we also check which pathway is activated. So most of these genes actually goes through the reddish IMD pathway, a transcript factor reddish, and it is the reddish, pre-reddish, and then when activated, it's going to break down into 2 fragments. And then you can see without a Borsicon, they are small, and with Borsicon 30 minutes, and they activate. And the maximum activation is 1. That's a typical response for hormones. Then we knock down the reddish and up to 70% efficiency. Then we check the antimicrobial peptide. These 2 typical ones. And when you knock down, then you add the recombinant protein in. What happens is this neuropeptide is not able to induce this antimicrobial peptide synthesis. However, there is one unknown gene. We saw this antimicrobial peptide, but when you knock down the reddish, the Borsicon could still induce their expression. So that's suggesting either this one is not an antimicrobial peptide, or they are regulated by a different pathway rather than a reddish or IMD pathway. So to test that, we check the sequence alignment with the NCBI database. We found that these unknown peptide sequence are very similar to a tennyson 3 peptide identified in another beetle transcript genomes and with no known function. Then we use the software to predict potential function, and we found that this peptide, the unknown peptide, with a 99% of confidence, is a kind of antimicrobial peptide. So then we test their functions. Okay. So we initially did the developmental study. This peptide, tennis-like peptide, highly expressed through the developmental state. And this expression pattern is very similar to the Borsicon. And then tissue specific expression also indicated that it's highly expressed in cell body and the hemocyte. These 2 tissues responsible for antimicrobial peptide synthesis in insects. Then what we did is we inject the Borsicon to see if this unknown peptide is indeed, or tendency-like peptide is indeed can be induced. And as you can see, inject the recombinant protein and tendencies will be dramatically increase their expression. And similarly, we did the in vitro dissect the cell body and incubate with recombinant protein, and the tendencies expression also upregulated. Then we're trying to knock down the receptor for the Borsicon, and then the efficiency is about 80%. And then knock down the receptor, and again, these people not able to induce tendency-like peptide expression. So that's suggesting this antimicrobial peptide is under regulation of people. And next question is whether this single peptide has antimicrobial activities. To do that, and and then we incubated this antimicrobial peptide with these 3 microbiomes: gram-positive bacteria, fungi in the middle, and E. coli gram-negative. And as you can see in the bottom, the expression incubation with these gram-positive bacteria is new kinesis like peptide upregulated. Injected with fungi, they are highly expressed. When you inject with E. coli, they do not have a response. Then we inject, we knock down these kinesis like peptide, and then test them with the injection of these three type of microbes. And in the middle, when you inject with fungi, also you knock down these kinesis like peptide, the mortality dramatically increase. So that means this peptide is really responsible and anti-fungi activity. With that, the gram-positive, but no barely any response to very low mortality injected with gram-negative bacteria. And when bacteria recombinant protein of this kinesis like peptide, then they inject incubated with this microbes. As you can see, gram-positive bacteria can inhibit 53%, and fungi up to 80% is already inhibited, but E. coli don't respond. And then we wanted to know which signaling pathway is involved in regulating the kinesis like peptide expression. And knock down this transfer analysis, as you can see on here. The top is Brancobacter incubate, and the bottom one is recombinant protein jack. As you can see, this transcription factor involved in whole pathway. And so if and also two are highly, when you knock down these two, and then the kinesin re-expression is downregulated. So these results suggesting that a kinesin 3 like peptide and peptide is regulated by versicon through the toll pathway by activation of DIF and also 2. And this conclusion is P-verse homodimer acting as a positive regulator of the immune response in tennis, Castanian lobby through the IMD and the toll pathway. The IMD pathway most of the gene, but the kinesin like peptide is through the toll pathway. So P-verse regulator expression of kinesin 3 like peptide, the novel antimicrobial peptide specific for fungi and gram-positive bacteria, and it via the DLGR receptor encoded by RIC, and then transcription factor DIF, and it also in the whole pathway. Last slide. I'm not going to talk about the particular candidate. So our lab is demonstrating last several years that this neuropeptide versicon can regulate all three pathways. Basically, whole pathway and eject cell pathway are also IMD pathway, but we don't know how they regulate, because if it goes through this pathway and the information is just whether they are through TAC1 or something else to regulate all these three pathways. So we are trying to figure it out in the labs. I'd like to thank my collaborator and the people working in my lab for this project.
I think you can once. Maybe you mentioned this. Is 10-3 LP actually
No, we couldn't find it, but it's in the beetles, Canabrian and other beetle species. The genome.
Our next speaker is Zheng Yongze from Taiwan. His talk is about the acid-based homeostasis in cuttlefish.
Okay, first of all, thanks for organizers, especially after Professor Thompson's invitation. It's maybe the first time for me to attend this comparative endocrinology meeting, and today my talk is about fish, but not a real fish. It's a cuttlefish, physiological adaptation, and how this kind of adaptation is related to the young hypophyseal hormones. And you can see that it's the embryos of the cuttlefish, just like their view, but it's super cute. And if we want to talk about the evolutionary adaptation about the cephalopod, we have to get back to the evolution about the cephalopod. Actually, the ancient cephalopod was derived from the very early class of the Ammonidae. You can see that actually we can easily to see the fossil of the Ammonidae near our seashore. Near our seashore. So why did it called Ammonidae? Because you can see the structure of this kind of fossil, and it is very like the ancient god of the Ammon, home-like hair. So we called it Ammonidae. Actually, the Ammonidae first appeared in the late Silumian to early Devonian periods. It's about 400 million years ago. Actually, in the Asian open sea system, Ammonidae were especially abundant, or the dominant species in our open sea, especially in the Mesozoic marine environment. You can see that this is the inspection of the ancient ocean. This kind of mollusk could athletic swimming in the open water system. Later we'll discuss about why they can have such kind of athletic kind of swimming mode. However, nobody knows why they're completely distinct during the Cretaceous period since about 100 million years ago. During the evolution, nowadays, we cannot find the Ammonidae anymore. However, there was still some related species or class near the Ammonidae, just like the Nautilidae, which is the Nautilus. Nowadays, we still talk about, when we discuss about the Nautilidae, this is a living fossil, of course. And another class is the Colloidae. This is the modern cephalopod. Of course, we know that the modern cephalopod is the biggest recent group of cephalopods living in our open water system, including the squid, cuttlefish, the topic of this talk, and octopus. Actually, the living cephalopod belongs to the mollusk. So how those mollusks could derive such kind of complex system and highly evolutionary roles in the phylogeny tree compared to the deuterostrome, the vertebrate that we are. The mollusk, the cephalopod belongs to the proteostome. During the evolution, of course, the early gastropods, just like the picture, there were the head, gills, and body, food, and funnel, and the heavy calcium carbonate shell, of course, on their back. However, during the evolution of pressure towards the shell loss, so the early cephalopod, as I mentioned, is about the distinct species. On their shell, their calcium carbonate have the space for their buoyancy and lost some weight, so they can have a swimming capacity to swim around the open ocean. And of course, the nautilus, we all already know. The loss of some shell space, and they can get a greater buoyancy for their swimming. And now, the squid and octopus of the cuttlefish, they get a greater metabolic pulse and increase the buoyancy, of course, and they can recolonize to the shell water open sea system. Why? Because their shells goes into their mantle. It's not like the early cephalopod anymore. Therefore, the environmental pressure or the physiological adaptation drive some physiological adaptation system or the morphological changes. Not only the adult stages, in the early ontogeny stages, the cephalopod is quite comparatively different to other species. During the development, they are going the direct development without metamorphosis. So while they are in their chorion, you can already see the embryos just like what they are in the adult stages. And they have a yolk sac for the nutrient supply. And outside is an capsule and chorion and the periventrally fluid surrounding the embryo. However, this kind of egg capsule created a diffusion barrier for gases and some ions, just like the oxygen, CO2, and ammonia. So this is a nature high PCO2 and nature high ammonia contents of the PBF. It's for PCO2, it's about 0.2 kilopascal. So this gradient is necessary for the diffusion of the gas across this membrane. And this is very important to create a diffusion gradient from their body to ambient circumstance. Our previous study have already found that if we incubate the cuttlefish larvae into different kind of acidified conditions, from their hatching to 2 months later, if a slightly decrease the pH level by PCO2, they can compensate their metabolic respiration. However, if we increase the PCO2 and down the pH level to pH 7.4, you can see the aerobic respiration was significantly decreased. So there was some metabolic adjustment to maintain the PCO2 levels in their body. Through the ontogeny of their gill function and development, we have already found that around the stage 27, the gills have functionally sodium potassium ATPase in their gill. However, in other stages, which kind of mechanism is responsible for the ion transport or maintain the intact homeostasis? So in the early development stages, as you can see that there was a thin layers of skin surrounding their yolk sac. And we have already found that based on the SEN and immunostaining images, we can find that there's some mesh-like structure surrounding their skin. And this kind of mesh-like structure, they can uptake the sodium ions, which we apply this image by the sodium green, the vital dye. So this mesh-like structure is responsible for the sodium uptake from their skin epithelium. Therefore, about 5 years ago, we construct a model like this. In the early life stage of the cephalopod, not only the cuttlefish, but only in squid, we have already found that compared to the tedious, this kind of mollusk skin, they also have a complex ion regulation machineries surrounding their skin epithelium. We can find that the sodium potassium ATPase and sodium proton exchanger 3, NHE3, surrounding the yolk sac epithelium. So the yolk sac is not only responsible for the nutrient supply, but also their skin is responsible for ion regulation. And at their head and their mental side, they also have some NHE3 and the mitochondria and sodium uptake capacity and the sodium potassium ATPase transporting ability. So we created the conversion occurrence of the ion transporter on the epithelium between the tedious larvae and the cuttlefish embryo. They all at the epithelium, they all have the basolateral sodium potassium ATPase as a primary driving force or responsible for other ions transport, just like the proton excretion, ammonia exclusion, or the calcium uptake. So from the evolution point of view, why the modern cephalopod were highly evolutionary developed? Because since they have to compete with the tedious in the open water system, this kind of conversion evolution features enables them to survive in the modern open ocean. So the next is, since we know that neurohypophysial hormones is a primary molecule to promote the ion and water balance in vertebrate, just like they were using the vasopressin, oxytocin, or in fish they were using ISOCCE. And through the hypothalamus and pituitary system, this kind of hormone controlling acid-base and ion regulation system is quite organized in vertebrate. However, cephalopod have brain too. Of course, we know they have a complex neural systems. Recent 3 years, there were 2 papers published by Australian group. They used the modern MRI technique to know that which kind of cephalopod log region responsible for visualization, sensation, mobilization, and another group also used the micro CT to clarify the ganglion system of the squid. And about 2012 and 2013, there was another group published that in cerebral, they will also have vasopressin and vasopressin-like peptide, just like in cuttlefish, they have sepiaptosin, and another homolog is called sepiaptosin. In octopus, octopressin and sepiaptosin also expressed, and sea urchin and echinopersin and C. elegans, they have the nematodes. But what's the function of this kind of peptide? Do they have the similar function as we observed in vertebrate? Our group also observed that in cuttlefish brain, they also have different kind of laws. 20% is the optical law and use the suspension image confocal, and we can localize in the optical cortical region. There was a complex synaptic system in their optical law, just like we vertebrate processes. But which kind of region is responsible for controlling this kind of neurophysical hormone? Still unknown. So during the early development, we checked the sepiaptosin expression, and using the in-situ hybridization, we can find the signal was obviously found in the optical region. And this responsive receptor STR2, sepiaptosin receptor 2, is also highly expressed in the 28 stages. Levels can be observed in their mental skin, in their hair region, and also in their yolk sac skin. So this area is important for maintaining the intact ionic homeostasis as well. And the receptor we observe is also could be found to co-localize with the NHE3 sodium proton exchanger 3 and sodium potassium ATPase. While we treated the early cuttlefish embryo under the acidified condition, we can find that the sepiaptosin and responsive receptor STR2 was highly expressed under acidified treatment circumstance. So nowadays we know that this kind of hormone is also released or expressed on one of the brain region, the optical lobe. And this kind of system will be also promoted and stimulated by acidified condition. So it is related to the ion regulation as well. But it is the early embryonic stage. How about the adult stages? As I mentioned, the sepiaptosin, this hormone, is observed in the optical region of the embryo. So we got a section and found that the sepiaptosin will co-localize with the neuron which labeled by ZN12, and it is localized in the subcortical region of the optical lobe. Where are the responsive receptor localized? They also expressed in the epithelial cells in the essential ion regulation organ, GIL, as well. So we also do the institutionalization work with the sodium potassium ATPase and NH3. We can find that the signals was co-localized together. And follow up, we want to know whether this kind of expression of the responsive receptor is really could be operated or stimulated by this neural hormone. So using the advantage of the gill structure in cephalopod. Compared to other mollusks, cephalopod's internal organs is a very closed circulation system. It's not an open circulation system at all. The hemolymph is surrounding their internal organs. In cephalopod, they have two hearts, and the branchial heart is connected to the gill. So we use the ex vivo perfusion approaches. And if we added the cephalotocin into the saline and perfused into the gill and take the bath and the perfusate, perfused after the cephalotocin perfusion, how will we have and the consequence after the cephalotocin perfusion? If we perfuse the cephalotocin into the gill, we can find that the 0.1 micromolars of the cephalotocin will significantly stimulate the potent secretion from their gill epithelium. And of course, the ammonia, we found that under the 10 micromolars of cephalotocin perfused, the ammonia was also be excreted by the gill epithelium. So nowadays we know that this neuron hypophysial hormone is all really related to the epithelium potent ammonia excretion from the epithelium. So it is another feature of the convergent evolution about the hypophysial hormone works in invertebrate. So that's my final slide, and there's a conclusion. And for the evolution point of view, as I say, that the cephalopod is a highly evolutionary developed invertebrate. Sometimes we call it a marine primate. They're very smart and they are well organized. So under the CO2 treated pressure, maybe if they want to make the hypocapnia inside their body, they have to maintain the intact ionic homeostasis. Maybe this kind of hypercapnia stress will stimulate the optical lobe or the ionocyte progenitor cells to stimulate the S epithelial ionocyte to trigger the bicarbonate uptake or the proton and ammonia secretion from the gill epithelium. That's why they can maintain the intact homeostasis as we observe in other animals. I'm happy to take any questions. Thank you.
I think we have a time for several questions.
So pardon me, my lack of knowledge on the secretion of the globe. So are there neurohemocytes in the globe that will not secretion of this hormone? It's going to be a hormone, and then also, with the embryos, the yolk sac is pretty substantial. Looking at that, and so you have to have a very large amount of is secreted and goes to that large space, not to be diluted. What are your thoughts?
Oh yeah. Actually, I have a very nice pictures, videos around that. As you can see that this epithelium nearby the real yolk sac, they have already know that their circulation is connected to their real mental bodies and hair. So it's possible that this kind of circulation can promote the controlling system into the yolk sac epithelium. Yes. Yes.
It's really fascinating to see how. So Again, I don't know anything about competition. Are these osmoregulators or these osmoperformers?
Oh yeah.
So can you measure the quality for osmosis?
Yes. I have to say that previous studies always say that this kind of mollusk, especially the marine mollusk, should be osmoconformer. But we found that compared to other mollusks, cephalopods seems to be can slightly control the proton and ammoniums and other ions as well, and slightly adjust their homeostasis. Just I mentioned, since they have to, especially in the early life stages, they have to retain in their chlorine. So the barriers between the ambient circumstance and the PBF they should have a prefab gradient. So the PCO2 or the high ammonia pressure, I think is a kind of driven factor to make this species have such kind of slightly adjustment capacity.
That makes sense. Another naive question. So when you see at acidified conditions, I think the pH 7.8, you can foresee these 8.
Yes.
What's the natural sort of?
Yeah, for the seawater, the
7.8 is quite not.
yeah, but you know that our natural water system is about 8.1. It's a little bit ugly. Yes.
Because cuttlefish also have able to so I just wondered if
Yes, I think it's related to the what epithelium nearby the hotspot. Yeah, sometimes it's responsible for the water or the air flux throughout the cotton epithelium. Actually, some of our previous study have already proved that this kind of epithelium have the sodium potassium ATPase expression. Yeah. Yeah, sure.
It's not also any good questions? Thank you.
我们现在试一下吗？
可以吗？那我现在试一下是吗？试一下我的这个更快。
真的。
有看到吗？
我们试一下。
你们喜欢
那这个能换页吗？
OK 吗？
啊，好的，能换页。
这没什么问题吧？
对，挺好。你是放的播放是不是？
我前面是放的播放，有问题吗？
没有问题，都挺好的，挺好的。
OK。
看一下我的。
啊，等一下，等一下，我把
不想就行是吧？不想。
等一下，我先退出，你稍等一下。 我是不是退出来了
啊，退出来了。
？OK， 好，那其他老师可以看看。
是。啊。
哎，朱勇老师啊，朱勇老师，我是王德寿，王德寿，王德寿啊，我就说那个，因为不是，好像我师兄刚好在那边吧，他这样两边的兼顾一些，我在这边还看不到。有陶亚雄师兄是他同意了，我给他说了，你觉得可以吗？啊？
没有，没
说。要试一下吗？
行，好的，好的。
那我这里我怎么直接共享呢？
我先打这个是吧？啊，
我还没。
还有，然后再共享。我共享一下。
这个呢，在这里看。下午这个。
这样就好了，是吧？
然后上午对。
翻页是可以，是吧？那就可以。
现在是李老师，现在可以共享。哦，现在是李老师在共享吗？
我已经退出来了啊，你共享吗？
来了吗？
我退出来了，退出来了。
哦，我现在试一下啊。现在可以看到，可以看到是吗？哦，现在有点字头，看到吗？啊，看不到吗？
我这儿也看不到，我这儿也看不到。
你也看不到，我们可能要重新来一下。
你把它关了。
是因为我们是刚注册进去，所以看不到。是不是我要用你后来给的那个？哦，把那个，然后那怎么办？要换掉，我们要重启。这个我们退出，重新启一下话。
啊，这个朱老师，我刚才是全图还是放映的模式？你刚才看到的。
你刚才是放映加提示。
对的。
是两个页面在一起。
好，那是对的。不要。
这个好的，这个。
现在是好的吧？
现在不是播放，现在是你的那个正常的 PPT， 你把它放到播放。
前提的，现在不是播放吗？
不是播放。
想播放。因为我是标题，所以我这个可能不一样，是吧？那我从一开始播放。 因为我是双颈看的，那我先说。啊，对对对。
他从这儿下，他那个不对。
嗯。
我在这边看的不是播放。
那现在呢？
没空想。
哦。
哎，这回好了，这回好。
这回好了是吧 ？OK 吗？现在是全屏了吗？
对，挺好的，好，这个好的。
好好，这个别的我就退出了。
现在。结束，结束。
好。还没有结束是吧？
没有，在上面，上面有一个结束。
嗯，啊 ，so OK， 好，我看到了 ，OK， 哪里？啊。哦，现在可以了，好。
他出去了，然后再进来。啊，李老师进来了，进来了。
Good afternoon， 看到吗？那个现在共享的可以吗？
现在对，现在不是播放。
右下角，我们这边播放。哎，你没摁播放，你摁那个播放。
哎，好了，可以，可以，没问题的。
可以是吧？好好好，那我们。
哦，
可以了，可以吗？
可以，可以，挺好的。
在。他的意思是说，陶老师，我跟陶亚雄师兄说，他在现场嘛，那么这一块主持的就是没有关系的。
对，要在现场的主持比较好一点。
因为现场的他能够兼顾两边，是吧？两边一兼顾的话，你在下面你又看不到那边，我在这个
上面没法搞提问，都没法搞的。
提问我们听着提问好。
完全听不见，因为他离那个话筒好像比较远。
要求他们到上面来提问吧，你要求他们就在你这个位置里面对，我陶师兄，哎，陶师兄在你旁边。啊，请陶师兄辛苦你了啊，有劳你。对，你在这样兼顾两边的话，这样子的话，你提问啊，这个最好邀请他们上台上来提问，这样大家能听得见那个做报告的老师们听得清楚楚。谢谢啊。好，林老师能看见，到时候。
Okay, maybe let's get started. So this session is supposed to be co-chaired by Dr. Yan Wei from UCLA and this one from Southwest University. But they decided they don't want to do it, so I'm gonna introduce all 4 talks. Okay. So the first one is by Dr. Liu Weijuan from Shanghai Ocean University. He was in charge of the first symposium. We appreciate that. And today he's going to talk about thermal perception in olive founder. The floor is yours, Weijuan.
Thank you, Professor Tao. Good afternoon, everyone. First, I would like to thank the organizing committee inviting me here to give this talk. Today, I'm going to talk about the thermal perception and thermoregulatory battery of CSS in Hollywood flounder. Fish are more vulnerable to the environment temperature change. They cannot, like humans, keep a certain temperature for their bodies. They are follow the environment temperature goes down and goes down, and follow the environment temperature goes up and goes up. So when the temperature changes to a certain level, the body will trigger the stress response. One of the stress response adaptive pathway is HPI X in fish. We generally finally produce the cortisol. Let's look at this figure for the fish cortisol production here. And we generally have the CH and neurotensin from acceptance, and they project the extron to the pituitary and stimulate the cell produce ACTH. ACTH enter the bloodstream, acting on the internal, stimulate produce cortisol. In fish, there is another system which is similar to the hypothalamus pituitary system, which is located under the spinal cord. We also have CH neurotensin neuron here, and the actually secretion there under the bloodstream and directly acting on internal system. And also can assist in the ACTH to acting on the internal system to stimulate cortisol action. Here is the special system in fish, and they generally located at the last 7 terminal vertebrae. And in there is a large secretion neuron secretion cells there. We call the Dogen cell. They have a long action and terminate in the urophysis. In the urophysis, the secretion product like CIH, U1, U2 storage and secretion there. We did measure the expressions and also hormone levels for the fish to localize the seasonal variation of this system. You can see the figure here. We have seasonal during a year, different seasonal plasma cortisol level and plasma neurotensin level showing seasonal change. Similarly, we have the expression inside CSF also show the seasonal patterns as well. But interestingly, this expression seasonal pattern will have the similarities with the Dogen cells number pattern as well. So it means during the seasonal change, they are actually not just change expression, they also change the Dogen cell number as well. So we break down the seasonal signal. There will be light, temperature, food difference, reproduction variations there. So in here, we look at the temperature. We want to know what are these response for this CIS and the temperature change. So this is a design of the experiment. We generally keep fish at 18 degrees for adaptation, and then we go for adaptive level. Within adaptive level, we choose 2 temperatures. This is one is a lower temperature, one is a higher temperature. So we measure the plasma hormone level. measure CSS expression level. We also look at the different urotensin 1, urotensin 2, new staining cells numbers. We also use the BRTU injection to measure any new cell generations. So this is the results we look at here. This is expression is a plasma level of cortisol immediately increased after 12 hours stress. No matter is hot and cold temperature stress. And we also have the CIH showing similar patterns, but we didn't see significant difference for urotensin 2 at 2 hour time. But if we look down, we can see urotensin 2 expression in CSS significantly upregulated in lower temperature at the challenge. And we also see lower temperature at 2 hours in CIH's expression level. But we couldn't find a significant difference in urotensin 1 levels. But interestingly, when the fish adopt at 8 days for the cold weather and the hot weather, and they actually have a significant increase the expression level for urotensin 1. So next, we want to know whether this organ cell follows expression and secretion change. They also change their patterns. So this is the staining of immunostaining for urotensin 1, urotensin 2. We actually couldn't see much difference when 2 hours until 2 days. There is no difference. But we can see at the lower, higher temperature, at 8 days, we can see the upregulator of the cell numbers for urotensin 1, but it's not for urotensin 2. Urotensin 2 slightly goes down in the cell numbers, but it's not significant. Then we use the BRDU injections. We want to know whether this increased number of this cell actually due to new cell generation. But unfortunately, both lower and higher temperature, we do not see a significant increase in BRDU labeled urotensin 1, urotensin 2 cells. So that means urotensin 1 positive cell increased urotensin 1 positive might be generated from the original organ cell, which is expressing both urotensin 1 and urotensin 2. But they are not from the new stem cell to differentiate to the urotensin 1 cell. The next question we want to know is what happens for the organ cells. We know the organ cell in there and expression changes, secretion difference. So we want to see, we have established long time there is ex vivo recording for the CSS. We choose the lower temperature in degrees, so we choose a higher temperature, 26 and 30 degrees for our test here. So generally we have the organ cell, this is the firing patterns there. Generally we have 4 firing patterns. One is the pattern is silent cell without any firing activity there. And we have boosting regularly firing patterns. We have weekly firing pattern like physical. We have constant firing pattern on electronic one. So as we have looked what's happened when the temperature change. So we can see this in B is 14 degrees, so we can compare with the original one is 20 degrees. We compare it, we can see that reduced temperature actually inhibit the firing activities and reduce the firing frequencies in our fish CSS for the organ cells. But if we increase the temperature to 26, we can significantly increase the firing activity and the firing frequencies for these organ cells. But when we increase to the 30 degrees, we can see lots originally highly firing activity like physical and atomic firing activities reduce, become bursting activity. And originally, quite silent cell is the beginning to show a pattern to physics and atomic patterns there. So it means that for 30 degree is different with 26 degree, they are changing the pattern. So in here, should have function change as well. Now we want to know what exactly happening in there when the temperature change. What is the sensor for this thermal sensor to sense how this temperature change? So generally we know it's a sensing temperature change all normally through these trip channels. Then we use the temperature change plus the trip channel and mechanism. Then we can actually measure whether which trip channel actually involved in this system. Generally, we know normally when we have heating low temperature, normally the trip A channel involved. So we try the number trip A channel. There is a trip A1 channel showing a significant reverse the inhibition of the temperature, 14 degree inhibition. So it means triple A1 actually involved in lower temperature to control the low temperature inhibition the firing activity of the organ cell in CSS in our fish. Now we did a PCR, see is any difference with increase to 20 degrees, and we can see there is directly inhibition for the 20 degree upregulated firing activities. Now we try to look at the 30 degree. We can see 30 degree there is another differential patterns showing again originally high stress pattern reduce original quite one is increase. But when we add this triple V1 channel, we show is reverse effect, very clearly reverse effect. And it means at 30 degrees still is triple V1 channel in working condition. Now we want to know whether this actually specifically is for the triple V1 channel. We use the triple V1 channels specific agonists, then different concentration like 1 micromolar and 10 micromolar, 100 micromolar to pass on this organ cells. We can see 1 and 10 showing dose-dependent upregulated firing activity firing frequency there. But for the 100 micromolar, showing slightly inhibition there, and reduced compared with 10 micromolar. So this is a much similar results comparison with our data showing when the temperature inhibition there. Now we want to know whether this TRPV4 channel also involved there. And we had at the 16 degrees, we had this antagonist in there. We can see antagonists can inhibit the firing activity for this 16 degrees. It means TRPV4 channel actually involves the regulation at 16 degrees. Same things at 20 degrees also showing this. This means 16 degree, 20 degree, they all involve for this TRPV4 channel. When we try to 26, can you hear me? You can see in here show is quite increase. Okay, fine. So I continue. But TRPV4 antagonists will not have any effect on this increased firing frequency. So it means after 26 degrees at TRPV4, it means TRPV4 is in the middle, and TRPV1 working on the lower temperature, TRPV1 working on higher temperature. So next we want to know what's the thermoregulatory factors. This channel actually direct operating on the carbon cells response, and so we still use the basic firing activity and at lower temperature TRPV1 system. Also, we carry out one simple evaluation for the TRPV1 channel to look at whether you are treating organ cell. Unfortunately, organ cell showing up any TRPV1 activities. But at the upper spinal cord, 8 to 20, find a number of cells which positive TRPV1 channel. So it means TRPV1 is not directly regulating organ cells. They use the indirect measurements. Now, to confirm this information, we call the transspinal cord CSS, and we find that they actually can, during the reduced temperature, they actually inhibit the fire activity organ cells. Now we count the spinal cord from the last seven spinal cord, separate 8 to 10 with 1 to 7. So after cutting with the spinal cord, we find that low temperature no longer have an effect on the organ cell firing. And also other than antagonists of the TRPV1 as well. So this means this is a confirmation for the regulation of organ cell is come from the 8 to 10 spinal cords, the TRPV1 positive cell. So what's actually the control when it's sensing the temperature, lower temperature change, what they actually control this organ cell fire? That is another thing to look at. So we use a number of the inhibitory transmitters. We find the GABA immunoactivity in these cells. 8 to 10 spinal cord. We also see the extron which towards organ cells also contain GABA. So we know the low temperature expression of the GABA receptors also upregulate. So that means that we look at the upregulation cells use TRPV1 sensing the lower temperature and secretion this GABA to the organ cells and inhibited organ cells for the fire. So here is another way look at to conform this. We use the GABA directly to add on the spinal cord to look at the fire activity of the GABA and inhibit the fire activity. To further conform this low temperature nutrition using the GABA, we use the 14-degree inhibiting the fire activities. Then we add the GABA antagonists. So we can see GABA antagonists actually increase the fire of this 14 degrees organ cells. So it means actually the lower temperature inhibiting was sensing by upstream the cells actually could use secretion this GABA neurotransmitter acting on the organ cell, which inhibiting the fire. So to summarize this, actually find the CIH, U1, U2 expression neuron in CSF, and also an increase in the one positive cell is due to the cell which express no can stem cell. The CSS can directly feel the temperature changes, and the TRPV1 working on the lower temperature, TRPV1 working on higher temperature, TRPV4 sensing of senses. And this work is supported by Shanghai University and also the National Science Foundation and also Ministry of Science and Technologies for the funding. And also this work was done by graduate students and also master students. Thanks everyone for your listening.
Okay, questions for Professor Peter? Anybody?
Professor Lee, very great, excellent talk. I just probably did not catch your talk. Is TRP expressed in the SNCC system?
Sorry.
Is the TRP expressed TRP A V they are good expressed in the coder system?
PTHRP or DRP?
Yeah, TRP. Yeah, TRP A and TRP B.
TRPA, yes. TRPA, yes.
TRPA and also V, right?
TRPA is not expressed in organ cell.
Okay.
It's expressed in upstream the cell which our action project to the organ cell.
Okay. Thank you. Thank you. Congratulations.
Thank you for your talk. I was wondering, there are some data showing that some TRPV sense also pH changes. So do you know the CNSS could respond to pH variation through these receptors?
I think CSS involved in there is research in Canada. They actually working on this, showing they actually involved in regulation pH change, acidification change. But we didn't carry out this specific work in our lab. And yeah, I hope next time I can answer your question properly.
Thank you. And I have a second one. Did you by any chance save the brain in your in vivo experiments to know if you have also changes in TRP AV expression in the brain during temperature challenges?
Okay, we'll move on to the next talk. So the next one is by my younger sister, Li Wensen from Shenzhen University. We're both trained by Professor Hao Ling. So today she's going to talk about metabolism and innate immunity in zebrafish during the competitive growth. Wensen, the floor is yours.
Hello, can you hear me?
contribution to organize the meeting, and thank you for your invitation. I'm originally from Sun Yat-sen University. The title of my presentation is Metabolism and Innate Immunity of Zebrafish During Compensatory Growth. Compensatory growth is a period of accelerated somatic growth with a steeper growth trajectory following the elevation of growth stunting condition. Compared with the control group shown here, that after the starvation following with the refeeding, we can have 4 kinds of outcomes: no compensation and partial compensation, full compensation, and overcompensation. And the term CG was coined in 1955 after its first seminally documented by Osborn and Bell in 1916. And now, CG has been observed in all vertebrate classes. For example, human and other mammals like beef cattle, finishing pig, and cattle and sheep. Other vertebrates like birds, reptiles, and amphibians. Now, CG has been documented most extensively in fish. In this table, there are collected about 18 species of fish, different types with different type of restriction and different type of time of refeeding. Most of them can achieve partial or full compensation. In the compensatory growth, after the dietary restriction and follow the refeeding and the compensatory response. During this process, what we most concerned is about the recovery of energy reserves and the increase of growth rate, especially in farm animals. As we know, feed is a major economic and ecological burden of fish farming. To reduce the feed quantity without loss of production may help achieve green, environmentally friendly, and sustainable development in agriculture. As we show in the table, there are a wide variety of restriction in feeding protocols in fish. However, in order to further promote this approach to the industrialization of applications, some issues must be addressed first. For example, the underlying mechanisms governing the response, involving metabolic, oxidative, and immune changes, and the practical scheme in CG. So in our project, we focus on the metabolism and the oxidation and the immune. First of all, we established our compensatory growth model in zebrafish. As we show here, we can see that the CG will happen after 1 week fasting or 2 weeks and 3 weeks. The CG happened in zebrafish. And we also found that the longer the fasting time, the longer it will take to recover. And during this process, we also check some hepatic enzyme expression level. And first is about in the period of food deprivation. We can see here that the time duration of fasting, for example, fasting for 1 week or 2 weeks and 3 weeks, it shows that the duration of the fasting time has little effect on the change of the metabolism. This are glucose metabolism, and these are the lipid metabolism. And we also found that the fasting for 3 weeks shows a rather stable trend. So in the subsequent study, we will choose fasting for 3 weeks in the experiment. And in the refeeding refeeding period, we use the fasting 3 weeks and then refeed 1 day, 3 days, and 7 days for the refeeding. And here we can see that in the refeeding period, the enzymes show a significantly change compared with the fasting group. So we can say that fully restricting fish show a higher weight gain ratio upon refeeding, and refeeding will result in a capital McRae, and also it will have a moderate increase in hepatocytes. And the PCNA, we show here that the PCNA significant increase. And in the liver TG concentration decreased after 3-day refect, but back to the normal level after 10-day refect. However, for the liver glycogen, it increased after 3-day refect, but back to the normal level after 10-day refect. Therefore, a detailed understanding about the enzyme change during this process, we apply the peptide transcriptome during the CG, and here we got about 4,000 unique differentially expressed genes, and geographically clustered into 4 groups, and we focus on the cluster 1 with about 1,800 unique genes, most eating 3 days after 3 weeks fasting, and also focus on the cluster 3, about 1,200 unique genes, focusing on the fasting and the downregulating in the first 3 days. And this data indicate that the fasting and the refeed 3-day stages are the stages where the internal environment undergoes drastic changes. And while the refeed 7-day and 15-day are stages where the body adapts slowly, so it change not very significant. And the signal pathway with the significant changes in the refeed 3-day phase include the DNA replication, and TCA cycle or static phosphorylation in the glycolysis, gluconeogenesis, fatty acid metabolism, and etc. We also carry out the validation of the transcriptome gene expression data, and most of the person correlation were greater than 0.7. And then we also have carried out the liver metabolites, and here we see that we got a 95% confidence, and in the B, we can see the 88 detective metabolites here, and most abundant is about the amino acids and followed by the fatty acid. With the disease or analysis show here that in the refeed 3-day metabolites among different abundance of metabolites, amino acid were the most significant, and also in the refeed 10 days, uric acid as well as amino acid are the most abundant. With the scatter diagram, we compare the refeed 1 day versus to control group, and compare the refeed 10-day versus to the control. And the data show here is that metabolites change in refeed 10-day are significantly reduced versus to refeed 3-day, indicating that the body's metabolism has gradually returned to the stability. And box plots diagram also show us the similar trend. For the summary, brief summary here, that zebrafish could reach full satiety after 3 weeks of fasting and refeed for 15 days. The liver display hyperplasia accompanied with increased TG but increased glycogen contents after refeeding for 3 days. And the metabolome data imply the participation of amino acid metabolism during compensatory growth in zebrafish liver. And when we fed 3 days, the liver TCA and oxidative phosphorylation show signs of improvement in temporal transcription analysis. So for the next step, we'll focus on oxidative phosphorylation. And in this part, we first detect the oxygen consumption. We found that the oxygen consumption was not significantly different. However, ATP production will increase about 130% from the refed zebrafish about 3 days refed. And also the cytosolic ATP levels were also higher in the refed zebrafish. And then we go to check the reactive oxygen species ROS accumulation in the refed zebrafish liver. We found that ROS increased in refed 3 days, but it will back to the control level at the refed 7 days. And similar format was observed in the MDA change. However, no effect of protein carbonylation we can see here. But the proteasome activity was activated, including the tumor tracing-like and the caspase-like proteasome. Then we use tempo and like a natural oxide that usually be used to eliminate the oxidative stress. Tempo treatment can show, so here will decrease the CG that on the body weight here. But tempo did not influence ROS and also did not decrease the ATP. However, it increased protein carbonylation. And the tempo treatment will block the ROS signaling in zebrafish. You can see that some mRNA transcripts targeting the NIF2 were all significantly increased. And NIF2 is nuclear factor. So it gave us a clue that ROS may regulate compensatory growth through the NIF2. Both AKT and mTOR signaling pathway were upregulated, and the tempo treatment inhibit the induction of mTOR, but have no effect on AKT. ROS accumulated in the liver due to the improvement of mitochondrial function during the compensatory growth of zebrafish. And it transmitted signals through the NIF2 and mTOR signaling pathway. ROS can retard the compensatory growth of zebrafish. So we conclude that mitochondrial ROS signaling are essential for the occurrence of compensatory growth in zebrafish. Therefore, about the immune response to the fasting and feeding during CG. Here we show that the zebrafish in the fasting group had an increased mortality rate compared with the control group. In the figure we show here is the survival rate, so a little difference here. And with the extension of refeeding, the mortality rate continued to decrease. And then here we first use the HEE to show the kidney morphology change during fasting and refeeding. We see that the number of melanoma-morphology centers, MMCS, in the kidney increased after starvation and reduced by refeeding. And HAVC1 expression increased after fasting It means that starvation may cause injury in the kidney and rescue by the refeeding. And further, we detect some oxidative, some factors related to the oxidative. First, NDA enhanced significantly after fasting, but decreased to the normal level after refeeding. ROS decreased with 24 days of starvation, but did not recover to the normal level after refeeding. That's a little bit different with the NDA. And nitric oxide increased after fasting for 21 days. Also, the activity of antioxidant enzymes like SOD, GSHPX, and CAT increased after fasting and recovered after effect for 3 days. Oxidative stress in the zebrafish kidney during fasting and feeding. We also detect the NDA. Sorry, is it? Yeah, the activity of AKP increased in the kidney after fasting and decreased after refeeding. Here we showed with different refeeding duration, refeeding and 3 days and refeeding for 7 days here, and it showed a similar pattern here. And also the respiratory burst response in kidney showed no significant change in different groups. The response was higher in the starvation and refeeding group, just so in this. We also detect some mRNA expression level about some genes related to the immune system. For example, the IL-1 beta, NF-kappa B1, NF-kappa B2, ARG2, and TGF-beta 1A and 1B. We can see a similar trend that the starvation will increase it and then decrease by the refeeding. Similar summary, brief summary here about the innate immune system. Starvation triggered zebrafish suspectly to the pathogen-like streptococcus during compensatory growth. And morphological changes of kidney appear in the process of compensatory growth. An oxidative stress in kidney was induced by starvation and reverse following refeeding. Some parameters of innate immunity are changed in the kidney after starvation. For the ongoing job, the first question we want to answer is that does NRF2 participate in the stress and innate immune activities during CG, and how is the mechanism and how they achieve this? We can see here when we use the WB, we can see that after starvation for 24 days, the NRF2 diminish very significantly. But after 3 days refect, it will increase compared with the staff. And also we detect the GSTP1 is the downstream gene regulated by NRF2. We can see that when staff it decreased and after effect will increase. And then we construct the transgene zebrafish with the CRISPR-Cas9 phenotype NRF2 in zebrafish. And with this model, we will go on for our next study. So here, summarization. As the first step to promote CG apply in fish farming, we have carried out some basic study about metabolic, oxidative, and immune changes during CG. Also, we did not know the mechanisms under. And for the long-term goal, we want to apply CG solution to effectively reduce feed costs in fish farming, and ultimately succeed in achieving the green environmentally friendly and sustainable development agriculture. And for acknowledgement, that thank you, Dr. Zhong Zhen Liao and Dr. Ji Longjia. They finished all the studies in the talk, and also thank you for some financial support. Thank you for all the attention. That's all.
Okay, we got quite a few minutes for questions.
Great talk, Vincent. I'm just wondering, have you also looked at housing density effect on your recovery, your growth, consistent growth, as well as changes in the immune system?
Sorry, can you please repeat again?
Oh, I'm just wondering whether you also have considered or have looked at the stocking density effect on compensatory growth, as well as on changes in the innate immunity.
Oh, no, we did not detect it. Now, maybe for the next study, we will detect it. Thank you for thank you, John.
Any other question?
Thank you. Very good talk. I have two questions. Are you talking about the kidney? So are you doing the head kidney? Is that right? That's the first question. Second is for the application for the aquaculture. You also mentioned during starvation, they might it's become easy to get disease sustainable. So how is the benefit to do the kind of certain period of starvation and then go back to feeding again? What the phenomena behind this? Okay.
Okay, thank you for your questions, Professor John. Can you hear me?
Yes, yes.
Yeah, yeah. You said the kidney. It is interesting. Maybe we are now focusing on the kidney. You see, just now I say something about that when the starvation, the kidney has been have had some injury, but when we feeding, it will be cured. So we don't know what's the mechanism under this process. Maybe next step we will do this. And the second question is about what's the benefit, right? Yeah.
Yeah.
I think. Yeah. I think first, if we can save some feed, that is a good thing. And for the next, maybe it will be good for the immunity, maybe we can diminish some cost in the drug or something during the agriculture.
Okay.
Yeah.
So first one I did not catch it. So you talking about kidney is the head of is the anterior kidney? You more focused around there, or kidney is very long. So
but in zebrafish is not very long.
Okay. Thank you. No. Thank you.
Thank you.
Thank you.
Any other question? Thank you, Winston. Hope to see you soon. Okay, let's move. Let's move on to the next talk by Dr. Zhao Shuangxia. She's affiliated with the 9th Hospital of Shanghai Jiaotong University. They are a referral center for studying rare genetic diseases in humans. So Shuaxia is a physician scientist, a really good place to visit whenever you stop by flying through Shanghai airport. You can try to visit the lab. So she will talk about HGF MET mutations and the thyroid problems.
Hi, good afternoon. Can you hear me?
Yes, yes.
Okay. Did you see the full screen of my PowerPoint?
Yes, yes. You can
Okay.
go ahead.
Good afternoon, Professor Lunju, Professor Qi Gongchang. Thanks for your invitations and the organizations for this excellent conference. And many thanks for the introduction of Chairman Professor Yang Chunhao. My name is Shuaxia Zhao. It's my great honor to have this opportunity to speak the presentation entitled The Deficiency of HGF MET Pathway with two-sided genetic by pattern-based cycle extension. Our research group focus on the pathogenesis of thyroid disease. Thyroid disease including congenital hyperthyroidism, autoimmune thyroid disease, and the thyroid cancer and nodules. As you know, congenital hyperthyroidism is the most common neonatal metabolic disorder in the world. Estimated to occur in approximately 1 in 2080 live births in China. Unless treated in the first few months of life, severe congenital hyperthyroidism can lead to growth retardation and permanent intellectual disability. Newborn screening for congenital hyperthyroidism has been conducted in China from some states. Data from newborn screening have enabled us to investigate the pathogenesis of this disorder. Most cases of congenital hyperthyroidism can be classified as thyroid genesis or thyroid homogenesis. Thyroid genesis accounts for 80 to 85% of patients with primary congenital hyperthyroidism and is a consequence of abnormal thyroid gland overgenesis, ranging from the lack of a thyroid gland, which causes estrogen, to a hyperplastic or ectopic gland. As you know, thyroid morphogenesis, including specification, boosting, extension of thyroid primordial, regulation of precursor cells, follicular genesis, and the complete differentiation and organogenesis. The expression of NGS 2.14B1 taxate and SSDS determines the specification of a group of cells from the ventral endodome at E8.5 to 9.5 in mouse and at J10 post-fertilization in humans. When the thyroid gland reaches that initial position at 14.5 to 15, it is expressed in primary thyroid. However, expression of this receptor and TT is not activated until follicular genesis begins at E16 in mice. At E16.5 in mice and day 17 post-fertilization in humans, the thyroid gland is completely formed and SLC 585 expression takes place. Synthesis of thyroid hormone T3 and T4 is initiated when the thyroid gland is completely functional. As for thyroid hormone synthesis, we call the thyroid hormone genesis. As for thyroid hormone synthesis, there's a lot of genes account for this process, such as UO2, UO2A, EP, gas reduction, etc., etc. And for the genetic hormone genetics, which is caused by defects inside the hormone biosynthesis, accounts for approximately 10 to 15% of primary congenital hyperthyroidism. In recent years, the zebra-fish has become a powerful model not only for the developmental biological studies, but also for large-scale genetic analysis and drug screening. Most thanks to the easy with which its embryos can be manipulated and its translucent body, which allows in vivo emission. This picture illustrates the advantages of the zebrafish as a model system of forward genetic and diverse genetic approaches to studying development and indeed in zebrafish. In our previous study, we investigated the causative genes of congenital hyperthyroidism by analyzing 21 potential cancer genes. These 21 cancer genes, including the genes associated with thyroid hormone synthesis, thyroid development, and thyroid hormone transport. The exons and the activation boundaries of these genes were analyzed by next-generation sequencing, examining the sequence spectrum in 110 Chinese patients with permanent congenital hyperthyroidism. In our study, we found that 57 patients, about 52%, carried by allelic mutations in 16 involved in thyroid hormone synthesis. Also, we used the reverse genetic testing in congenital hyperthyroidism patients, and we found that for percentage CDC1 varies from this repeated. In zebrafish embryos, CDC1 knockdown caused the defective thyroid primordial genetics and hyperthyroidism. The thyroid cells were stuck together and CO2 dissociated from each other to form individual follicles in CDC1 deficient embryos. CDC1 promoted the beta-catenin translocation into the cytosol and suppressed the formation of cell adhesion complexes. We used the same strategy and found that 3 mastermind-like family genes vary in congenital hyperthyroidism and revealed that both canonical and non-canonical not signaling pathways affect the thyroid hormone biosynthesis. Since less than 10% of cases with thyroid genetics can be identified by non-cancer genes related to the thyroid development, the underlying molecular mechanism for the thyroid genetics remains that unknown. So in this study, we focused on this molecular mechanism. As we know, the endowed zebrafish thyroid consists of individual follicles of variable shape and dynamics lying between the first gill arch and the bulbous anthrosis. Although morphological is different, the development process of the thyroid gland is convergent, and zebrafish thyroid gland also undergoes classification, fooding, migration, and photogenetics. So, to discover genes essential for thyroid morphogenesis and development, we conducted an in-vivo modified genetic screen in zebrafish using TC and TSH. In situ expression testing as straight up. A total of 1,606 active families was screened with embryo, among which 112 X2 mutant families with normal development stages, except for thyroid dysfunction, were identified. Through a formal genetic screen-based ENU for the regulatory of cell development, we observed three families named 018O16, 0122, 000036, and 034O60 with similar dot-like dot-like morphology, which were named as dot mutant families. We performed the whole active sequencing in the tissues and controls to identify the molecular impairments responsible for the dot phenotype in the above families by self-segregation analysis combined with fine positional cloning. The linkage analysis indicates that the potential potential gene was probably localized on chromosome 4 in 18O16 family and on chromosome 25 in the other family. After filtering out DDSNP database and control sequencing data in 15 zebrafish families without scientific genetics, 55 homozygous variants in 16 genes on chromosome 4 were identified from the affected embryo samples from of the 018O16 family, and the active family were localized in chromosome 25 medicine. Finally, by positional cloning, mutation in SGSR8X NAT R284N and NAT E270K were identified from this three family mutants. We also verified this mutation find primers in the embryos of the mutant particular. An interesting coincidence was that NAT is a effect of SGF encoding by SGS-R15. The total length of SGF is 712 amino acids, and the CK8X mutation in SGS-R from SGS-R family is predicted to produce a truncated form Latin homocyst. Latin homocyst also means to further confirm the deficiency of the SGS-R or MAP led to the phenotype of dog-like styles in zebrafish. We injected embryos with SGS-R morpholinus or MAP morpholinus. We found both of them could phenocopy the dog-like style phenotype in 5 days post-fertilization embryo by which with a season curve. To clarify how SGS sequence pathway is involved inside home genetics, we examine the expression of SGS-R and MAP during 7 stages of thyroid development. In our studies, we found at 48 hours post-fertilization, the thyroid primordial transformed here, and SGS-R was expressed adjacent to the thyroid gut. As the thyroid developed, the expression of SGS-R was decreased because it remained low at 54 hours post-fertilization and was difficult to detect at 62 hours post-fertilization by which. To confirm the SGF-R and MAP expression pattern, we searched a capital zebrafish thyroid cell sequencing database and found that SGF-R was expressed inside the stone cells, and MAP was only expressed in some of the thyroid cells. Also, we can see the TI-284 and the TC-270 70 was localized in segment domain. This two mutants did not affect the location of mass and memory. Phosphorylation of the mass receptor as Y164/5 and Y1349 were mildly diminished in the mass to 221 k-newtons and were significantly damaged in the mass L 288 n-newtons. This mutant was conserved in humans. Injection of the constant TTV mass and characteristic as well one cell state significantly rescue the scientific genetic of embryos as well as phase post-fertilization in the dot mutants of the bigger fish mass I284N family. In the XTFR mutant family, injection of XTF protein at 48 hours post-fertilization yield gas significantly rescued the abnormal cytomorphology in embryos as post-fertilization of the dot mutant. We know TTCO and NEAT are specifically expressed in maturated status follicular. The sheath of the stem tissue in the homozygous of embryos at 5 days post-fertilization were also like resolved in HDSR K8X and the MAD I284N family, marked by typical and unique codes. We mark the maturity status follicular stage T4 immunofluorescence at 5 post-fertilization and 10 7 days post-fertilization. And from that, the numbers and the total volume of mature folliculars in homozygous embryos of these two mutant families or citrate when compared with the embryos. Notably, the concentration of T3 and T4 in the tissues of the endowed zebrafish with homozygous mutation was lower than those in their WT control. The number of the viral follicles was lower in the 1.5 months post-fertilization zebrafish than in the WT tissue of the MAD mutant family. The distribution of the size of follicular in mutants in the MAD 200, I200, and the 810 family was also different from the WT tissue. Also, the size of the primordium in the homozygous embryos of the morphine fused to viscerally extend along the laryngeal midline to the late stage outside the development in the embryos from 56 to 72 hours post-fertilization. We know the initial cells immediate activity by SCL and the MAD pathways were mediated by multiple parallel downstream branches of the MAD pathways. 3 downstream switching branches of MAD, including STAT3 here and STAT3 here and NADK so it has been recorded. So in the developmental side of morphogenetic, the group switching pathway plays a role. Therefore, we treated the sequence inhibition of this signaling pathway. We found that inhibition of ERK signaling with MAD inhibits U0126 induce a nearly perfect signal part of the oxide in this post-fertilization embryos. Also, restrict the overexpression of continuously activated mast cells in thyroid, restore the cytogenesis of the 5 days post-fertilizing homologous mutation embryos in this mutant family. Previous studies report that SGF induces the cell spreading and disruption of cell adhesions through the mass energetic snail integration pathway to promote the scattering of NDCP cells. Interestingly, the scattering and migration abilities were more active in the cancer cell line CPC1 after treatment with human SGF. Moreover, the effect of SGF on the scattering and the migration of CPC1 cells was abolished by knockdown mass expression or treatment with mass inhibitors. Snail, although known it can having transcriptional repression, is upregulated by SGF mass switching pathway. Directly by activating by MHC. Consistently, we found that SGF induced the increase in snail and decrease of ectoterin was restored in CPC1 cells after mass knockdown or treatment with the mass inhibitor. Compared to white-type embryos, ectoterin was domestically increased in thyroid of homozygous embryos in SGF-ISOCCE mutant, and the effect of SGF mutation on ectoterin expression was found the primordial food reactive by injection of human SGF in the homozygous embryos. Similarly, increased ectoterin in thyroid of mass I284N mutants could be decreased through injecting the zebrafish particle. Yeah. Or to activate the snail. Okay. The last column. Pathway. Ectoterin inside the cells was increased when embryos were treated with mass inhibitors U0126. Scientists connected as fast-based post-fertilization embryos of the SGF as K80X morphine was partially restored by injection of positive snail plasmid or treatment with the attenuated cytopenia. Also, the bifurcated index of the primordial was dramatically lower at E11.5 in mass conditional knockout in thyroid tissues of mice when compared with WT with First, activation in the thyrocyte at the free edge was significantly stronger than those in the cell at the middle. This is in the middle, this is at the free edge. At the middle of thyroid, while phosphorylated Earth intensity in thyrocyte at the free edge was free. That is that in the middle cell of the thyroid primordial in mass secure mass. This finding indicates that Earth activation in the leaf cells of the classical migration has been impeded in the thyroid primordium of mass secure mass. Moreover, thyroid function of secure mice was components that are shown by elevated scarcity levels. Yeah. In summary, we found that SGF mass is indispensable is indispensable in governing the elongation of the size of primordium along the brachial vessels during cellular development, mediated by some regulation into hidden expression in cytokines while identifying neural pathways. I would like to thank Professor Michael Song and our science team. Okay, I finished. Thanks for your attention.
Thank you, Dr. for your wonderful talk. Any questions from the audience? So, Professor Chen Xue Yan from NIH.
Okay.
Thank you. Thank you very much for telling us the result of your beautiful work. It's very comprehensive study. So I have a question about your dog family. You identified MET and an HEFA mutation, and then you discover that there is some morphogenesis defect. My question to you is, you also observed a decrease in T3, T4, and a reduction in the follicles. My question to you is that have you observed any growth defect? Because T3, T4 are important for the growth.
I cannot hear the question very clearly. Adam?
Okay, my question to you, in your dog family, you found that dysgenesis, that means morphological defect. And my question to you is whether you observe other phenotypes such as the reduced growth in your fish. 因为你的那个你找那个 dog family， 那你说有 MET 跟 HGFA mutation， 你又发觉那个 T3、T4 都降低了，对，然后那个 thyroid follicle 也减少。我要问你，就是说，因为 T3、T4 跟成长有关系，所以我问你，你有没有看到，你的 fish 是不是比较小，还是 compare with the wild type？ 你的鱼有没有比较小？
是的 ，OK， 整体的好像没有太大差别。
所以你看到的只是那个 thyroid 的样子不一样，但是呢，你没有看到功能的退化，对不对？
Another question? So I have a question for you. Even now you go back to the human genome sequencing database, for example, the UK Biobank, right? Did you find mutations in these genes in humans?
You mean we cannot find any mutation in HF or MET in humans?
Yes, yes.
And yes, we cannot find it. Also, we did the detect the UK biobank, but we have almost 1,000 computer factor mutations. We cannot find any mutation in this gene.
Oh, really? So that means they're really essential, right? What about your TSH receptor mutations? Do they also have thyroid dysgenesis?
Yes, we patients with receptive mutations. Yes, we have some continue to have the patients have receptive mutation. What's the question?
Those mutation, the patients with those mutations also have not just thyroid hormone production problem, but also thyroid dysgenesis, morphologically different. Can you image them? Image the thyroid gland. Yeah.
Okay,
Okay.
Thank you. Professor Tom. I cannot hear that.
Any question for you? Thyroid. Thank you, Dr. Zhao, for your wonderful talk. So the final talk for this session is from Dr. Zhihua Gao, Zhejiang University in the beautiful city of Hangzhou. I really like the title, The Beautiful Neuroendocrine System. I'm sure quite a few of us will agree with that, right?
Yeah.
The floor is yours, Dr. Gao.
Thank you very much. Is this okay? Great. So, and my screen shows up pretty well. My voice is okay, and the picture is fine, right?
So you're not showing the
I'm no. I'm showing it in full screen. Can you see it?
No, for us.
Okay, so let me adjust this a little bit. So, yeah, I already did it.
It's not here. It's not there.
Okay, stop stirring. Okay, and then okay. So is this okay? What about now? Maybe okay. This. Does it show up now? Just go. So I am doing it in full screen now, but it doesn't really convert, right? So, but then can you do, okay, just 1 second, let me try another way. What about now? Okay, So, sorry for the trouble. Good afternoon. Many thanks to Professor Zhang for organizing this meeting and Professor Zhu for the invitation. It's really a pity that I couldn't go to the meeting. I look forward all in the future in person. So I'm trying to move my slide. Please let me know whether it works. Does it work? Is it going? Okay. System in the red with a focus on the microcellular oxytocin neurons. The neuroendocrine system comprises the hypothalamus pituitary, which is like a small spoon located at the bottom of the hypothalamus. This small spoon has very powerful function that helps to maintain the homeostasis of the whole body. And we know that there are actually two types of neuroendocrine cells. One is the powerful cellular neuroendocrine cells. The cells are pretty small with a diameter between 10 to 20 micron. They project to the medial eminence. Over there, they release the hormone release, or the releasing hormone to regulate the function of anterior pituitary. And there's another type of neuroendocrine cells that are much bigger with a diameter between 25 to 10 micron. They directly project to the posterior pituitary. Over there, they release hormones to regulate the kidney and uterus function. And these cells are the major focus of my talk today. So those microneuroendocrine cells constitute the hypothalamic hypophyseal system, along with their soma and long projections to the posterior pituitary. While oxytocin is primarily produced by microneuronal cells located in the hypothalamus, primarily within the periventricular nucleus and the supraoptic nucleus. Oxytocin is released from the posterior pituitary. Over there, they reach to the uterus and the mammary glands to regulate parturition and lactation. As I mentioned, so oxytocin, when released into the bloodstream, they mainly regulate the baby delivery and breastfeeding. That is the role of peripheral oxytocin. However, recent years have revealed diverse roles of central oxytocin in the brain, and the central action of oxytocin mainly regulates parental behavior, affiliative behavior, collaboration, as well as it also attenuates the stress and promotes the positive emotions. However, it's really interesting is that oxytocin neurons are very limited within the hypothalamus. And when these neurons project to the posterior pituitary, release hormone into the peripheral circulation, those peripheral oxytocin barely passes through the blood-brain barrier to re-enter the brain. So, however, the receptors for oxytocin are widely expressed in the brain from the olfactory bulb to different parts of brain and as well as to the hindbrain. So then the question is, if peripheral oxytocin barely passes through the blood-brain barrier enter the brain, then what is the source of oxytocin in the brain? Regarding this question, there are some proposals. One of them is the dendritic release of oxytocin from the magnocellular neuroendocrine cells, as shown here. However, as shown in this cartoon, hypothalamus is only a very small part of the brain, and dendritically released oxytocin from the local dendrites by diffusion to distal brain region may be very limited. So then recent studies have showed that oxytocin neurons can also project to multiple brain regions from the forebrain to different parts of that, including the hippocampus as well as cortex. That suggests that there may be targeted axonal release of oxytocin in the brain. However, there are two types of oxytocin neurons within the paraventricular nucleus. One type is the marginal oxytocin neuron that project to the posterior pituitary, as I just mentioned, and those are the endocrine neurons. The other type of oxytocin neurons are smaller, and they are powerful oxytocin cells that mainly project to the spinal cord and the brainstem. And those cells are non-endocrine neurons. So then the question is, which type of oxytocin cells contribute to the central actions of oxytocin in the brain? If there is, if magno oxytocin cells contribute to the central action, then we may be able to see and coordinate peripheral and the central actions. And these cells are likely project, send their main axon to the posterior pituitary, as well as send the collaterals to different parts of the brain. On the other hand, powerful central cells may also send their projections to different parts of the brain, such as nuclear accumbens and amygdala. In this case, there is very likely that there is no coordination of peripheral and the central actions. So to address this question, whether microneural cells laterally project to different parts of the brain, we did this experiment. So previously, most studies have directly injected the virus into the hypothalamus. By doing this, they can label both powerful and the micro cells. However, in our method, we directly injected the retrograde tracer into the posterior pituitary. With this way, we'll particularly label neurons specifically project to the posterior pituitary. And by tracing their fibers within the brain, we will be able to identify that those are indeed the collateral projections of macrocellular neurons rather than different neurons that project to different regions. So by doing this, my student accurately inject the virus directly into the posterior pituitary. As shown here, this virus can retrogradely along the neural stem back to the hypothalamus. As shown here, we were able to label neurons within the supraoptic nuclei and paraventricular nuclei, as well as other areas that also project to the hypothalamus. And this figure just shows the distribution pattern of retrogradely traced cells within different layers of hypothalamus. By staining this retrogradely traced cells with vasopressin and oxytocin, we verified that this retrogradely traced cells indeed contain vasopressin and oxytocin. As well, we carried out we also verified that using this retrograde tracing method from the posterior pituitary, we particularly label microcellular but not hypercellular neuroendocrine cells, as shown here by the thyrotropin releasing hormone labeling, which is a marker of hypercellular neuroendocrine cells projecting to the media elements. So we then made a thousand of slices throughout the whole right brain using the micro-optic sectioning tomography by collaborating with Dr. Hui Gong's group. And this method is very high resolution view of single cell, single labeled cells. As shown here, so this is a three-dimensional reconstruction of the hypothalamus neurohypophysis system. So this area, all the traced cells are located within the hypothalamus in different nuclei, and by doing maximum projection, we were able to and then we were able to fully quantify those projections towards the posterior pituitary. We found that paraventricular nuclei and supraoptic nuclei only contribute about 50% of this total input. Other accessory areas and additional areas that have been unnoticed will also alternatively contribute to the peripheral effects of vasopressin and oxytocin by projecting to the posterior pituitary. And this is a three-dimensional maximum projection of the hypothalamus neurohypophysis system. As shown here, so different nuclei are all stacked together, and at the bottom, that's the posterior pituitary, and all the cells, they send their long axons, goes bottom to the posterior pituitary. And here, that's a second projection for this, that you will be able to appreciate that our neuroendocrine system is such nicely organized and beautifully organized with a heart at the center of the system, and a butterfly extending their wings towards both sides of the hypothalamus. And along with that, we were able to see that in the brain, there are also tiny processes extending out of the hypothalamus. And this long process project to amygdala, cortex putamen, and the different parts of cortex. So this is likely the collateral projections of the macrocellular neuroendocrine cells. We further verified this by single-cell tracing, as shown here. A typical representative neuroendocrine cells with its one axon projecting to the medial eminence towards the posterior pituitary, and the other end goes to the nuclear accumbens. And there are other different neurons that also showing collateral projections to different parts of the brain. And this video gives an overview showing the projections of macroneuroendocrine cells within the brain from paraventricular nuclei to the from paraventricular nucleus and supraoptic nucleus to different parts of the brain. And we further verified this using dual retrograde tracing strategy by injecting the green fluorescence virus from the posterior pituitary and a arbitrary tracer from different parts of the brain. So with this, if the cells collaterally project to posterior pituitary or in the brain, then we will be able to trace cells as a yellow cells that have both the red and the green signals. And indeed, we trace cells using this strategy. We then also use this similar strategy to selectively label oxytocin cells. So, for example, we made a oxytocin pre-reg by injecting the Cree-dependent retrograde tracer directly into the posterior pituitary. We were able to label specifically oxytocin, oxytocin neurons, and we were also we could also see their fibers within different parts of the brain that suggest their collateral projections. We then use chemogenetic tools by injecting a HM3DQ virus into the posterior pituitary and label the oxytocin neurons by injecting the ligand of the receptor CNO. We were able to activate these neurons as shown here by the C-force labeling. Activation of these neurons leads to the elevation of oxytocin in the bloodstream, as so here as well. Conversely, if we inject the DI virus into the cells using the same strategy, we were able to inhibit oxytocin neurons as shown here by electrophysiology analysis. And accordingly, the level of oxytocin is reduced within the blood. So using this method, we found activation of oxytocin neurons only promoted social interactions. For example, if we use this three-chamber assay, so normally rats would prefer to interact with the side containing their conspecifics. By activating the oxytocin neurons, we could observe increased interactions towards the concept specifics, as shown by this projection and quantitative analysis. So activation of micro-oxytocin cells not only elevated peripheral oxytocin levels, but also promoted social investigation. And conversely, if we inhibit micro-oxytocin cells, we would see that the original preference towards their conspecifics were suppressed. So this suggests that, if we further activate oxytocin neurons by inhibiting their terminal projection within the cortex putamen area, if we further activate micro oxytocin neurons, we will see that they actually promoted locomotion. And this promoted locomotion promotion is mainly through oxytocin release within the cortex putamen, as we could block the movement by locally administering oxytocin antagonists. So together, I've shown you the three-dimensional reconstruction of the whole hypothalamus neural hypothesis system, and we found that these neurons not only project to the posterior pituitary, but also extend their collaterals to different parts of the brain, and one of them is to promote locomotion and promote social interactions. So with this, we think oxytocin neurons, they are like an orchestrator, and they orchestrates both peripheral and central actions through their collateral axons. And with this, I'd like to thank my collaborators and my group members and all the people that helped us on the way to characterize the three-dimensional structure of this beautiful system. And thank you very much for your attention.
Okay, thank you for your nice talk. Live one button. Any questions? Hi. Dr. Pei Sansai from University of Colorado.
Thank you. So is there any way that you think that the activation of the oxytocin neurons can be dissociated, because you use chemogenetics, which has a very profound effect on the neuronal activation. So if you use optogenetics, high or low frequency, whether or not you can disentangle the secretion versus the neural hormone effect versus the neurotransmitter effect.
Yeah, that's a very good question. So we have, easy to say, but how to do? We've tried to manipulate these neurons with different frequency using the optic stimulation. But sometimes we think we can probably only see the central action. We don't really, without affecting the peripheral hormone levels. However, I feel it's not that reliable, because the ELISA analysis of oxytocin in the bloodstream is a bit tricky. We have to extract the samples and reach the measurement, and sometimes even by doing that, we were not very sure whether it is so accurate. So we got to find a good way to measure the oxytocin level in the blood. On the other hand, we are looking for new tools by using oxytocin sensors within the brain to particularly track the release of oxytocin as a neuropeptide in the brain. But very much thank you for your question. If you have any suggestions regarding the protocol, I would really appreciate that.
I think people are getting tired. It's a long day. Right? Peter, is a long day. So how do you measure the blood oxytocin level?
So we use ELISA from Enzo, but then because oxytocin is bound to other proteins, we have to extract it with the particularly C18 column to get rid of the contaminants. Then we will be able to measure the level of oxytocin. Even by doing this, I still think the method needs more improvement. Maybe SBAC measurement would be better.
And those are the binding protein. Can you separate the vasopressin and the oxytocin?
There are different ways to measure vasopressin. So for measurement of vasopressin and oxytocin from the blood, there are ELISA kits. So in our system, so for the first part, we only we directly inject the virus into the posterior without any specific cream. That way, we are labeling the whole magnocellular cells. Later on, when we manipulate the cells, we use particularly oxytocin rights. That way, we only selectively manipulate oxytocin neuron without affecting vasopressin because we check the vasopressin level at the same time.
Okay, thank you. I think that's all for the session. Thank you all for thanks to all the speakers for this afternoon session.
Thank you.
画面关直播。
还有存档吧，设定应该都有存档。